All right, we're on bottom. So I guess we're just
Let's find a
gonna
rock.
head up. Yeah.
Find a rock. Yep, just find a rock. Try that one right there.
Yeah, that one you mean?
I think they're all gonna be like pick something up.
That little overhang.
Yeah. Want to push on these things? You find any rocks, banter? Sure.
So can you just do like a welcome ocean research vessel found for ROV Sebastian intro just so it's in the beginning? And a short degree.
Yeah, welcome to dive 430 on Sebastian on RV Falkor. Sorry, we're on our RV Falkor with ROV Sebastian at 1,800 meters on Titov Seamount. We are gonna work our way up today to about 1,100 meters, and yeah. We're gonna start off by trying to break off a rock.
Dropped out. Like flicker, then it just won't black. It'll signal. Alright, man. See if moving any of the not taking anything, some sort of this one.
So, pick a rock.
Anything significant?
Yeah, you can go ahead and just kind of poke this one.
That's huge, bro.
It's a huge one.
Okay, then we can come up here and see if you could break off a piece of that ledge. It's definitely.
Break some off. You get a big piece.
Yeah, so we need a decent sized chunk.
I got faith in that.
I mean, I don't think that's gonna just fracture the crust. I don't think.
Alright.
Fly up.
Sure. Before we do, Derek, do you want to try and see if two is back?
Yeah, sure.
We have another. This has come up a couple times since the attempt. Did we manage to capture the sea pen the other day that is known to be elusive? And yes, Russ, the ROV pilot, used a push core to push down into the sediment and he transferred the sediment with what turned out to be with the sea pen, at least half of it, into the quiver. So it did work. It was successful, at least half successful, which is more than we can say we have been so far for that particular sea pen.
Try one of these. The big one right here in the middle.
Sure.
So we just got on bottom. We haven't seen. There was one little fish when we first came down. We haven't seen any corals. And first thing we're gonna do while we're at our deepest depth is try and find a rock for mineral because we're interested in the ferromanganese crust and what the thickness is, what the chemical composition is, and kind of helping understand the geology and the oceanography of the area. We'll see if any of these are loose. I'm going to be a little impressed if they are, looking at how well they are coated and cemented in. I'm Brian Kennedy. I'm a PhD candidate at Boston University.
And I'm Alexis Weinig. I'm a research scientist with the U.S. Geological Survey. That looks like it.
Yep, that one looks
Is-
loose.
Can we get just a picture of it real quick before we pick it up? Yeah, there you go. You got it. Little tube worm tube on there.
Right, got it.
That's good.
Okay.
Thank you.
Mm-hmm.
If we can bring it just up to the camera and rotate it, that would be. There went a chunk of it.
Let me bring some more lights on and turn the lights off.
Okay.
Okay.
We can just keep rotating it, kind of 360. It's very rusty on the bottom.
Yeah.
Okay. That's great. Thanks. We can put that in the rock box.
It's gonna be sample 397.
Yeah.
Oh.
Coming in with the torch man.
Would you say that was like roundish?
I call it roughly football shaped
It's good.
with a couple chunks of crust missing.
Alright, I'm gonna fly up back. It might be easier to use the starboard arm with the little red cushiony things.
So the reddish brown color you're seeing on the rock is actually more of the natural color of the rock, and the super black stuff is the ferromanganese crust that grows on the rock over time. So you see the crust on the top where it's exposed to the water, but you get a more true coloration of the rock on the underside where it's been protected from being precipitated on by the sediment.
We'll let that sediment settle away a little bit before we go back in.
Do you want to film again, Alexis, before we pick it up?
Yes, I'll take another zoom just in case the rock has changed, but I'll let it settle out a minute before I try to do that. It's about 10 centimeters. Okay, doke. Okay, that's good.
Fish photo ball line.
Did I miss it? Oh, I feel like I must have been looking at the chat.
Okay.
I think we got the imagery we need.
Yeah, I think we're good.
No.
Nice.
Got the rock.
Rock.
Now back to regular scheduled biology.
Rock acquired. We did our job for the geologist.
Yep.
Now we can move on. So Tim won the sample competition last night.
What was the sample?
So,
What was the sample competition?
It was just we all betted how many samples we thought we would be at by the end of sampling after all the associates were accounted for
On the last one.
on the last dive, and Tim
Somehow
Tim
he controls the
won.
number of associates taken, won the
And the
competition.
master sheet and the.
No, the master.
No, I
Alexis
control the
controls
master sheet.
But still.
it was close. Our numbers were 2 numbers apart, and he barely squeaked in.
Dopey eyes should
He
not be
came in
included
screeching
any
with the ee to get that last little sample from a rock.
Does he win here?
Huh?
Does he win prizes?
Yes, but Randy doesn't know that he won yet, so he has not received his prize yet.
So we're coming up on a stalked crinoid here.
Yes.
future, we have a pale blue matte background. Can we not use the pale blue track line?
I didn't pick the line color. Would you like me to change the line color?
Yeah, we make it a little easier on me.
Wait.
Oh my gosh,
No.
that is so I could not see that. That
It's
is-
like green.
Yeah, okay.
That line is
Can I drive for a second?
is kind of hard to see. I couldn't even see that until Chris just said that.
It's a green line. But
Mine is blue. This is
this is
pale
blue.
blue.
I don't even know how you determine the color of the line. I know how to change the color of the waypoint.
Or if we can make the line like thicker.
So we're diving on Titov Seamount, which is
I have to
a
do
geode or
extra
flat top
because
seamount
you have to
in
make
kind of the
control.
southeastern corner of the Howland and Baker unit of the Pacific and Rhode Islands Marine National Monument. It's our second dive on the feature. We were here yesterday on the kind of southwest side. Now we're kind of on the northeast side, covering a similar depth range. So we're going to come up pretty much straight up this fairly steep slope here for the first few hundred meters of the dive and then trek along the ridge, hopefully to make the edge of the top of the flat part of the feature, which is where on the west side, previous dives from the Okeanos Explorer found a really diverse and abundant deep sea coral community. Got a fish coming up here, looks like probably a halosaur.
Yep. Wow, our first waypoint is 400 meters in elevation.
Straight up.
Okay.
Straight up like some 40, 50 degree slopes.
This is our second dive at Titov Seamount. We dove here yesterday on, were we on the southeast side?
Southwest.
Southwest side. Lots of halosaurs.
I'll just do it as not. It's more just complaining to complain.
Yeah, thanks. Like over 2 waypoints. You make them red.
This is a long track. It's 2,000 meters too.
Okay.
So
Alright.
we'll need
Could take a look at what that white dot is, Chris.
Where's that?
On
Oh,
the
right.
yeah.
my bet is Titan.
Oh, good guess.
You're fine. Looking at the cameras. Bring down the lasso.
Yep, it is a chitin.
It's a chitin or polyplacophorus. It's a type of mollusk. It's really ancient lineage.
That's good. We can keep
Are you
going.
happy?
Yeah. They have
them.
Nah, we'll leave them alone. They have a really muscular foot that's on the underside of the animal that's latching on to the rock. You can find chitins all the way down in the deep sea, though they're not super abundant in these areas. You can find them up in intertidal zones, so if you go tide pooling, you'll find chitins clinging onto rocks up in that area too, so they're really widely distributed. Looks like a crinoid.
I'm gonna change that one. Sorry, I made the waypoint.
That's alright.
darker.
I'll just go really fast so I get to a darker area in the map.
Do you want me to put another waypoint in here?
No, you're fine.
Here, I'll send this over there.
Got a unstocked crinoid hanging out on a rock. Oh, we got a pycnogonid. Can we take a look at the sea spider?
Yep. Right.
So these sea spiders are coral predators, corallivores. So this expedition is paying special attention to what eats corals and sponges and trying to get a better sense of how that plays in a deep sea ecology. So seeing these coral predators both on and feeding and not feeding is important to get a sense of how much time they spend actually eating coral versus other organisms.
We've seen some interesting behavior from pycnogonids. We saw one a couple dives ago feeding on a holothurian or a sea cucumber, which was really unusual to see. They're also known to feed off of anemones. I think we've seen that a couple times on this cruise.
And these aren't actually spiders. We call them sea spiders, kind of colloquial, but they're not related to spiders very closely at all.
Yeah,
and
they're not
they're-
actually arachnids.
Yeah, and they're super strange that they have no respiratory or circulatory systems. So they don't have gills, they don't have lungs or anything like that. They literally just diffuse gas through all of their skin, more or less. So that's why you see they're so thin and long, as they have to have a high surface area to volume ratio to be able to do gas exchange without any kind of actual circulatory or respiratory system.
It has something in its little paws. I don't know what you call
Yeah,
them.
I'm trying to remember that is. Yeah. Yeah, I agree. It does appear to be holding something.
But I don't see the proboscis out, which is when you find them feeding on anemones or gelatinous organisms, they have a pretty large proboscis that they will insert into their prey, and kind of works like a straw. They just
Suck the life out of it.
suck up the nutrients.
Starship Troopers.
So 10 centimeters, he's maybe about full length across, 20, 25 centimeters.
So medium-sized one.
Yeah.
Look at the botryoidal texture on this rock.
Yeah.
So this kind of small,
I think
lumpy,
that's good,
cobble
Chris.
appearance
You
of
can
the
keep
rock
going.
is indicative of heavy ferromanganese encrusting, or incrustation, maybe? Is that right? Incrustation?
I don't know if that's a
Encrusting?
word.
I don't either. You have to excuse us, it's 4:30 a.m. or 5:30 a.m. on the ship. Yep. It's my story and I'm sticking to it, Deb.
Yeah, so I wouldn't,
There's our first coral.
oh yeah, there's,
Good stuff.
in the distance here, looks like we have a coral right over here. I'm not 100% sure, but I don't think pycnogonids have eyes. Do you know?
I don't know, actually.
But I don't believe that they do, or if they do, they're very rudimentary, so they're really just light sensing organs. So they don't actually see, they just sense their environment through other senses.
Spider.
Yeah.
When we were messing around next to one the other day, it didn't appear so.
Right, right.
So at this depth, there's no sunlight penetrating this far down, but there is a decent amount of bioluminescent organisms. So a lot of organisms down here do have some rudimentary light sensing, but it's looking for bioluminescence, not necessarily light from the surface, like we use, or most shallow water organisms use.
Looks like we have our first coral of the dive.
Bless you.
Do either of you want anything from the mess?
I'm okay, thank you.
Found the Seamount.
I missed the HG2, yes, pretty much.
The what?
HG2, the one that looks down.
Oh, yes.
I missed it. It's not there now.
It looks like we have some egg masses on this coral. Looks like a plexaurid, and 2 egg masses, with an astroschema, so a brittle star in the center. Saw a lot of Plexarids yesterday. It's hard to Plexaridae is a family of corals, but it's hard to give a more detailed description of them. It's hard to get down to genus without having the sample in hand, so we generally just say Plexarid, but the same type of brittle star is often found on this family of corals. And I don't know what those eggs would be, but they could possibly be from a fish or from a gastropod. We see different types of eggs laid on corals of different, kind of all over the place. I think that's good.
Right. I'm gonna go up a little bit more on that campaign so I can see how this. Brighten that up a little bit. Maybe do a different light as well. I'm gonna go all lights around.
None of the contents.
Don't do that. Is the upper light on? Try inner. See what inner does. That's simple.
Some crinoids. Yeah, that's good.
We have to put HB2 somewhere so we can see if it's working. So let's put it 0, 0, 0, 0. Let's do
It's
that.
interesting, those like slabs of
Yeah,
rock that have come down.
got a little coral
Looks
here.
like a little. I'm gonna snap zoom, Chris, on this little.
Yep.
Yep. You know what I'm doing.
Then on the
Okay. Looks like a bamboo coral. So probably. I don't know. I'll just say unbranched bamboo. That's okay. That's good. I can keep going. There's a question, what does the ivory part mean in corallivory or spongivory? And does it mean creatures that eat corals and sponges? And that's exactly what it means. It means we're seeing a coral or a sponge being preyed upon by some other type of organism. So what we're seeing, we know that sea stars can prey on both corals and sponges. There's evidence that aplacophorans, so mollusks with like a worm-like body plan, can prey on corals. The pycnogonids, like we said before, the sea spiders. We don't often see eating coral directly, but I think there have been some sightings of that, but we see them more often eating larger gelatinous animals like the anemones. I'm gonna zoom in right in the middle there, Chris. Looks like we have some zoanthids growing on a dead sponge. And then there's a little live sponge behind it. Little ball. We've got a predatory tunicate. See the
Yeah, it's the stalk that actually caught my attention more. That stalk is thick and fleshy as opposed to the thinner, more fibrous ones I'm used to seeing with the predatory tunicates.
To the one left.
yeah.
Alright.
Pretty big, 10 centimeters across.
Yeah.
12.
Alright.
That's good. Yep. We also see snails or gastropods that will feed on corals and sponges. What else? Any other groups?
It's all the ones I can think of that actively feed, but we also, are beginning to, notice some other associates that here cause through abrasion, cause damage to the tissue as well.
Yeah, so we often see the
and the corallive jellyfish.
The little
Forgot the corallive
jellyfish.
There is a little jellyfish that we believe is eating coral polyps, but we're pretty sure it's a new species, and we've only collected it a couple times in 2017, and so we've been actively looking for it this expedition and haven't found any yet.
urchins can also
Yeah, that's true.
eat corals, but the brittle stars that we often see on the corals, so the stars with the really long arms that are wrapped around the coral colony, there's no evidence that they are eating the coral itself. It seems that they're eating the same food source that the coral is eating, and that's determined through stable isotopes. So you can run analysis to try and determine which trophic level, so which level in the food chain something is eating at, and what the analysis that have been done with ophoroids and corals is that the ophoroids are eating the same level of food, so the detritus in the water column that the corals are eating. So it's likely that they are using that coral to get up into the water column to be able to feed, or possibly even kind of picking food off of the coral polyps.
Thanks, Mike. Tell Casey I said hi too.
Tell Casey I said hi too.
Little Casey.
Hello to the Okeanos from the Falkor. The NOAA ship Okeanos Explorer is also in U.S. waters, but on the Atlantic side. They are diving today as well, so you've got multiple channels of deep sea TV to tune into. So it should be a fun day in both oceans.
This is always one of those fun things, telepresence moments, where we're here, literally almost on the other side of the world, around Howland and Baker, and we've been texting our friends on Okeanos pretty much continuously as they've gotten ready to start their field season, and they're shaking down Deep Discover and getting ready to explore. What are they doing this year? They're in more South Atlantic? Or no,
it's
they're
the
doing corner
canyons.
rise seamounts. That's right.
Yeah,
They're going up
seamounts.
the New England seamounts and corner rise.
But I think right now they're on the
They're just outside
shakedown.
of
Yeah,
Norfolk.
yeah.
Somebody said they were watching them this morning.
Yeah. So I think they're live. They're
Yeah,
doing
they're
a
live.
shakedown, but still broadcasting the dive. So I think they are in the southeast right now.
Yeah, they were in and out of Norfolk, I believe.
Okay. There's a snailfish. No,
You probably know if it did. Cuskeel.
cuskeel. Oh yeah, that's a good time to mention. What is that website called?
Ocean Education.
The new website that's been launched as a partnership between NOAA, Ocean Exploration, Nautilus Live, and Schmidt Ocean Institute is called deepoceaneducation.org, and it is a joint effort to have information about where each ship is and if they are diving. So it's a nice central location to be able to check out kind of 3 of the more telepresence, the ships that do more telepresence during their dives. So that are streaming live, that have scientists on board and also at shore that are contributing information to what's happening in a dive. Over here, Chris, looks like we have a coral. Called the Deep Ocean Education Project.
Ben, how's your cooking?
Wonderful. Go on.
No, I think we got my appreciate it.
No, I think what?
You knew I was gonna say that, but
There's a sea star down there too. I don't believe the Nautilus has started their season yet this year, so you've got 2 out of 3.
Smells good, so.
that's disappointing, sorry.
That's okay.
Yeah, look at that. That's cool.
That is cool. It's nice, like shot of each ship.
When we use the Nautilus, Tom, you just go live.
I think we're still moving a little bit. And that the Deep Ocean Education website launched really recently, so I'm sure they will continue developing that website to have more and more features as time goes on. I think that has been a request from the community for quite a while to have kind of one space to go to be able to know who's in the water or where in the world the different ships are. So it's a nice resource for ships that are not necessarily U.S. based, but more operated around U.S. time.
Ship watching a ship.
So right on.
Ship watching a ship.
What's this cable? Is this not showing you? Are you on it?
This looks like a hemicorallium, so a type of corallid precious coral. Has another brittle star. We often also shorthand call these OFs because it stands for Ophiroid, which is the group of one of the groups that these brittle stars belong to. Last night I was watching and somebody in the chat was curious why they kept hearing the word oaf. So it's just shorthand for Ophiroid, which also stands for brittle star. And it doesn't look like there's much going on here. Even so, sometimes there are small polychaetes that will burrow into the skeleton of hemicorallium. It doesn't seem to be a parasitic interaction. It seems to be kind of commensalism where both are benefiting, but this it creates a much more fragile skeleton, and this one doesn't seem to have those holes or burrows inside the skeleton. It can be hard to tell on the seafloor, but when you bring the coral to the surface, it's quite evident. All the polyps are retracted in. And we have a little overturned sea star, but we'll take a look at the underside. So that actually can be,
Poor guy.
it can be quite helpful to look at this side for diagnostic reasons.
Nothing there on me. Stealthy. Ninja Russ.
So don't have coke in the game. So don't have coke in the game, so don't have the cage.
And this sea star will eventually right itself, so they have the 5 arms that are flexible, and they have all those little tube feet. You can see them starting to move on the arm on the left. So it'll flip itself over and continue on its merry way once we leave it alone.
Did they flip themselves over?
Yeah. Yeah, I was worried there. That's good. We can
picked
keep
it off the
going.
rock the other day. I was like, let's put it back where we found it.
Someone asked what color would the coral and brittle star be. They are that pink color and that orange color for the brittle star. We have white light down here with us, so we're seeing them as if they were on the surface. How deep are they? And does it say? No. Oh, look, right off Philadelphia. Hey.
Is that Elvis?
Okeanos.
Oh, okay.
Did this EDP funding for the US Science fencing.
Oh, that's amazing.
Cool.
They're gonna go that way, man, like,
Saw that. We saw that.
you know how the jar trees? Yes, it looks like there's a coral nut up in the back left.
Oh. Looks like a little sponge right there.
Little sponge. Long spine urchins sitting on top of the rock.
Yeah, we see a lot of those.
Yeah, they've been quite
Kind of
prolific.
across all the different sites. I feel like we
Right.
see them at least at some point during a dive.
It's like another tunicate.
Some of that encrusting sponge, maybe. Oh no, it's just like a low-lying sponge.
Threw up Google Earth, Alexis.
Oh, okay. It's on the YouTube. Well, that made me sound old on the YouTube.
You are as well on the
Deb has Google Earth up to kind of geo-reference everyone to where we are in the world. So we are in U.S. waters, but we are far in the Central Pacific. So if we zoom in to where we are, we're down here in the Howland and Baker U.S. EEZ or U.S. Protected area.
Pacific Rhode Islands Marine National Monument.
Part of the Pacific Remote Islands Marine National Monument. It's my have quite a hard time remembering that one.
PB&J. No judgment.
Yeah, here's a good little coral to look at. But we are on Titov Seamount, which is at the southern border of this protected area. and we dove on it yesterday, and the Okeanos Explorer dove on Titov Seamount in 2017. So we've had a couple opportunities to survey different parts of this seamount that has a summit of around 1,150 meters. It looks like a Victor Gorgia, but it definitely has some tissue damage, which is kind of surprising for this group of corals. So this Victor Gorgia, this family of corals, at least as far as here in this area of the Pacific, they have this very bright purple color. And yesterday we collected one, and when we were preserving it, the coral color is just completely dissolving onto the gloves that we were wearing, which isn't that pigmentation doesn't normally rub off as you're sampling the coral. So I think it's just a very thick pigmentation. Has an astroschema brittle star and that crinoid hanging on. That's good, guys. We can carry on.
So marine protected areas come in a whole range of different flavors, from kind of special management areas with light protection all the way up to full no-take protected areas. So the marine monuments, like the one we're operating in the U.S., are highly protected areas where very little or no extractive activity is permitted at all. There's something we haven't seen.
Oh, a small little Iridogorgia, it looks like. So very.
So when you're trying to kind of assess the level of protections within a monument, it's really you have to look at the rules and regulations of each individual monument, or MPA specifically, to kind of gauge how strong the protections are in that area.
I don't know if I lost that little coral somewhere. They're very faint sometimes, and they don't have large branches or bright colors. I know, me too.
Yeah, I totally can't see it.
It was a small Iridogorgia. I think
Yeah.
it was a very young Iridogorgia, so
Agreed.
type of Chrysogorgia. Well, we can keep going. We'll hopefully see others
Yeah.
as.
It's raining,
Oh
it's
my
raining,
gosh.
it's lightning and-
Yeah.
I'll come in here and you'll see.
Okay. Yeah, the original way. Yeah, there it is.
Yeah, it's just
Wow.
camouflage.
It's like optical illusion.
That's kind of sweet. I'm gonna try that.
Okay.
Put that a little bit. That's just nice.
Has some tissue damage towards the top.
Yeah, it doesn't look like it's doing so well.
Yeah, not sure if that one will get to be like one of those big
Can we
meter long.
Can we take a look at what are these little
I think these
nubbins
are
are?
xenos.
Somewhere we did it before, actually.
Trying to do it on pilot one because it's the same as the background. But I couldn't figure it out. I definitely points to change head blue. And I didn't figure out how to change the body.
I think these are xenophyophores, so single-celled organisms that create these larger structures. They can have multiple nuclei. Like that one in the back definitely is.
Yeah,
I think all
I
those
think you're probably right.
little sand look like they're made out of sand or out of the sediment. They agglutinate all that sediment around them and create a structure. That's what that looks like to me. Oh, there was a chrysogorgid. Some of them really do blend in with the.
There's a big slime star ahead of us.
Yeah. Big pink hymenaster, which we saw a couple times yesterday. They always look very like pillows to me, like cushions, but when you bring them up to the surface, they're actually very rigid. They're pretty hard. And these ones have the common name of slime star because they create a lot of slime if they are disturbed, which we're not looking to deal with. Oh, we have a big fish right up there. Just kind of chilling there. Any idea what that is?
Not one I'm willing to put out there yet.
Okay. We'll get some images and then just carry on. We do not have a fish biologist with us. So while we can at least ID most corals down to family, we aren't as good at IDing fish, especially at different sizes. Sometimes there are certain fish that we see pretty commonly, like rat tails and halosaurs or cusk eels, but there can be slight differences to fish, especially in the head morphology or the shapes of the fins. So I try not to give too many names to fishes since I'm probably wrong every time. This is a nice little ridge pavement feature. Oh, good morning, Adam. Good, how are you? No.
We got a short nap.
Had a nap. Little sponge here. Take a quick snap zoom on the way.
Yeah, I'm really not sure what that fish was. It had a dorsal fin consistent with a rat tail, but the shape of the caudal fin and the break between
Yeah, that really
the
tall.
secondary dorsal and the caudal fin, most of the rat tails don't have that. It might have been some type of hake, but that one we're gonna have to send to Bruce Monday.
All right, we can keep going. It's not a chimera.
No, definitely not a chimera.
No. That is one that I know.
Yeah. Best guess is a hake of some type, but this is deep for those. No, definitely, it wasn't definitely not a cusk. It had the segmented multiple dorsal fins instead of one continuous long one.
I didn't see it. So.
I'll look up a little bit. It's like an Anthomastus mushroom coral.
I'm not used to seeing xenophyrophores on rocks at this kind of density like this.
There's a lot of them, you're right.
So today's dive covers basically the same depth range as yesterday's dive. So this will give us an idea. We really, no one's fully characterized a single seamount. So we really are still kind of guessing at the differences on the different faces have, the different currents, and how the different sides of seamounts may have different biological communities. And so this seamount will have at least 3 dive tracks through a similar depth range between the 2017 Okeanos work and our 2 dives here, that will hopefully get a little bit better sense of how the community composition on changes or doesn't change,
Oh, there's a
depending
little
on
jelly right here. Can we zoom in on that?
oh, it's another one of the
little
little
red.
rock-hugging jellies we've been seeing.
I think that was, man, how many dives ago?
A while.
Yeah. Yeah, we saw this maybe, man, I don't know.
Yeah, sometime early in the cruise.
We're on dive 11,
12.
12 right now. Maybe this was, we saw in the first couple dives, we saw this same jellyfish in this same type of posture where it's stuck onto a rock. And we're not quite sure why they do this, but they are stuck on really well because we've prop washed them, and they've got a lot of thrust upon them from us being around, and they don't detach, they don't seem to go anywhere. So that would be a good one if anybody wants to put that up on Twitter and ask why a jellyfish might be stuck to a rock like that. There might be a person with more jellyfish knowledge out there that's willing to help give us an answer. That's good. Thanks, Russ.
Looks like you got a little bamboo whip on the backside there as well.
Yeah. I'll just do a quick in as we come around. Yep, you can see the bamboo nodes at the base. Just an unbranched bamboo from the family Isididae. That's good. Thanks. I think that's my Anthomastus. Here's another whip coral. Looks like another bamboo. Come in a little bit tighter. The chitin in the background too. Don't see any associates. Alright, that's good. I know we're looking to sample some more associates today, especially crabs, because we didn't get any yesterday. So when we have a chance, when we have the right number of them and everything, we can keep an eye out for that. There's lots of xenophyophores.
Yeah.
Now that you
lots,
pointed it
lots.
out, I'm like, whoa.
So we've already come up almost 150 meters, and I'm sorry, I'm doing bad math. 50 meters since the dive started.
There's a little something in the floating by us. Okay. See it down there? Yep. Okay.
Tina 4.
Yeah. Or, yeah, you can see its arms out too. Or arms, feeding appendages. Very dark ctenophore. I'm gonna lose it as we. That's cool. We got it. Wow, looks like it's like off into the abyss. Let's go cliff jumping. Okay. Oh wow. It's quite the maneuver.
And off the ledge.
Yeah. It's a brittle star. Yeah, I know. It looked a lot more dramatic. That was a cool move though. I could like feel us like jumping off the cliff, turning around, looking at it. Sometimes you forget, I mean, when you're on board, the ship is moving, and so we're moving while the ROV video is moving because the vehicle is moving underwater, and every once in a while it does feel like you're down there experiencing what it's like to be on the seafloor yourself. Have you worked with man submersibles? No? You don't want to? You neither?
Makes no sense. It's an ROV for it.
I mean,
Yeah.
fair enough.
Because
there's
Yeah,
air conditioning.
biscuits.
Oh, there's a. Can you look at this coral over here, Russ? I'm moving to it. Ew, jeez.
Steel coffin? No, thank you.
If it's on the beach and it's, the hatch is open and I can look at the side, I'll be alright.
I think it's quite enjoyable to go in a submersible. It's fun.
I'd like,
Say next, we've never had the opportunity.
yeah, I'd like to do it once.
After you do it once, you want to do it again. I can
Okay.
tell you that much. This looks like an Iridogorgia, so from the gold coral family, Chrysogorgia. We have some branches towards the, not the base, but the lower half that are. Can we bring the ISO down a little bit? Thank you.
I don't see any associates other than looks like a little mysid flying around.
Yeah, a couple of
Or a
mysids.
couple, yeah.
Which those are really difficult. Like you can't. They're pretty difficult to sample. But I would be curious. I think that they feed off of the what is caught in the coral tentacles.
Yeah, steal food out of their mouths.
Yeah.
Literally.
So we're talking about the little shrimp that are swimming around on the coral. Little red. They have red eyes, and we see them pretty ubiquitously across different types of coral, and they're always kind of perched, swimming around the polyps, and it seems that they're probably using them as, as a fishing net. So the coral's actually catching what's in the water column, and then the little mysid is coming along and having a snack, but not actually eating the coral itself. So these are octocorals. You can see when we do the close-up that they have 8 tentacles on each polyp, and the pinnules, the little feather-like projections on each tentacle, allow the coral to have a larger surface area to collect particles out of the water to feed on. And each polyp is an individual animal. So corals are colonial, at least most corals are colonial animals. So we call them a colony, each coral a colony, because it's made up of a bunch of individuals that all work together to create a healthy overall coral colony. All right, that's good. I thought this was going to be more vertical from the waypoint.
Think a little further up.
Okay.
We put one onwards, it
all the
may
way
be.
up to one.
Yeah.
Okay. So we get this part and then they get to do the vertical
So
stuff.
super boring until you get off at
Cool.
waypoint 1, and then somebody will get.
That's what
Maybe you just
that's
fix
what
the landscape.
that's
Would you
what I
like to-
planned it. That's how I
It's
planned
like
it.
apply an accelerator.
Huh?
Apply an accelerator.
I know. We do still want to see the bottom.
I don't know. Still got 300 meters to cover in the first before waypoint 1, so vertical.
Thanks.
Looks like a little stalked crinoid, and then
And
a
a star and a
non,
unstalked.
yeah, and then actually we're gonna take a look at that sponge, Russ. Okay. Oh, and there's a big picnic going over there that we're gonna look at after because it's eating
got food.
something.
Yeah.
Oh.
Oh, jump.
Oof is jumping ship, jumping sponge.
Yeah, there we go.
Let's get a picture of that oaf. That looks like the one that we sampled off the sea. Looks like that. Look at that pretty flower of
Yeah.
a oral disc. Somebody asked earlier, how do we tell if the sponge is live or dead? And I guess we're never really sure if they're fully dead, but normally these sponges, when they're live, they are like a white, almost bluish color, and so as they die, they become that more brown, sandy color, and some of that is sediment buildup on the sponge, and I think some of it is just the tissue. As it dies, it changes color. Take a look at this little crinoid.
Alexis.
Yes,
Just
dead.
wait for a second.
Let's see what we have up here.
Who's getting eaten?
We have our second pycnogonid, or sea spider of the dive.
Don't shoot the messenger.
We'll sit down to take a nice look at this, because it does seem like this sea spider is feeding on whatever this is.
I feel like it's like that's probably a really good example of a beautiful situation.
Looks like another
Okay.
sea cucumber.
I think
Can
you're
we have
right.
the brightness up a little bit of the ISO? That's better, thank you.
That, remember that top left novice?
Yes. So So it does look like this is another instance of a pycnogonid eating a sea cucumber. So a gelatinous echinoderm that crawls around. This is the second time we've seen this type of behavior on this cruise, and the second time I've ever seen it. So you can see when we talk about the proboscis, it's that kind of snout of the spider that's inserted into the cucumber, and it's just really like dissolving, I think, the inside of the sea cucumber and sucking up those nutrients.
So it's like an acid or something?
Yeah, it must have some type of chemical that it is using to dissolve the inside of the cucumber so that it can digest it. Probably digest it before it actually brings it into its body.
Who sampled it? Oh, I don't want that on the ship.
Yeah,
We did on another dive.
We have a couple of sea spiders already. Yeah.
With the actual,
Oh, that
its
was
food.
or
Not
sea
with its
cucumber,
food.
or whatever it is.
It is a sea cucumber.
It's eating, sucking it. It's kind of scary. Hey, Logan, send that to BBC.
No, it's some waste.
Waste two's back.
Yeah.
Is it a duck thing or a thing?
So-
Can we lasers on this? Create a
Yeah.
measurement.
He doesn't like you, Chris.
Can we turn lasers on? Thanks.
Nice.
Oh, one of my eyeballs is watering like a lot. There's lasers right below it.
Yep, I got it.
So it's like 30, 40
Yep.
centimeters in
30
diameter.
30 plus, looks like.
All right, that's good, guys. We can carry on. That's one of the, we don't know how long this process takes just because we don't have the ability to sit and wait and watch this cucumber for hours or days. I mean, this sea
Even
spider.
weeks.
So it could take a long time for it to fully, it could sit there and just be preying on that cucumber for a long time, or it could happen relatively quickly. We just don't have, we don't have a scale for that.
So because the sea spider is so slippery, does it have to feed more often because of the energy storage?
I don't know. There are people that study picnogonids like exclusively, and there are a lot of different types of them, especially in like Arctic waters. So I'm sure there's some more information about it. Like when there's a coral over there. When people do like trawls up in the Arctic, they get a ton of them. Like there's a lot of them.
It just seems so.
I know. Yes, their body plan is.
You store the energy in a different way.
Yeah.
My guess down
There's
here
another
is they
one.
probably don't. They really don't. I doubt they depart. Once they get something like that, they just
Yeah.
stay there until it's consumed, and however long it takes them to consume it, they hang out there. Is my guess.
This looks like a Swiftia, a different type of plexaurid. We'll get a closer look at it in a second. It's missing some tissue. All the polyps are closed up on this one branch, but all open on the majority of the colony. Oh, there's a little, here we have coral predation. Corallivory.
Oh, that's
I'm
cute.
sorry, ISO. Sorry.
Two of them.
Oh yeah. Two little sea stars having a feast.
Is that little snails that are little white ones?
Yeah, I think so. It's hard to see the foot of the snail, but sure looks like it. Look at that one like hanging off. Looks like a Christmas tree ornament. That's Maxoon. So that one is right on the edge of where that
Yeah.
tissue is coming off. And then up here where there's
Oh, it's third star.
oh, it's the third little star. I've never seen that many on one coral
I've never
colony.
seen these little baby sized either.
They're so cute. There's a barnacle. There's a anemone down there as well. Let's see. They all have really munched on this one section of the coral. It's also the only place that you have an oviocanthid, that spiny-armed ophoroid. The other ones are all astroschema. Oh, well, that one's not. Let's see if there's any others over here. Please.
It's the 23rd.
I'm gonna go. Oh, there's an A-plaque offer on that. On this branch right here.
Oh yeah, got it.
I'm gonna just try and get all three of those in one shot for Randy. Well, there's 1, 2, 3, so we just gotta find 7 more. Alright, that's good.
Let's go look at
Oh yeah,
if
there's
that's
another.
a spider or
I think it's another big pygmagonid. Which, man, 3 this early in the dive seems to be a lot to me. Fish. Mhm. Okay. Let's see what we got here.
that one's huge.
Yeah, what, like with 50
Yeah.
centimeter
Approaching
legs.
half a meter.
Not feeding, just hanging out. Okay. Can carry on. There was a coral right up in front of us for us. We can take a look. It's another swifty, I think, so I want to see if we see any other little sea stars predating on it. Yeah. It doesn't look like there's dead tissue like the last one.
Zoom in on that white sand just behind it on this white balance.
For sure. Just like up here.
Yeah, sure.
There we go.
Thanks.
There we go.
Alright.
Yeah. But yeah, looks like another Swiftia, but no predatory sea stars, just the ophiroid that normally accompanies these corals. We'll get a good shot of the coral disc for Tim for identification later. There is an A-plaque offerant on the bottom branch. Right there.
Another one on the
And
seafloor.
another one, yeah. They look different. Okay. Let's look at this. I'm gonna just take a look at this star before we move. It's not close enough.
Appearances here regarding the
Not one that we haven't seen this one on corals before, but we have seen it on the seafloor before. Yeah. I remember that like kind of fuzzy texture.
Okay. I don't remember that.
Okay. Not yesterday, but at some point on this cruise. There's also this one polyp octocoral up here that I think Steve would be very interested in. There's a group of these Alcyonesians that are only one polyp. I just can't tell if it's that or if it's a young Anthomastus. It's very light colored, but sometimes Anthomastus can start out that way before they become much redder. Okay, I think we can carry on. Just a little whip coral. Somebody off in the distance. I think you see where I'm. There's a comment about very quick turnarounds on dives on this expedition. Yes. If we have the mapping data in hand, then we are trying to optimize our time in the water. So that means planning dive tracks ahead of time, and then just acquiring any additional mapping we need, which can be done in just a few short hours. And so if we're not needing it to move far, like last night, we dove on this same Seamount then we're trying to get Sebastian back in the water, and the ROV guys are really great about being able to turn around the vehicle really quickly for us, and so here we are. Very little sleep, very many hours of dive time. This, does this look spongy to you?
No, it looks like those little zoanthid clump, hydroid clump
Oh,
things,
okay.
that we were seeing a couple days ago. Yeah, I think that's a clump of hydroids.
Yeah, I think you're right. There's something in the middle there, the purple. Wonder. Growing on some type of old stalk. It's a pretty healthy colony of hydroids, though. You can see down the side of this ridge. Let's take a look at this sponge while we're here.
Sponges do not look like they're doing well in this part of the.
No, there's definitely a lot of, it seems like a high sedimentation rate. I mean, the rocks don't seem heavily sedimented, but sponges are definitely, seem to be catching a lot of sediment.
I don't know if that's an umbellula or a metallogorgia, but there's something over there.
I think it's a crinoid.
Or crinoid. Oh, you're right. Crinoid. Crinoid.
All right, that's good. We can keep going. Yeah, if it makes you feel any better at home, we on the ship can't watch all the dives either. So we work in shifts where on the science side we're working a 4-hour shift, 8 hours off, quote unquote, to off.
Yeah.
Which means we're either processing samples or doing data entry or planning dives, and then we do that twice a day. So Brian and I are on 4 to 8 local time, both AM and PM, and then we have, you've heard Tim and Anna, who are from 8 to 12, and then Steve and Anamare are from 12 to 4. So we have 3 groups, and then you'll hear Randy, who's chief scientist on this cruise, so she's not running a watch, but she is kind of floating around and coming and going as she needs to while taking care of all of her other responsibilities as chief scientist. And then the ROV team has a different way of running their schedule. Russ, do you want to talk about your watch standing?
Yeah, we do like a midnight to midday schedule, and then we have one guy that runs 6 till 6, so we have a crossover between the shifts so that we've got knowledge of both periods of time, kind of maintenance or breakdowns or any issues that could or may occur, you know. And that tends to be what it works best as well for contact with families at home and that kind of stuff as well. So we kind of set the watches up to suit the guys that are on board as well. You know, if everyone's got a particular choice that they want, we'll try and accommodate that, and everyone will work to what works for them. You know.
Yeah.
We're very flexible in that kind of respect.
Yeah, that's great. Especially because, like you said, we do try and stay in touch with people at home while we're out here, and especially ROV teams and ship's crew are out for much longer than us as scientists often. So, trying to accommodate everybody's ability to call home when it's not in the middle of the night at home is helpful.
A happy team is a productive team.
Yes, it's true.
And this is a particularly long expedition. This is the
Yes,
longest
that's a
I've
good
ever
point.
done. We're normally out here for 34 days, days at sea, which turns into about 32 and a half actual days at sea. But I think the longest I've ever done before this was 28 days.
Yeah, I think my previous longest was actually on our last expedition on the Falkor to the Phoenix Islands protected area, which was 30 days. And some of the length of the cruise is due to situations with COVID right now. So on previous cruises to the Pacific, we have either left or arrived into Hawaii, and either left or arrived into American Samoa or Samoa. So normally we don't have to make the transit from Hawaii all the way down to this region and then all the way back up to Hawaii, but that's what we're doing for this expedition to make travel more manageable, since our science team especially is from the United States. Some crinoids. We haven't seen a huge diversity or abundance of corals or sponges yet. Kind of sporadic little corals. Can we take a look at this little polyp here? Little cup coral. I think we saw one just before this too. I didn't say anything. This little scale worm on the rock. Just based on the very thin base, I'm going to say Javania, which is what we've collected a couple times on this cruise and is pretty common around this region. Cup corals are scleractinian corals, so different from the octocorals we've been seeing in that they form a hard skeleton, but cup corals are solitary, so it's just one polyp makes up the whole organism versus the colonies of corals when we talk about octocorals or colonial scleractinians. That's good. Thanks, Russ. That looks like the one that the sea star was eating.
I agree.
Big crinoid. A couple more sea cucumbers. If we can set down, Russ, we'll take a good look at this coral, because we've been seeing it a couple times today, but we don't have any close zooms on it yet. Lots of sea cucumbers right here. I better beware of those sea spiders a little further down. nice big octocoral polyps. So we can come in. Looks like possibly a little worm living in in between the polyps there. Another. Oh, it's moving. Some type of polky worm.
Is it polychaete or nudibranch?
I don't
See any chaetae, do you?
Oh, I just realized I don't have my glasses on. Is that in focus for you?
Yeah, it's
Okay.
close.
I don't think it's a nudibranch.
Yeah, I think I agree with you.
There, you can see its head.
Yep.
It's kind of cool to watch it crawl along. So the polyps are probably only a couple, maybe a centimeter or two, so the worm itself is quite small. Alright, that's good, thanks. So we've come up about 100 meters, I think, since we hit the seafloor.
Definite increase in the density of sea cucumbers up
Yeah.
here, and diversity, frankly. Several species here in relatively high abundance.
Oh yeah, look, 1, 2, 3, 4, 5, 6, 7. Sea cucumber party. Yeah. Lots of sea cucumbers. Thanks, Russ. You found us a coral.
Fantastic.
Yeah.
Oh, I'm on it.
Stand by. You're also live on the microphone.
It gets better and better.
He was all like casual about it before.
Wow, I don't know if I've ever seen sea cucumbers in quite this level of density on rock before.
Sorry, I moved the camera before you.
It's okay. Thank you.
Okay, we'll just kind of. Let's see here. Looks like a bamboo coral. All of the polyps are retracted. Or except for one little branch up there has them out.
That's a bear.
Yeah, 'cause right now here this is well, I guess that's yeah, no, this Alberta begins in June, so Monday will be the 26th.
Seem to have a gall on the. Is that a? It's a gall
It's hard
or
to tell
it's a
from this
snail?
perspective.
Yeah. But it is a bamboo coral. Do you want us to try and see what that is, or?
No.
Okay.
So right now,
Good.
it's 11, so it's noon right now.
Let's just take a look at one of these cukes while we're down here.
7.
They always kind of look like very slow, gelatinous Triceratops to me. You
I
see
can
it?
see why, but
It's fascinating. You can see the sand. It's just
Yeah, inside their gut. Oh, we got some action.
oh yeah. There's clean sand.
There's the clean sand. So these are responsible for consuming a lot of the nutrients that's left in the sediment. So they just scurry along, and you can see it putting sediment into its mouth with those front appendages. Do you know the names of
no,
those?
I do not.
And then they just keep going, and the sand comes out nice and clean. And this sand must be very clean because there are so many cucumbers. There should be a cucumber transect. All right, that's good. Thanks, Russ.
You don't have to count them individually.
Huh?
Keep a tally.
Somebody probably will when they go back.
There's a
Oh yeah,
Big
slime
slime
star.
star.
Right, hanging up on underneath that little ledge. What are we at? 1780. Looks like we've got another coral up here. Just do a snap zoom. I don't think we need to sit down.
You're not gonna want to focus on that big cup coral?
No.
Okay.
So Swiftia is the pinkish coral, and then there's another cup coral next to it. Don't see any of those little sea stars on there again. Gonna keep an eye out for those. Okay, that's good.
Yeah, or make it no more than 4.9 because Hannah has a column 10 and
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 sea cucumbers that I can count in this one view.
The edge there. Oh, good news.
Jeez, yeah.
Yeah, definitely like this one.
Looks like a little sponge again. Little sponge. You can keep.
Alright, I'm just,
Oh yeah.
a little bit trench out. Just to keep it from shooting up too fast. Other than that, there's
Or you look these big things?
We have. There's many of them. They're sea cucumbers.
Left here.
We have not looked at that.
Do you want to look at
Yes. At least just briefly.
Todd likes to study the ISO, so next is the arms, moving shit,
that?
and,
We go to that.
yeah.
She's
Camera, ISO, just knocking them off the list.
Next.
This is today's Brian. It's just a shift. It's just a shift to show for college students.
Okay.
Just getting set up here to take a look at this little plexaurid. It looks like it has more biomass of oaf than it does coral.
Yeah, it does. It's all the branches are on one side.
We're looking at ISO.
That is some nice ISO work. I haven't seen good ISO work like that in a long time.
I mean, have you not done it? I don't know if it's that.
Since the development of cameras.
I think she's saying out of it. You don't know how to do it.
No, that's not true at all.
She learned from the best. I just taught her.
See? I'd be nowhere if it wasn't for Adam.
Thank you.
This little ophoroid on that plexaurid, that's good. More sea pins. Sponge. I'll just take a quick zoom over here on this Iridogorgia, then we don't need to stop off at it.
Steve's gonna be disappointed with the amount of distance we've covered.
When is Steve not disappointed in us?
Well, that's true.
There's another Singapore one added.
Oh, that's the one. We need that. Alright, that's good.
Alright.
She still hasn't heard back from Terry.
Oh, look at that.
Oh,
Look
there's
at-
a little
That's a
hermit crab in
one
that.
of those long air shells again that look really hard to drag around.
Yeah.
Look at it.
Yeah, let's take a look. I think it's just a hermit crab in there, but he was
Yeah.
moving
It's
fast.
just a long, uncomfortable looking shell for the hermit crab. Still can't get over all of the sea cucumbers.
Sometimes you just gotta take what you can get. When you're a hermit crab in the deep sea.
It's true. I guess that whole shell's dissolving in the deep sea makes them a hot commodity.
And there's just, yeah, I think some of it is also just, there's not that many
It's true.
gastropods down here.
The homolids really have the right idea where they just steal anemones and use them for
Yeah.
shells.
You look at it.
Okay.
You can pinpoint that? We're trying to figure it out.
buddy. That one seems to scurry off
Body
again.
show where she was away for the council. The body would show where she was
But I feel like maybe their abdomen's like shaped to it as it grows. And then it doesn't have to change shell for a while.
Guess that's possible.
There's another plexarid with an ovaroid. Little hermit crab behind it too. So many sea cucumbers. It's a stampede.
Or more likely, don't scare them will cause a stampede.
Yeah. Okay, that's good.
So we're up about 1765 meters now, and we started just shy of 1900 meters as we climb up this this ridge on the northeast side of Titov Seamount, which is the southernmost seamount in the Pacific Rhode Islands Marine National Monument, the Howland and Baker Unit. We're just south of the equator. We crossed about, I guess, a day and a half ago on our way down from a couple unnamed seamounts outside the monument boundaries. This, as the current schedule stands, this is our last dive inside the monument, and then we'll do a couple dives in U.S. waters south of here before starting our trek back to Hawaii. What you're looking at right now is a predatory tunicate. So these are actually relatively close relatives compared to what you've been seeing to humans. They're chordates.
very difficult to sample.
And yes, they're very gelatinous, so they're very difficult to sample.
The movie?
Yeah, I'm trying to get you quote. Give you quote.
I don't know that movie at all. It's a bad movie?
No.
Sounds like it's a bad movie. I have my doubts. Step Brothers? Oh, I've never seen the movie. What? I've never seen it.
Purple crinoids and a solitary coral.
Never ever. I'm gonna fail that assignment. It's only
3 hours long.
We-
Yeah.
First one, all three.
We've gotten good images when we can keep heading on up.
Alright, he's trying to convince me to watch Lord of the Rings or
Well, if you haven't seen Lord of the Rings, you have to see Lord
I'm
of the
just
Rings.
surprised he's never seen it.
I have to admit, I'm kind of surprised as well.
Yeah.
You read the books?
No.
You just
don't think we need to spend a lot of time with it. I just want to get a little bit of a flyby. Yeah, that's that same plexoid we've been seeing all morning. That should be good enough.
Alright, can you bring down the ISO just for a second? It's gonna be one good picture.
All the wrong way.
That should work.
Too
I haven't gotten any updates on my delivery. So I don't know what's going on.
Oh, no refrigerator on range.
What? Of your Beatles album that's still missing in the next
I got that. That just took like
Sure you did.
3 months.
You got that just like you got your for whatever.
My Bronco, I still got the Bronco. It'll be a gift to myself when we get back from San Diego. I hope.
Hopefully you'll get it next year.
Some shrimp.
Yep, shrimp, sea cucumbers. Saw one plexoid.
So a lot changed since that was last year.
Found out Chris has not seen or read Lord of the Rings.
I'm sorry, what?
I've not seen nor have I read Lord of the Rings, and it is not on my to-do list.
Yeah, I'm with Adam on that.
I'm alright with it.
What?
I'm alright with it.
You don't have to be a fantasy lover
You don't.
to watch Lord of the Rings.
I did try once. Obviously.
Those are movies I put on if I like.
Need to see
Yeah,
it.
no, I really just need something like relaxing in the background.
The first one.
Yeah.
Like The Hobbit.
Yes, yeah. I was more of a fan of the Lord of the Rings movies than all of the Hobbit movies.
All of the ones?
The Hobbit movies, like
Oh,
they
yeah.
made the Hobbit in multiple movies.
Yeah, I didn't actually see them all.
I don't think they were as good.
I read the book though.
I didn't see it.
The book.
Get into
I mean, I read the
books.
book when I was in like 7th grade. I feel like.
Yeah.
Never read the books. I just started and I was like, I just watched it.
The books are really good.
I'm sure they are. I just don't think I was in the right frame of mind.
I have one friend who rereads the books every year.
Whoa.
I know.
That's
Sort of like I'd love
dedication.
to read the Lord of the the Game of Thrones books, but I just
The Song of Ice and Fire.
I don't know.
Are you?
Yes.
Did you read those?
I have read those.
What?
Well, there's a reason that I read those.
Why?
I was in Chicago airport and I was on my way to Russia. And I was going to be in Russia for 3 months and they were for sale on like a big thing. And I was like, yeah, that'll work.
What books were these?
Song of Ice and Fire, Game of
Song
Thrones.
of
Yeah.
Yeah.
And there's way more books than there was like TV show and all the rest of them, but like
Yeah, so purchased the first 4. And then
Yeah.
once I read the first 4, I was like, okay, I'm invested.
Yeah. I didn't read all of them. I read the first couple, and then I just
I read all of them.
But I'm not surprised, Brian.
I read the Harry Potter series while surveying
Did you?
and it kept cutting off data and my boss at the time was like, you don't stop that, I'm taking Mr. Potter away. I get like sucked
That was
into
like-
the book.
When we were in quarantine before coming on the ship, those 3 days, every day they had Harry Potter on Bravo, and I was like, yes, yes.
Yeah, I've never read those or seen any of those movies either.
What?
No, I'm kind of with you on that one. I think I've read one book and seen 1 or 2 movies, but I never got into that series.
I didn't make it. I didn't read them as a kid. My sister was super into them,
Yeah, it was an adult.
so we always went to all of the, like, book releases at Barnes and Nobles and did all of that. But the movies are another one where it's just like that's nice background noise.
So we're kind of getting out of the corals and the sea cucumbers up
Yeah.
here now.
Back to deep sea. It's a nice crinoid.
But still keeping a lot of the xenophyphores.
Yeah, not as many as a full, or maybe
True.
I'm just now.
No, I think there's.
I might be
I don't know.
blind
That's
to
a fair.
them.
There's a fair number.
got a text message indicating it would be
About your, what do you have, a
Well,
fridge coming?
a fridge and a new rain, so
Somebody home to accept the delivery?
sort of
Ooh.
he's there, but he can't be bothered.
That feels dicey.
It's on the porch. It's gonna come up to the refrigerator and stove on the porch.
Yeah, doors be stripped off.
All the electronics gone.
Okay, the day before I left, I went to open my stove and the door fell off, the glass fell off.
Oh no.
And rusted out. 5 years. That's what living in the Northwest does to your stove.
Wow.
Wow.
5 years? Did you have a chance to fix it before you left your wife with no stove?
Nope.
I couldn't fix it, it was done. And
Did you
didn't have
wanna
time.
buy a new one?
She's like, well, it's so hot in the house right now. She's like, I'm not gonna
That's
make
true.
it."
Yeah, I get that.
She's grilled.
So the top still works, just the oven?
Top works, yeah.
Okay, so.
So the range works, just not
Okay.
I
the.
fixed it once before, but all the clips like rusted out,
Wow.
and it just fell apart. Like, literally fell apart.
We gotta get
I think you
replacement
should look at
things, cause they
Dory.
just slide in and out, right?
That's what you should do.
Nope, okay.
Play pots.
I do have to agree though, in the summer, even, I was in Philadelphia for a while, and now in West Virginia, I actively try not to use the oven in the summer. Like, what can I cook that does not require turning
Yeah.
this large heat source on?
Pretty soon you're gonna be cooking on an 18th century stove.
I know, I got
Should
that
see her
fire
posters.
burning, well,
I took
what,
a train through West Virginia once. It was amazing.
it's beautiful.
Yeah.
Everybody can come visit me.
You have to see her house. Her house is like 200.
I live in a 200-year-old log cabin.
It's pretty awesome. Getting that picture?
Yeah,
Alright.
it's a bamboo coral.
Oh my gosh. 7:20, we missed breakfast?
We haven't missed it.
I went.
Well, I mean, like, it started a while ago.
Pin it to the bottom.
Lovely mushrooms.
I could smell it when I left the room. That was a bad choice. Now I'm starving.
Is this before?
Even though I had cereal at 2 a.m. Oh no, I had cheese. Post sampling snack. It's a tradition.
Sediment drape seems to have increased here a bit as well, as the corresponding with the drop off of
Yeah,
as
Right.
much biology.
740 meters. So it's been a pretty steady climb. We haven't
Yeah.
really had anything super steep yet.
We had one little ridgeback that kind of questioned. Oh, here's our first tripod fish.
When we first started coming up, it was
Yeah,
not for a long
not for
period.
yeah.
It's kind of like,
And it's been like we've been on top of a feature. We were on top of like a ridge for a while, but there just wasn't a lot of abundance there.
I just got pulled forward. Looking out the
That's an interesting. So, the unknown explorer got a response on Twitter about the jellyfish that was on the rock. I don't know how to say that genus name, but from George Mage says that they might have modified tentacles that help them hold onto the rock while the other tentacles are up in the water foraging for food. If food is abundant, that's a great place to stay in that area. If food isn't around, leave and find a better spot. So that's interesting. They don't actually spend their time foraging in the water column, but they just find a rock to stick on, kind of like corals do. Hi, Tim.
I have to do something with that.
Pretty good.
Okay. That's okay. You're good. Do you want to go eat breakfast?
No, I just got to live.
Okay.
Yeah. Yes, that is my kitchen. That is a
Not too
KitchenAid.
much. It's been very sparse.
Door
A couple plexaras,
An
a couple over to
induction
Gorgia,
stove.
some bamboo whips.
We'll pass it down when you get back to me.
Yeah, I got it.
Just keep an eye on that, so we do it every day or so, so just on the video, not present.
So is that ice making? Is it local power?
It It is yeah, it's a little pockets of it. It's lower than my last one.
It looks nice. It's different at first.
So, here we are looking at this.
Ooh. Wait, they're already in?
Yeah, so he did his job. He gets to stay. I don't really pay rent, so he's got to do stuff.
That's cool. It has like little drawers.
Yeah.
I like that. You see his new kitchen setup?
It's a lot of drawers.
I'm kind of stoked
You want to
that
take
We
a
can
look
take
at
a
this
look at
core?
this core.
Bottom,
Yeah.
because Meadow always opens the fridge and then she just gets inside.
Oh.
It could be dangerous.
Well, she gets inside and she like starts eating, so but she's so
Oh.
small that she can get almost inside, so the door almost closes. So one day I walked by and I was like, oh, the fridge door's open a little bit. When I opened it, there she is eating a carrot.
On the bright side, at least he's eating healthy.
Well, it's only because she can reach a chocolate. I figured with the freezer on the bottom, you should be able to open the bottom of the freezer, but what can she eat out of the freezer?
What? Ice cream?
Popsicles.
It's hard for us to get ice cream to our house when someone's adults in the ferry.
Oh my gosh. You need to just buy a liquid nitrogen container and just
I know
put that
people.
at home. Yeah.
What was that liquid nitrogen container working?
Okay, we have a nice. Let's see what we'll have here. Before I make any comments, we'll take a closer look.
I'm going to hold this.
Oh, sorry.
Do this touch. Alright, thank you.
Or I can give you the
That's fine.
Okay. I would think this is a paragorgid, so bubblegum coral. Based on the way the polyps are attracted, especially at the ends of the branches where you have those big balls of tissue. So with the hemicorallium that looks similar, often the polyps don't retract all the way into the tissue like that. They are still little barrels on the outside. I think this is our first paragorgid of the dive.
Another microbe wand.
Yeah, another hermit crab in one of those shells. The Nastroschema on there. Just wait a couple seconds. Okay, I think that's good.
Sorry. Adam, who's the most famous Canadian author?
Margaret Atwood.
Margaret Atwood?
Sure.
I've read a couple books by Margaret Atwood. She's a Canadian historian. I really enjoyed her work. She did Paris 1919, 6 Months to Change the World, about the development of the tree beside it and also she did more of the piece. The granddaughter of George who's prime minister of England during that time, so she was not that one right there.
What's that?
It's a pale looking
Oh no, let's take a look at this
and a
one.
prettier one, okay.
Alright, what
There's
the-
more going on up there. Looks to me like some possible predation. There are lots of anemones.
I can fly. I'll sit down. Switch an angle. There you go. I'll free fly if you want to sit
Sorry.
It's gonna free fly if you want to
Oh,
zoom in.
sorry,
It's a
yes.
an angle to really sit down.
Okay.
Let it clear out the-
Yeah.
Swiftia?
It's 50, yeah. Let's bring that back up a little bit. Thought maybe it was gonna be one of those sea stars again, but it just
No,
looks like a bunch of
looks like a bunch of
anemones.
anemones.
Got tricked.
So you think these are budded off and created so many anemones, or do you think this was a multiple individual recruitment event?
I could not tell you.
Alright, I think that's all we probably
That's
need.
good, actually. Yeah. It is very sloped. You can see in that video.
You might also be coming into that we get a little ahead of them, so pull to the side and pull straight back.
There's nowhere to gorge yet. It's very faint.
What's that? Oh, it's a
We're seeing a lot less of everything than
Gotcha.
compared to what we were seeing early. I mean, we didn't have high abundances of corals, but we were seeing more regular
Yeah, we were seeing
instances
them
of
regularly, and then there were all the sea cucumbers. and
Yeah.
Now we're into
And-
just kind of.
the xenophyophores are also seem to be.
Yep.
I wonder if there's something then about the quality of the sediment in that region. If there's
This honestly looks sandier, whereas what we saw earlier looked muddier to me. Because I'm starting to see a little sand ridges and some stuff like that showing up in the sand patches.
yeah.
Why don't we take a quick look at that
We'll take a
sponge?
snap zoom at the sponge.
Oh, yeah, I was like, I don't see
I know.
it.
Actually, that might be have a starfish.
I think it's an ophoroid.
Yep.
Yeah.
Oh, off jump.
Dislodging itself. Oh, it looks like a big scale worm on the rock to
I
the
always
left.
see it.
Yeah.
A couple of those little guys.
Yeah. And a big worm up there. That's good. We can keep going.
There's something orange on the
I
top
know.
of that rock.
Oh. Oh yeah, there is. Good eye.
Maybe we're just my scientific opinion.
Looks like another Swiftia. Can we come around on either side of it? Yeah, that's great. Oh, there's another little sea star down there.
Oh, well, we should probably take a minute to image as well then.
Do you think we can set down, Chris? Okay. Oh, there's a coral out there.
Camera back. See what that. Then I'm gonna back out.
Mm-hmm.
It's pretty beat up looking.
It is pretty
Coral.
beat up looking. There's also a black offering. Good. So this is our maybe fourth Swiftia, the second one that looks like it has some predation underway. Another very small little sea star on the bottom. Oh, it's a
It's not,
gastropod.
yeah.
It looks like a sea star at first.
Oh, it's a grazer.
It is a
Breakfast?
yes. Do you want to go eat breakfast, Brian?
Sure. Let me finish this entry.
Okay. And then up here, there's an APAC offerant right here. And also an APAC offerant right here.
But we've only taken one rock. It's the only sample we have.
Okay.
We've seen. very few corals, frankly.
Okay.
Decent number of flexoids, but the same, looked identical ones we were seeing yesterday. And
He's trying to.
Do you want to zoom on an override, Tim?
Yes, please.
Actually, I haven't seen the switches in person yet, so.
there you go.
All right, have a good breakfast.
Thanks.
Okie doke. I think that's good. Yeah, there's a coral to the left. Let's go look at that.
So we're on 398 is the next sample? Can I go eat?
Okay. 398 is the next sample.
Okay,
Yeah.
great. Thanks, Alexis.
Yeah.
Is there any thought about collecting some of those puzzles with you, right?
This is yeah. So we
No,
saw
just
one
got
with
one.
sea stars before. It didn't. We haven't seen 10 of them, right?
Of course.
So you can get a piece, but not
Yes.
the whole thing.
Yeah. Well, and like with the predators,
Yeah, it's
you know.
All right. Thank you.
It's my first Swiftie I've been awake for. I just wanted
Oh,
to
is
let
that
out out there.
Momentous occasion.
Yes, really. You gotta celebrate something. And you wake up. Oh, what is
Have to get organized.
that? A hard coral?
I don't know what this is just yet. I was thinking it's a Hamanaster sea star, but it may be something else.
Oh.
What's that? Yeah, I'm not sure what that is.
Set up very large.
Oh yeah. Yeah, that's fine. Let's go over to the coral island. Okay. Get organized.
Do you want a side view or a
Straight on view would be great. If you can come around.
right here or come around to the left?
Yeah.
All right.
That'd be great. Okay. So they did do one rock sample.
Oh no, this is a hemocrallium or a paragorgia. Probably hemocrallium with the skeletons. It's got a nice oak on it, or astrocetus, season.
The lasers on?
I believe so.
Mother? Oh, there they are. Just want to get it started. That's good. You said corallium.
Yeah. Do you say
You
paragorgia?
good, Chris? Settling in? It's fine.
I think so, anyways. Just based on the skeleton.
I haven't seen it move either. So sometimes the only way you can tell the difference between a paragorgia and a corallium is to see it.
Wave at it.
Just to wave at it. Yeah.
Do that.
No, we're okay. Let's see what we got here. I think.
I like it.
Yeah. Can you? Yeah. Can you wave at it?
Yeah. You wanna get the,
I just want to be sure.
see how those-
We're 17, 12 meters.
You know you see it. Don't
I'm
touch
just-
anything with it.
Let me just check out what else we got. Can you bring the iris down a little bit?
Yes.
Have I got it?
Down there. I don't know which one it is. I don't know.
It's
Last
the
time.
ISO is upper left one. There you go. So you would just give it a little brush there, Tim?
Yeah, still touch.
Do you want to zoom in a little bit on the corals?
Roger that. Hold on.
Oh, look at that associate, potentially.
Ready?
Yeah. What did you see?
Is this bulbous thing that's in there?
No, see the
Yeah.
wavy?
No,
He just touched. Yeah.
Oh, is it? So it's
Did you see it? Did you see how it went? Like it started
It just
here,
snapped back.
and then it went all the way across with the slope.
with the polyps coming in?
Yeah. But it also didn't really move much.
How long did they take to open?
Don't know. Varies, I think. Yeah, I see that. I see that gall-like thing. That this thing here.
Yeah.
It's been pretty adapted at
Yeah, all right, I'm gonna get with it here this morning on this.
just general, obviously it'll slide slightly, but I've been in a waypoint for
I think it's just a overgrowth.
Yeah.
and I've made a transect
All right.
line, which I think it's like
See how many of those we see today. That's great, Chris.
basically shifted that way. Whichever.
No, leave. I was like, leave.
I took it as she wanted it on the recorded video, so yeah, I mean, that's fine. I'm not convinced they want it on the screen. I think it's just.
So we're currently diving on Titov Seamount inside the U.S. What is it? Yeah, we're inside a monument. We're inside the Pacific Remote Island Marine Monument. It's 1,700 meters, heading upslope. I haven't even seen our waypoints yet. Can you come out on the nav screen? Let's see what we're doing. Oh, okay, yeah, I remember this. Okay. Great, thank you. We got about 500 miles to our next 500 meters to our next waypoint.
Yikes.
Yeah.
Yeah.
It's a stark contrast to the dive on the same seamount yesterday. We were sort of in corals all day, in fields of Crinoid corals and plexarids. I think they saw some plexarids on the earlier watch.
This call has 4 people on it. You cannot have 4 people on it because
Well, no, the people doing the tour was just me and one of the presidents.
Hey.
You guys in the library.
okay, that's fine. So we don't have enough channels on sound.
What's new? Let me turn up a little bit here.
So maybe we ran it out.
Oh yeah,
Oh, you asked for it.
another Primnoid. Yeah, can we take a quick look at this, Chris? We'll just document it.
The weather is the weather. Weather outside. That's basically what you said.
So initially, when we started dive on the seamount like this, we want to document representatives that are here. When we first see the coral for the first time, so there's been some predation or something on this coral as well. I don't know if
Didn't
it's
see it.
aplicofrans maybe on this. I don't know.
It's on that top branch. Those aren't polyps, are they?
Those
Or
are
they?
polyps. I don't.
Okay.
I think so.
Yeah, you're right. Sorry.
How are you doing? He's coming around.
Yeah, you're correct.
There we go. I think there's aplox on the ends. Can I zoom in, Chris?
I get you in closer.
It is on the
I think this might be just fine. Anything obvious to you? Let me go back out again.
I don't know if that's regrowth or new growth at the top of that.
Here, the top of the
Yeah.
one, the middle. Yeah, I agree.
saying that we were gonna get a GTA in San Diego, and that would be the last one. So,
Yeah, I'm gonna let you fly, Chris. I don't think you got it.
yeah, I know, I'm just saying it's not anywhere.
Wow. It is some sort of growth over the top of that. Then it's bare. It's these bare patches, like you see in the center. I feel like can be caused by crinoids or things like that just abrade the coral. But I don't see anything here. Nice flying.
No?
You see something here?
No?
What is that on the end down here? That nubbin.
That nub in the middle, like something.
The nubbin down here, right there. Something around. I think it's just a polyp, but this is
So, looks like a potentially.
one of our primary goals is to study corallivory, the predation on corals, and we want to make sure we document damage to corals and. It's just
I
a
don't
polyp.
believe it is being very sandbox. I have to go down there. I can't go further down.
That's all right. That's all right. I think it's just a polyp. I think it's fine. Chris, that's good.
Yeah,
All right, let's make some notes here. All right. So we'll continue to head upslope.
Try to get ahead of him, by the way, don't bring him down to the side, he'll be in front.
Make sure I got it going on there. How did your samples look yesterday?
It looked fine.
Yes.
Oh, from those
From the
sponges?
sponges, yeah. We collected some sponges yesterday for a experiment with respirometry.
I mean, they breathe.
They breathe,
Oh, yeah.
yeah.
And they respond a lot better to deep sea bacteria than they do to terrestrial bacteria. This column on the right, the bottom three are E. coli. You can see that.
Oh yeah. Yeah, right.
There's bacterial over here, but yeah.
Yeah, right. Interesting.
Yeah.
All right.
Hello.
Good morning.
Good morning.
How are you? Just getting underway here.
Just getting underway. Did you eat breakfast? Yeah, did you eat breakfast?
Or do
Would you
want
like
it?
breakfast?
I'm okay. I'm all right for that.
Do you want to turn it off for a second?
Yeah,
Quick meeting?
yeah, right. All right.
Back. Oh, anthomastus,
There's
potentially.
anthomastus. Yeah.
Crinoid.
Crinoid.
See that one? Coming up on maybe a bamboo whip here.
Ooh, that would be nice.
Oh. It's a short one. Do a snap zoom. We're going at a good speed here to. Let's take a closer look at this because I think it's a Predatory. We got a purple crinoid in the background. I'll tell you what, I cannot get focus on this one.
Pretty far away.
Yeah.
Alright.
Oh
See, it's
man.
outward at the base. What do you think?
I'm trying to.
Hang on, I'm
I
missing-
think it. I feel like I see one stripe, and I can't see within the polyps.
I don't see any of the characteristic bamboo nodes. Wow.
Yeah. Polyps are pointing. Man, can go either way.
Well, it may have changed since you.
Just trying to see if something in there too, something red in there. I can't tell if it's further away or what.
I just want to hear this when I pull this in case somebody sort of stole it.
Yeah, I think it's.
Right? I'm going in here. Settle in.
Man, I got my name.
Good?
River and still.
I don't know what's going on with my focus here.
Well, we went in pretty quickly. We zoom back out maybe, and then go in
Well,
again.
I don't think I have focus control. But anyway, it's a really large polyps.
There we go.
Oh, I think it's a. Have you got focus control?
Yeah.
So I'm just trying to.
That would do it. So I think Primnoid,
Part of a.
based on the nose
Yeah.
stripes, like you said.
No, I think so. I think so.
The way the polyps are
let's
casting
call it
shadows
that.
in the stalk, it like makes you think
Yeah.
maybe, but I don't think so.
All right. Sounds good. 6 meters. Yeah, got it. Thanks. We'll keep rolling. Given that we're in this U.S. monument, we are limited in our collections.
What kind of jelly is that? Or whatever's floating? Yeah, it's fine.
Where you see a Tinaphore or something?
Yeah, that's probably what it was. It was very clear.
Oh, I see it now.
Kind of floating in there.
Yeah. For example, we really are not allowed to collect any whole animal unless we see 10 of them or so.
Yeah, 10.
So we just have to document things, take a count of what we see, and just be very careful. In general, we rarely collect whole animals like corals, sponges. We just collect bits and pieces of them to give us what we need to identify them, to do the work we want to do. What's that? This bushy coral over here. Let's take a look at that.
Is this a dead skeleton with things
It is
on
a dead. I said coral too fast. Should have known better.
Well, it looked like one.
Yeah, it's still shelf. It's still running, so yeah.
It's definitely got some
It's a coral skeleton
hydroids
loaded with hydroids, yeah. Curoid in there. Yeah, that's good, Chris. That's right. Yeah, I'm kind of fine with that. Let's see, up on the slope. Let's see, I anticipate our depth to, we're now at 1681. And is this the right?
Yeah.
Okay, so our waypoint's at 1434, so we got a ways to go up slope, so just keep making tracks.
Yeah, it's pretty far.
We dove here yesterday, last night, whatever it was, and then mapped for 2 hours to fill in some gaps around this seamount that was discovered in 1961. The Cricigorgia, yeah, sorry, I almost missed that. Take a look at that one, Chris. I don't know if they've seen a Cricigorgia yet on the staff.
Alright, I'll send it to you.
Yeah.
We're gonna touch.
No, I don't think we're gonna touch it. We're gonna just document it.
Got a crab in there.
Looks like it's got an associate in there. We are documenting corals and sponges and the animals that live on them. On all these seamounts we're diving on to look at various questions that we have scientifically. Some are biogeographic, like what lives where and what depth. We see a lot of depth zonation with corals and the animals that live on them. We were south of here in the Phoenix Islands 4 years ago, doing the same sort of work and identifying animals like we're doing it today. And that is a Cricigorgia. Small, large polyps, and you'll see inside.
Yes, it doesn't. Oh yeah.
That's I'm gonna go.
Where's an associate, right?
Oh yeah, that's a crab eye looking back at us.
Yes.
See him now? You see the belly of the crab. You see some of the legs
Uh-huh.
move. You have to sit up and set up on it and just watch it for a minute. See it move. See the claw up top. That's a Europtychus crab. I'll go back up again. I don't see anything on this Cricigorgia, but these crabs live almost exclusively in corals like this for their life. Now you can see the other arm and claw of the crab. These are really small, sometimes the size of a quarter, a U.S. quarter, even a nickel. All right, that's great. Thank you. So we'll document that those crabs are in there for probably the half of this dive, and then maybe we'll be able to collect one and confirm its identification. Those animals that live in there can be very specific to those corals, so that only that crab species will live only in that coral species, and that's called fidelity. And so can be a high degree of fidelity, and we've seen with depth that fidelity increases with depth. It gets more, and the coral associate will be more distributed across different host corals when you get shallower. So we like to cross a lot of depth. We started today at 1781 meters. And now we're at 1680, so we've gone 100 meters in elevation since the start of the dive. We'll continue to go upslope, and we'll see if we lose fidelity or have a transition in the coral associates that are in the host, living on the host. Is a stalked crinoid. Here, we saw those a lot yesterday too. Put up the iris a little bit. All right. Okay, press on. Another chromatulid crinoid, an unstalked crinoid. Look at that slope. You can really see that. These corals and sponges love hard bottom substrates, so that's why we focus on vertical and steep slopes in our dive planning, our tracks.
I heard you over there. I was like, is Alexis here?
Goodness gracious.
Teaching me corals. Right about there.
Tulip, and the one before that too.
Okay, let's get a snap zoom on this. I don't know if you've seen this before on this dive, Chris, or not.
None of this stuff now.
All right. There we go.
And if you want to be a big, take care of the iris for that. Give him something to do. He needs responsibility.
I'm losing it.
Listen to a word we say. I'm
So-
more than happy.
Send it to them.
So we got a stalked glass sponge here. I want to document that's here on a long axis, long stalk. This is not like the one that we looked at yesterday for your experiment, was it?
No, much different than material.
All right, that's great. That's
We
fine.
took a
Let's keep
piece of
rolling.
one of these tulip ones though for culturing a few dives back. Several dives back.
What's that?
Yeah, see, I don't have any iris control. All right, Ben, can you lighten us up? Yes, please. My iris is not actually working.
Perfect.
So, all right.
Big sea spider.
Yeah, let's take an image on the sea spider. Oh no, I gotta turn that off. That's not gonna work.
We saw like 3 of those earlier, Tim.
Today? Oh, you did? So you got them.
We
You
saw
got 'em.
one eating a sea cucumber.
Oh, that's scary.
It was like sucking it into
All right, we'll keep going then. We got another one though. I'm sorry, Chris.
Yeah, we see something.
Yeah, I didn't know that. Okay.
Yeah, those things definitely are the things. I mean, nice sea spider, but at the same time,
Bridge.
they are listed as a thing of nightmares from the deep sea.
Nope.
Oh, I can't.
Open it up.
No, I know.
Please.
Wait.
I'm having camera issues this morning. It's me. What's a shrimp in the family Nematocarcidae, long legs?
Is there any chance we can search that waypoint just down a wee bit and I can back
Okay.
Put it back behind
It's
you.
another crinoid coming up.
Put it behind you.
Bravo. Thank you.
There's a old stalk skeleton.
Go to the right.
I didn't see anything.
I'll send just that one next. That's it.
Oh no. Oh, that's okay. Well, we were really in corals yesterday all day. We were on the different
No,
side
now
of the
we're
seamount
paying
now.
for complaining about the PRIMNM forest.
Yeah. Well, we were
Not
just seeing the same
complaining,
thing
but, you know,
over and over.
we were
and
ready
over
for
again.
more diversity,
Yeah.
and kind of went the opposite direction here.
A lot of rocks. There's another stalked crinoid. There's another whip. Just a quick zoom.
Yeah.
going a little bit. Oh, hell.
Crinoid or web?
No.
The old age question.
Yeah, just going down the thing here. Yeah, yeah, thank you. Yeah, I think it's, you see it was a bamboo whip.
Yeah, you got
Yeah,
that one node
it
at the
is.
bottom, right? Is that what you're
There's
going
the
off
base.
of?
Yeah. I think the one we saw before might have been a bamboo too. Then this has got a funny branching pattern. I was trying to get into the branches here, how they.
Spin around, you better look at the branches.
Yeah. I won't touch the camera. Sorry, you guys.
Fly around it.
Yeah.
Kind of side.
Just trying to keep focus on it. Just a branch whip. Yeah, I don't know what bamboo coral clay this goes into, but we'll document it and we'll look at it later. It bifurcates. Two branches coming off at the same point, then it branches again. That's kinda neat.
what we collected for Les the other day.
Is it? Oh yeah. Yeah, that's great. Thank you. There's reminds me of an Isidella.
It's an Isidella.
Isidella coral. That's what it looks like with it. It's like a... Hello.
I'm just checking it.
Yeah.
I'm gonna go break down.
Yeah, they already sampled a rock. I don't think I have.
We don't know what kind though.
No, no, yeah, no.
It's football shaped, I guess, 10 centimeters.
No.
Is that rock earlier that looks like heart? Heart, like a
Yeah. Did you sample the first rock?
I flew and back into
Yeah, but I mean, it was you saw it was it really dark and crusty?
Yeah.
Yeah, this is
Yeah,
not,
okay.
this will all.
Then we're good.
Yeah, it was the, see, definitely the whole,
But like, this is literally like half an hour.
very,
So, yeah, okay, so I'm just saying that's, there's not like.
Yeah.
No, but.
All right.
Yeah, it really, it doesn't matter. It is like slicing it, you can't cross it all in.
Come up a little bit so we can see what we got up here.
the
Can't
geologist.
locate it across the line, so it's gonna be a tricky map.
What's that?
That's a geologist.
There's a group of geologists at USGS. Right. No.
How difficult
But
is it
anyway,
to
I
extract
think that going to like, if you're gonna try to do your fuel.
I don't think it's difficult at all. I don't think it's difficult at all. I think it's just
Is what it is,
It's
the
part of
other
it.
option is we just sit and wait till however many hours. Yeah, I'll find out.
Are you okay?
Yeah. Yep.
Let's run a look.
I was like,
Yeah.
what was that
We're
case?
good. We're good.
Can she sit in for you for a little?
She's working. She's got stuff to do. That's
She's about to.
yeah. We're good. All right. 1,650 meters on Titov Seamount. Going up to a depth of 1151 today, which is the top. We'll be up coming up to the rim of this seamount. Named after yeah. Yep.
Change of plan. Hello, we are having a bit of a watch change here, so forgive the radio silence for a second as we sort ourselves out.
Morning.
Morning.
Good morning, Randy.
I'm still waking up too. Good morning. We have Chris and Ben flying on the Sebastian. That's how tired I am. On the Sebastian, on Sebastian, our ROV, and what a beautiful day for flying it is. You guys want to say hi?
Hello.
Hello. Hi there.
We also have Debs, our fantastic marine tech, who is. Oh, yep. She's waving. You can't see her, but she's here. She is managing our live stream and working on our mapping and helping us coordinate our imagery and sorting our data. And really, I don't know what on earth we would do without her. So thank you, Debs. She's probably doing more things than that I'm just not aware of, because that's Debs for you. Making all the behind the scenes things work. I am Randy Rojan. I'm faculty at BU, Boston University, and by hook or by crook, the chief scientist of this expedition. Sorry, you're stuck with me. We are doing seamount after seamount after seamount after seamount. So we are in a big run of seamounts on the Tokelau Ridge, which is pretty exciting. But right now, so you'll see us, that's why we're in the water so much right now. These seamounts are all really close together. We are hanging out. Can you just zoom on that for a second and see if that's a bamboo coral for me? Like, or a broken piece or what? We are on Titov Seamount. Yeah, exactly. We're on Titov Seamount, which is in the Pacific Remote Islands Marine National Monument, which is in the U.S. waters, in the Phoenix Archipelago, which is a 10-island archipelago that's actually cut in half by the equator, with 8 islands and a bunch of seamounts in the Republic of Kiribati, which we explored. Oh, that's a hermit crab in a shell. Thank you. I'm
It's
good.
the third one we've seen that had a shell.
Okay.
Pretty weird.
That is interesting. Thank you, Chris. So we were in Kiribati in 2017. We did 17 dives there, and now here we are in the U.S. side of the Phoenix Islands, where there's 2 islands and a bunch of seamounts above the equator, and a little bit below the equator, where we are right now. And let's see, this is our 12th dive of the expedition.
Yes.
I think so.
That is a
We're all pretty sleep deprived, so we're gonna go with 12. So here you go, guys, a dozen, and pretty soon you'll get the baker's dozen as we go to
Started
another
at
seamount in a day.
4:19 and we're on 4:30.
Yeah,
It's 12.
that's 12.
There you go.
12. Hey, I can count even though I'm tired.
It's weird when you have to actually count the number. 'Cause you can't just subtract.
Yeah, it's kind of one that we're on.
It's
Yeah.
true.
One off, yeah.
So we are going to take a look at this little coral. Thank you for anticipating my every need as I get set up. I really appreciate it. Can I snap zoom?
Hey, Lloyd.
Randy, there's been a request from Outreach to hold shots a little bit longer. So
Yep.
if you snap zoom, then just, if it's something you think is a good shot, then hold for at least 10 seconds.
Okay, I think this is just a snap zoom.
Okay.
So we're going to keep going. But thank you.
Before we do that,
Yep. Are we going to white balance? Are we going to focus? Okay. No, I'm just wondering what we're doing. Working on our focus. Okay, so
that's the greatest one. Alright, cool.
I'm looking at the chat and I see that you guys are disturbed by our silence, so let me just explain that sometimes when we cap off, we're talking about operations and logistics, and you really don't need to hear about what's for breakfast. I mean, maybe you want to hear about what's for breakfast. It's delicious. Or who hasn't slept enough and should probably, or who has a meeting, or who needs to swap out an experiment, or who, whatever. So there's all these different things, and so we usually just turn the audio off to keep those sorts of details. We give to give us a modicum of privacy. So forgive us when we turn it off. It's not personal. We're just trying to work out, who needs to be where when. So when you hear a little bit of silence from us, that's what's going on, and then we come right back in and get back to the science.
But hi, everyone.
Oh, look,
Morning.
you're here. Oh, good. And we have my watch partner here. Please introduce yourself.
Hi, I'm Anna Gauthier. I'm currently finishing my PhD, and I'm co-mentored by Randy at Boston University, who's with me today, and Jonathan Kagan, who's at Harvard University, based out of Children's Hospital in Boston. We are asking a lot of questions about deep sea coral immunity this time we're here. Last time I was here, I was culturing deep sea bacteria to ask questions about mammalian immunity, so we're excited to flip the question around and get some of these primordial responses and what's going on. And I'm hoping after maybe some of these more rocky outcroppings, we might get a little more abundance of coral, but we'll see.
But that's the thing about rocky outcroppings. So first of all, it's just, you never know what you're going to find. This doesn't really look any different from, geologically, although I'm not a geologist, but it's
No.
rocky, it's slopey, it's lightly draped, it's lovely, it's encrusted, it's botryoidal, it's all the things, and yet in some places we find tons of corals, and in some cases we don't. We're going to take a look
Another
at this.
spider.
Another picnigonid.
That's the 4th one today. Interesting.
Okay, I do not have the Slack open. Do you?
I can.
Okay. Oh, what are you eating? Okay, so one of the questions we're asking on the seafloor is who is eating who? Picnogonids are sea spiders, which are what we think are generalist predators. We've seen them on a number of different things, but I have no idea what it's perched on top of.
Looks like
take
a
a look.
sponge.
Sponge, anemone, gelatinous blob, sea cucumber. let's see. Either way, they remind me of the Dementors from Harry Potter, where they just like suck the life force out of you and then just sort of extend their proboscis and go to town. But they are. We're here to study corallivores, and these are certainly corallivores.
Is its proboscis on anything?
I can't tell. No, I don't think it is, but I don't know what's going
It's
on.
on that sponge, is it not? What is that?
I can't tell what it is until we get a better angle on it. Right now, it looks like.
But you can see it's almost like that long elephant-like nose in front.
Yeah,
is
exactly.
the proboscis, and it doesn't look like it's touching anything.
Yep. Exactly. Right now I'm going with it's eating a UGB, an unidentified gelatinous blob. But we'll get a closer look at it and try to identify what it's eating.
Not a UGB.
Not a UGB.
It really has those legs up. Yeah, it
Yeah, it
does.
does.
The research sea spiders have swirly straws for drinking.
All right. Tell me when I can. Oh, you got it. Oh, thank you.
What is that? Is it a sea cucumber?
Mm,
Or was that a thing it's behind it?
is that a remnant of a sea cucumber? Is it a
Like a-
very squished up holothurian?
It's still a UGB.
It's still a UGB.
Still blurry.
So our pilots are amazing, and we sit there and say, please look at this teeny tiny little blob this, on the bottom of the seafloor, 1,632 meters below the surface of the sea, just below the equator, in the middle of the Pacific Ocean, where we haven't seen people for days, and we're 1,000 miles from anywhere. And they're like, sure, no problem, got it. Because you guys are stars. What is that? An anemone? It doesn't really look like an anemone.
It's hard to know if the sea spider already got to it.
I know, it looks kind of collapsed. Or at least very, understandably.
Nervous.
A nervous UGB.
Yeah.
It looks a little.
So, I wonder if anyone on
No,
YouTube's gonna
we
throw
need to
something
get a
out there.
better view of it. Chris, can I ask you the impossible and ask you to spin around the other side and look at it that way?
I don't know.
Thank you. Once again. Oh wait, you know what? Actually, let's take a quick snap zoom on this down here, because this might be the same thing. I'm just making sure that's okay before you fly. Let's take a look at this UGB. Aha.
Almost
Well,
looks like a jelly.
I don't think so.
No,
Is
it's
it-
not.
It's
One
not
of
a
those,
predatory tunicate, which
okay,
is I
that
know
was
what
what I
you're
was
thinking
thinking.
about. No, I don't think it is.
It's just an encrusting sponge kind of
It
thing.
looks kind of like that.
But it's more translucent than I would expect.
Hang on, here's another one. Hold on. We just need to get some angles here. We'll figure it out. You got this. I think
That's definitely what it is.
It's the same thing.
It's a sponge.
no, I'm leaning towards highly. Right hand. Here's another one.
You're leaning towards UGB.
No, I have thoughts. Check our Slack channel, see if anyone else in our group has just identified this thing and knows exactly what it is.
No.
This may just be my ignorance that I don't know what it is because although I'm leading this expedition, I work in many, many different ecosystems and I kind of sometimes work in the deep sea but often don't, and so there's often things that I don't know, and I rely on my extremely experienced team to be able to say, oh, that's a whatever it is, and turn something from an unidentified gelatinous blob, a UGB, into something. I know, it looks and then, I don't know, here we go. Oh, okay, maybe this is the same thing, uncurled. This to me looks like a sea cucumber, exactly. This is a holothurian. But so everything else could just be curled up. It looked kind of like an anemone and other things. That's a very...
Maybe a half-eaten holothurian because we saw one eating one earlier, so maybe this spider ate
Oh,
half
a
of it.
half-eaten holothurian. That's possible. But what about these things? They look almost anemone-like, but not.
Sea spider, tell me your secrets.
Oh, Anna's making up a song about it.
That's all I got though.
Yeah, I don't know. Maybe it's not something.
Touch it.
Um,
Touch it.
Can we, yeah, let's poke it. Please.
Gently.
Gentle.
Tiny.
Let's do a gentle.
That is nothing but gentle.
Boop is the word that we've been
You a
using
gentle
for
boop.
a while.
Okay,
Yeah.
I was trying to come up with a fancy science term for you, like a let's. I don't know. I'm too tired to think of
I
it.
think boop is
Let's-
good.
let's
Gently
boop.
investigate it with compression.
Let's boop the UGB.
Stand by. Banter.
Stand by for booping.
Can't really see it unless it's not gonna help you at all.
Or we could collect it.
Hold on. I'm gonna have them spin around and look at see what it is on the other side.
See the shadow underneath. Is that there?
Sorry, I'll stop talking.
Okay, so what does it feel like?
It is.
Doesn't feel like it.
I mean,
I don't think you touched it yet. There you go.
oh, there it is.
Alright, well
It's
not
gelatinous.
Yeah, but it's not super like
It doesn't
it
explode
didn't
upon.
rip it. Yeah, it didn't rip apart.
You sure? It looks a little drippy on the bottom. Does it feel spongy or does it feel?
It has some give.
There you go. That's an email.
Okay. I just can't tell how much force you're putting on it, right? So it's
It's like a balloon, water balloon.
it's like a water balloon. All right.
You want to spin around with the one that does the spider?
Yeah, let's do that. Thank you. I don't know what this is, but I'm sure it's known. I'm sure this is me not knowing.
Problem is this Google search term do you use for something of this nature? It's like small gelatinous blob at the bottom of the ocean.
It just didn't change at all when we touched it, right? Like I didn't see any change in no behavior, no change in morphology, no change in shape. It didn't retract or coil.
It
just,
knows it has been booped now.
It
Yes,
just
I think it
acted
does.
like a water balloon. Oh, perfect, without exploding, which is a good thing. Good job not exploding the water balloon.
Well, hopefully maybe this other angle will reveal something.
Oh, well, thank you guys for asking the Twitterverse, because thank you, Lisa, because I am definitely not sure.
Let's get a good image for them again.
All right,
Once Chris is sit down.
All right, I'm gonna zoom in to the one that is that is next to a pycnogonid. We don't want to assume, although goodness gracious, that does not look happy.
The other ones looked, yeah. Like, this looks like a deflated balloon.
Hang on. Let me send a. Hold on. Picture to Alexis. I don't know. I don't know. Well, maybe we should try the slurp one.
Okay. You want to do that one, or you want to go back down to the other side?
Let's go back to the other side. Sorry
Okay.
to make you move back and forth, but this one, whatever it is pretty damaged. I assume it's damaged. Maybe it's in its happy state because it's best friends with the pycnogonid, but I doubt that.
Yeah, like he's hanging out with the scary
Yeah.
spider thing, so they nobody bothers him. So that would be.
Yeah.
I feel like that's actually the truth. that one we booped already.
Yeah. One of the great things
Thanks,
about
Lisa.
thank you guys about all of you on the chat is that you're willing to help us out, which I love. And I think it's also really important that one of the most important words I learned when I was a graduate student years ago are the words "I don't know." I think it's really important to be able to be really honest when you don't know something, and then that's how you learn. So we know a lot of things, and there's a lot of things we don't know. And in the deep sea, it turns out there's lots of things that nobody knows. So we get to figure it out. Yeah, I think that's a good one. Either one of those two, whichever one's applicable. But in this case, it may just be.
Down below that level is not.
So far, we still have a UGB, unidentified gelatinous blob.
It's interesting.
Although I'm
that
pretty
this
sure that,
other
I mean, that's
one-
definitely a holothurian. It just
Yeah.
doesn't look like the same thing.
No, it's
I
just because they're both clear, but
Yeah, I know. And roughly the same size and in the same habitat, but I don't think they're the same thing. Alexis doesn't know either. Okay, so I think we are justified in our UGB designation, and we will pick one up. Thank you.
it's like, what's the life cycle of a holothurian? Do they have like a sessile like stage?
No. But, good luck. I mean, it could be an egg of something. It could be a whole creature that is just, we're looking at it from a funny angle, right? And we can't figure it out. It could just be, it could be something new. It could be like something simple, like I don't know, like a ctenophore that we can't really see all the features for, right? It's just, that's the thing, it's pretty featureless. If we had some of the characters, we'd be able to kind of get it to some taxonomic level, but all I see is the gelatinous blob with, looks like a big circle in the middle with no
That
no
feels like an air
features.
balloon.
So,
Or a water balloon,
when you
excuse
don't
me.
have a whole lot of, general invertebrate biology can get you pretty far if you know the major characters, but if you can't see any of the major characters, all you have is a UGB.
Oh, is it, here, drop it and we're going from the other side, dude. Oh, hold on a second, maybe not.
Meanwhile, Pycnogonid is moving on to greener pastures. He's decided he doesn't like our commentary, and he's showing his displeasure by
Alright, I might be able to.
moving
I
on.
think there's an anemone downhill. He's probably got his eye on,
Right. Yeah, I'll try
or
this,
she,
see what happens.
or they.
But this not gonna work. Yeah, fly out. Sorry, our host is underneath our arm, so we have to fly out.
Oh, got it. No problem. We're going to reposition ourselves here so that we can get a better angle on the UGB.
We move it, yeah.
And yes, we're hoping that while we suction it, we will be able to get a better angle on it, and then certainly when we bring it up to the lab, we'll be able to figure out what it is. Should be pretty easy then. We have microscopes. We have a lot of different tools with us up in the lab to be able to help us determine what we're looking at, which is partially why we sample. We can also, if all else fails, preserve some of it for genetics or send some of it to some of our taxonomic expert colleagues at various museums like the Smithsonian or the Museum of Comparative Zoology for an assist. Phone a friend strategy when in need. So we'll see. But either way, when we're trying to think about pycnogonids and them as generalist feeders and trying to figure out what they're eating in addition to corals, that's why this is an important data point, because it will tell us a little bit about the types of things that they eat. If this is cnidarian, then we know that they, it tells us a little bit about their kinds of preferences. If it's not, then it broadens the family of organisms that it's feeding on. I'm tired. Okay. Pycnogonid
We get the
eating
point.
a unidentified. I'm putting UGB in our notes. Gelatinous blob. There, now it's official. UGB.
Mystery clear orb. I see that's another name. Do you like the word orb?
a good word.
Alright, there's our guy.
Yeah.
This
That'll
is,
work.
yeah, I don't
That'll
know.
work. This is great. Let's, it's very small. Maybe it's before you take it, can you stick the lasers on it for 1 sec?
Yeah.
So we can get a little side, we'll bring it up to the lab, but just in case it falls apart in the suction sampler. There we go. That's good. I can grab a measurement from that. Thank you.
You just waste your time right now, so I'm just feeling better.
Yeah. So we couldn't see a mouth. All we could see was sort of like a center circle, which almost looked anemone-like, but I didn't see any tentacles curled inside.
No.
Now it's possible they're tucked all the way in and we just can't see them. But they're all sort of very tucked in, and so that's why I went to a couple of them looking for evidence of what might be. No worries. Yeah, absolutely. What are you looking for?
Another pen. Put off.
I have a Sharpie and a pen. Aha! And this unidentified gelatinous blob is going to have the distinction of being our first biological sample of the day.
When you're ready, now slowly start doing. We're not slow. Just a little stress on the animal as possible. Might have to flip it, I don't know.
Oh, there we go.
That's beautiful. Is that jar 1 or?
That's 7.
7, thank you.
And sample number?
398.
So this is the situation. I can't tell if
There
it
it
was,
is.
alright.
That's in. Yeah.
Alright, so what we'll do, just to make sure, is we will,
And that's at 1632 meters.
Yeah. So who won the bet last night about the sample number?
Hmm?
Who won the bet last night about sample number?
Tim.
Tim. All right. The man who doesn't like chocolate or coffee.
He had a chocolate mousse last
All
night.
right. Well, maybe he does like chocolate.
Yeah.
Okay, sample.
I don't know. Do you see it?
Don't really see anything.
I don't see anything
Trade is
either.
on the- can't really, hold on. See if I can get better light there.
I don't see it.
What I can do is go back to that one and send it, the section back on.
I think.
Or we can keep it and put it once, section one, see if it, I can't, it was clear, so I.
Yeah. I think we can try to maybe collect one more.
Okay, no
And
problem.
then into the same jar.
Same jar,
And
alright.
then hopefully they'll both show up, and I think that's fine. There's plenty of them all over this wall. There's a lot of them. So this does look like it was attached to the wall, which is part of the part of my anemone thinking. But again, I just didn't see. We're hemming and hawing. So the only thing to do is make sure we have it. So we're not sure it actually got in, so we're going to collect another one. which will also give us, actually, can I ask you a favor? Can I plug this one? If I can zoom all the way in, well, oh, we have it?
Something that looks like it might have been just
Oh, good.
spun around, but we'll collect another one just in
While you're
case.
doing that, can I zoom all the way in while you're
Yeah,
plugging
you
it so I
can
can see the attachment
Yep,
piece
yep,
to the
I'm
wall?
trying to find
That'd be
another
great. So we'll call it the same collection number. And I want to actually watch the attachment point. Cool.
Is that right there?
No, that's a sea cucumber. You want the one behind it. Yep, right there. Yeah.
Alright. Ready?
I'm gonna.
Oh yeah. I'll zoom out so you can get
Okay.
his
Yep.
thing on there and then let's get close. Alright.
Yeah, if you can take a picture of that and stick it in Slack, that'd be great. Hannah.
Taking a video.
Inflated.
Whoa.
Alright.
See, that doesn't look like an anemone from this angle. Not even close.
Stuck in the bottom rock right underneath it. There you go.
Nope, I don't think it's an anemone at all.
It's tenacious.
Tenacious UDV.
We rotate it all to get that corner down a little bit, you know, where it starts to get the cut off is. Just trying to get in there a little bit more.
It is definitely changing it is like just like a water balloon.
It looks like a really tiny version of those tunicates, like where the little fit, they look like little only hair dryer things
Exactly. But I don't think it's a tunicate.
or whatever.
Yeah, I know what you mean.
Alright.
Perfect.
So there it goes.
Perfect. All right, well, we'll see what.
There goes the limit. Saw that one definitely.
Great, we definitely have one. Thank you very much, pilots. That's awesome. We're really, we will see what it is. It's some insane unknown hour when we come up. All right,
Yeah, we'll let you
thank you
guys
very
know
much.
when
So we will,
Onward and upward.
yeah,
Right.
the only thing,
Yeah, that's
can you
right.
just put putative corallivory on that so that we know what it is? Even though it's not corallivory, just put
I already made it.
pycnogonid predation on something. Okay, so as soon as we get ourselves repositioned, we will move on. It could be an egg, it could be another organism. I just need to see a little bit more of the features and characteristics to be able to figure it out. And that is the beauty of Sebastian. It is not just a camera, it's all the things.
Yes. Alright.
All right, let's go flying. I don't even see it anymore, do you? He moved away.
That was on what, 7?
7,
Yeah.
Is that it? Or is that
No,
a
that's
crinoid?
shrimp. And that's a crinoid, yeah. Oh, no, you might be right.
It
Yeah, that's
is.
the pycnogonid. He's moved on.
Surprised there was a sea cucumber below him. I'm surprised he didn't go down that.
All right. Never know what you might find in the deep sea floor. I feel very inadequate as a scientist right now. Where all I have is unidentified
Is
gelatinous blob for us. I have nothing.
Is that a sea pen?
Yeah, that's a sea pen. Can we take a quick look at that Umbellula sea pen? Thanks.
Is that one of my nemesis sheets?
And
So there's 2 things right there. What's up, Zoom?
Yeah, it looks like there's a a little crinoid next to it, and like a large, tall, likely bamboo next to it.
Cast a nice shadow.
It's it's probably the first time in its life it's ever cast a shadow.
Well, it's doing a lovely job.
All right.
See both?
Yep, I'm good.
All three, actually?
1, 2, 3, 4. 1,
Oh, I
2,
didn't see that one
3,
down there.
4.
Yeah, I
Yep.
only saw those three.
Okay, good. That's an urchin. That's a crinoid. This is an Amalula sea pen, and I just want to see if there's any associates on it. Because we've seen some interesting sea pens on this trip, and sometimes there's fish eggs and other things associated with them, so I'm just taking a look. And I don't see anything, but my goodness, it's beautiful. Let's see if we can get a better focus on this. I am working on it. I'm terrible at focusing. Okay, now let's try. Oh, there was an associated. It looks like some kind of small.
Just hold that
small
shot.
crustade- Oh, that's a worm. It's a scale worm.
So yeah, your side is you're doing it.
Oh, wow, look at this.
10 seconds.
Okay, 10 seconds. 10 seconds.
Okay.
All right. Thank you very much. That was great. Okay. And then let's take a look at this coral over here. Doop, doop, doop. Let's get some focus. Gorgeous. Yeah, another bamboo, unbranched bamboo coral.
I don't know what you did back there. Hey, what?
Just sort of scanning for associates or any sign of anything. And nothing else, giving us a good look at this little coral. It's not so little. Oh, there we go. We've got a little mice and shrimp on it. Up we go. Where's that little amphipod? I think it was a little amphipod actually. Yep.
Not just pretty fish.
Okay, that was great. Thank you very much.
The other two down there don't.
No, it's an urchin and a crinoid, which are fantastic, but we know what they are. So let's go.
What waypoint are we on?
We are. I can't tell what waypoint we're on. I have no sense. We are approaching. We're halfway to waypoint 1.
So we need to get up to 1434 meters, and
Been
we're at
very efficient today,
1627.
You know, I'm good
That's
with this.
okay.
If we're stopping, it's because there's things to look at. Some interesting little sponges. We're going to take a quick look at those actually, just to characterize them, please. So we're on Titov Seamount, which I learned yesterday is named for the cosmonaut something. Well, his last name is Titov, and he is the first person to circumnavigate here.
That would be Yuri Gagarin.
That would be what?
Yuri Gagarin.
The second person to do it, which is Titov.
Okay.
All right. Which is very cool. So let's take a look at these guys. I love the sponges. They are so interesting. So this is some kind of hexactinellid glass sponge, stalked. 2 of them. It's always nice to be on the deep sea floor with a friend. We have a little urchin in the background, and, ooh, excellent, a tiny chrysogorgid in the background. So let's take a look at that while we're here too. Right behind the urchin. See that?
Yeah. Quit touching it there.
Antics abound in the control room. So in reality, for those of you who are wondering what it's like to have life on the ship, everyone here is really amazing. This is a group of people who work really well together. Pilots, the scientists, the crew, within each team and across each team, to make sure that all of this is possible. And it's all good-natured fun, which makes this truly enjoyable, which is extremely important on a 34-day mission. It's a lot of time. After everyone's been alone for a year and a half, all of a sudden to have all that time together on a ship is such a joy. Wow, it's so little.
Set up some Nicholas like right.
Oof. Thank you.
Oops. Slid off. Hold on. Zoom off. I'll get you right on.
No worries. So one of the things that we're looking at on this expedition is coral associates, so organisms that are associated with coral. And we use the word associates because it doesn't indicate what that association might be, whether it's symbiotic or parasitic or predatory or commensal, because the ecology of these interactions aren't well known yet. And one of the questions and hypotheses that's out there is whether or not associates find corals when they are young and when the corals are young, and then they grow together. And I don't know the answer to that, but this is a very tiny coral with a very tiny associate. So either way, they found each other early. Aww. And so I see a comment in the chat. Yes, it's true. It's fun, but we are not playing. This is certainly not play. This is extremely, extremely hard work. It's months, if not years, of preparation, and then we work around the clock. We're all completely sleep deprived and exhausted. But I guess my point is that a sense of good humor and people that you enjoy working with makes the time feel a lot more pleasant and a lot more fun. And there is an element of scientific play and curiosity that keeps all of this really fresh. Wow, look at how close we're able to get.
Yes.
Yeah,
Good. Fun.
Let's see if I can get some better zoom here. Here we go. Look at this, Anna.
The ring wasn't designed for the hook. It was designed for the bunch.
Oh, it's so tiny.
It's another one of those things.
No, that's a sea
Oh,
cucumber.
that is.
That's a halibarian. This thing has like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19.
If you white balance without
43 polyps. That's a very small coral.
Oh, it's just getting started, maybe.
I know. Can we get, is there any, hi Russ.
What would you like?
Lasers, and you. Hi. And you should say hello. Here's a little watch change. We've got Russ in the room.
So what will happen is, because the laser, you've zoomed in quite a bit.
I know. Let me zoom out quite a
Yeah, if you
bit.
zoom out, you'll see the lasers just from bottom there.
I don't see him. No.
Yeah,
Yes, I do.
yeah,
Can we bring the lasers up so that they surround the base of that teeny tiny coral? The urchin also helps for scale. This is very small. This
It's quite
entire
tiny.
thing is like less than 10 centimeters. Yeah. Okay.
Which, imagine how tiny that squat lobster is then, if that's 10
I know,
centimeters.
it's so little. All right, well, that was a really valuable observation. It's so little.
I like the Quidditch next door though.
Fair enough.
That's
Yeah, right. Thanks, Ben. That was an awesome collection of play.
So is this another one of those?
I don't know.
orcs,
So
though?
we're going to take a look at this little we're calling this a UGB, an unidentified gelatinous
We collected
blob.
two.
Alright.
We've already collected 2 of them, but we're just going to take a look. If you two know what this is, since you see a lot of things on the seafloor, we can see some structure here. Ooh, 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 ooh.
Hey, that looks like an anemone. Look at
No,
that.
it looks like a tune. Oh, no.
Interesting.
What are you? What are you? All right, let's take a better. I don't think it's an anemone.
Could it just be like a very young Holothurium?
No, it's attached to the wall.
Oh, okay.
We pulled 2 of them off. Now I think it's a
You know what you could use?
tunicate, because you
I've
see
used
there's
it a lot
2
of times. It's amazing.
opens. Yep.
Good old Google.
Yeah, I agree. Hold on to
Eyeball.
What is in it? Is that like
Well, so this
If
is
it
this
is
is
a
this is a
tunicate?
gut,
okay? That's what I was
right?
wondering.
And so it's full of sediment or whatever it's eating, and then there are some. I mean, this is its internal intestinal structure.
Okay.
So I don't know what it is, but I feel much better now that we have this imagery. I think there's two holes, which is what's making me think tunicate or potential, right? So you see this one here? I think this is another one right here above it, right? It's really hard to see, but it's.
Yeah.
I don't know if you guys can see this at home because it's very, very subtle. We have an incredibly good screen in front of us that gives us we're looking at this in 4K, and it gives us a chance to see things with great detail. But the pygmy hornet is eating this. Adam, have you seen this before? Do you know what it is? Sometimes the pilot sees things that you guys see things with all different groups all the time. Well, we've collected 2 of them, so we'll figure it out. Oh, Steve's typing. Maybe Steve knows. And then we'll move on.
Okay.
Okay. Well, let's.
You sent your camera, that's right.
Yeah, that sounds good, and I think we can go. Thank you very much. So Steve also thinks potential tunicate, so I feel better in that guess. So we'll see when we look at it, but that's 2 votes for tunicate, 1 vote for ctenophore, and
We
many
didn't
votes for
see the
unidentified
mouth, though,
gelatinous
with the
blob.
other 2 though. That's the thing.
I think tunicate's I think right. Well, let's take a look at this one. That is another. That's a solitary predatory hydroid, and these are amazing. I love looking at these.
I don't think I've seen one of these yet this trip myself, which is not
Oh,
saying
they're
much,
just
but
gorgeous. They often have a little isopod or a shrimp inside of them.
I
Tell me
actually
when I can zoom all the way.
Yeah, just.
Yep, that's why I'm not going for it yet.
definitely something in there.
Your ISO is on the upper left knob.
Thank you. How did you know? And I'm reading my mind.
Okay, I believe we are stable ish.
All right. Oh, beautiful. I love these. Maybe they'll just turn towards us and wave.
Is that part of it, or is that
Yeah.
an anemone in it?
I can't quite tell. I think it's part of it. Yeah, it's part of it. It's sort of got this internal structure to it. But they're just stunning. All right. Gorgeous. And then while we're here, you know what I saw behind it? UGBs, 2 of them. Let's take another look. Maybe one of these days we'll figure it out. And then I really will move on because we have a lot of other things to explore, but it's bugging me that I can't get a good view on this. That's a tunicate. Look, see,
That
here's
is,
the 2
yeah,
holes.
yeah.
All right, it's some kind of tunicate. It's not the classic predatory tunicate, but it's a tunicate. Good. I feel much better knowing that. Let's see if this is the same thing. Now we're getting some structure. Yes.
So close. Oh,
Very clear.
yes.
Oh, there we go. Our UGB is some kind of tunicate. Good.
Says it has the commensal polycate
polychaete inside.
Yeah.
I'm not sure if it's a commensal
that's
polychaete
true.
or if that's just. I mean, it might
It's
be
internal
because that's not
structure.
what you typically expect to see in a tunicate. So, all right. We have some kind of tunicate on the bottom, potentially with a commensal polychaete inside and a hydroid to the left. We're good. Woo! Let's keep flying. Thank you. That was great. Let's see if he's turned. Nope. Still going the wrong way. Alright, let's I'm sure we'll see another one. So it's so interesting. We're on the opposite side of a seamount that we dove yesterday, at roughly the same depths, and we're seeing very different organisms. We're just on the north side today, whereas we were on the south side yesterday, and so it is pretty cool to try. This is a great way to try to. There's I love those stalked crinoids. This is a great way to try to dial in on why things are where they are in the seafloor. Okay, so we see some crinoids as we fly up this little mountain. Maybe it's not so little. Not so little. Let's climb the mountain.
Climbing away.
Okay. Sorry, Anna. Oh, can we take a look at that little pink coral? Sorry, it's just below me. Did you see it? It's very slight. I assume it's a bubblegum coral or a hemicorallium. But I just want to take a quick look. There it is. You see it?
Yeah.
Teeny tiny. Oops, wrong way. I'll get better at this.
Okay.
All right. First, I'm
There's
gonna go
an
look at this thing down
Anthomastus,
here
too.
first. Yep, good call. Anthomastus. I'm gonna come back and look at that. First, let's look at the coral. Hard to tell at this angle, but it looks like a corallium. I'm sure it's. I'm pretty sure it's some kind of corallid. It looks pretty rigid. All right. Then let's scroll down here and take a look at a couple things to look at. That's a crinoid. And where did it go? This thing. What is this thing?
A large encrusting sponge or a rock sponge?
A
Oh,
large.
you mean on the sponge?
No, I mean. I just want to make sure that's a sponge. It's a
Yeah.
sponge. Okay. Cool. And then wasn't there another little coral? Yeah, an Anthomastus. We'll just zoom on that, make sure that's correct, document it. And then
Oh, there's one above it too, whatever
Yeah.
that is.
I'm coming. Yeah, it just goes to show you, right? We think, oh, there's not much here, and then a quick little zoom in would show
Stalked crinoid.
that there's actually quite a lot here. It's just a little. This is a very small Anthomastus coral. Just a few polyps, really. It's starting to grow. And then here we have a hermit crab and Yeah. Ow. Just land a little. Little anemone.
Oh, he has an enemy on his shell.
Yep.
And it's not a decorator crab either. It's a hermit crab.
You're right. It's a period. Okay, that was great. Thank you. Another urchin. I'm gonna keep climbing this mountain. Maybe I'll just zoom in a little tighter though so I can see what we see because there's quite a bit here. Hey,
Good morning.
good morning. There's a lot of these tunicates. My goodness, they're everywhere. You see them? These UDBs, well, some kind of tunicate now. They're absolutely everywhere.
It's funny how you can go so long without seeing something like that, and then you see a million of them, and you wonder, is it?
Was it there the whole time?
Yeah.
I know. Well, luckily we have the footage. We can go back and look. You know, I thought that pycnogonid eating one. I'm not sure we would have stopped to look, but now we know that a picnic. Well, we can assume, given the shape of that tunicate and the pycnogonid right next to it.
Yeah.
I'm not sure it's entirely. We didn't see it happen, but it seems reasonable to suggest that pycnogonids can add this tunicates to their predatory list.
It's definitely a hypothesis at the very least.
Yep. We are 1,616 meters below the surface of the sea on Titov Seamount. We're going to take a look at whatever this is right here. Thank you for anticipating my every need. Is that rubble? There's a fish.
That's what I thought you meant was the two fish.
No, I wanted to see what this schmutz is on the seafloor. Yeah, can I do this now too? Even closer? Okay.
Another tunicate.
Not rubble. Okay. Okay, thank you. That's great. Another Anthomastus.
His eye.
Let's keep hiking up the mountain. So again, we kind of were glancing at the chat while we go, and you're absolutely right. Somebody awesome will get to count the tunicates or really go through this footage. It's really important for us to acknowledge the fact that we have an incredible team of very talented students back at home who kind of cut their teeth, if you will, on deep sea video in order to get their taxonomic identification chops, and they do a great job. You go from starting as a freshman and knowing really nothing to then taking marine biology. I mean, you know something, but not having really worked with this kind of footage before. And by the time they graduate, they are really experts in deep sea taxonomy, and they know how to analyze footage. But we're going to take a look at this right over here too. I think it's a big scale worm, but I want to get a closer look. They learn how to analyze data, and there's a lot of sub-questions within this footage. And so we are not just deep sea footage, they help us sort plankton from this area. That is a scale worm, it's cool. They help us identify shallow coral reefs and look at photomosaics and ID corals. So we rely and count on our collaborations with large teams of undergraduates to they get to learn how to do this kind of work, and we're very grateful for their support. So this is a big team of people, not just on ship, not just at home, from the Schmidt Ocean Institute, but from all of our universities who help us turn this footage into a scientific discovery, right? So papers and findings. Thank you, that's great. That will propel all of this into what I always say, go from a qualitative world to a quantitative world. Climbing the mountain. Thank you. I think that was sarcasm. All right. So I keep just talking while we're looking for corals and sponges. I've been noticing these like mucus nets for years now. They always have these like triangular configuration. You guys see that? I've always wondered what kind of critter makes them. It's got to be some kind of like worm or something, and I'm sure they're casting that mucus net as a way to capture food particles, and then they'll probably suck it all back in, reabsorb it, eat it, and then cast a new net. But I don't know how
Sounds
to.
lovely. Process, just in terms of your description.
Oh, it's. I mean, it may seem gross to us, but it's really a very effective fishing net. Mucus is like super sticky and it traps stuff. I don't know. Doesn't seem like the worst way to go to me. You don't have to deal with anybody else's stuff that you have to suck out, right? It's just your own. I'm kind of good with that. Oops, wrong way. So we're not geologists. So there's a little masses coral. So we can't answer too much about the geology of this, but I can tell you that what you're looking at is a ferromanganese encrusted basalt, because we're on the side of an ancient volcano, probably in the 80 to 90 million year old range. We're going to look at this coral very quickly. And one of the things we have been doing is collecting rocks for our friends at the U.S. Geological Survey, who will help. That's a unbranched bamboo wood coral, so we're good.
I'm gonna go and fly closer.
Oh no, yeah, we can, but I'm also okay. I know what it is. So they can tell us a little bit about the geological history of these seamounts, the mineralogy of the crust, and help us infer something about seamount age and seawater chemistry, and maybe even tell us a little bit about why one side is different from the other, which might give us some insight as to why, even though everything looks rocky to me, like there might be some chemical differences here in the rocky crust substrate that makes it great for corals on one side of the mountain and not so great on the other, or whatever. So there's a lot of insight to be had here geologically that we don't have because we're not geologists, but I think, again, the phone a friend strategy.
Yeah, I mean, it doesn't look. We were talking about the shape of the rocks yesterday and how that was a potential factor in recruitment, but these look just as
Yeah. And
lumpy.
Speaking of which, we might want to see, when was the last time we collected a rock? It's been a while. We could collect a rock. There's a lot of loose rocks here.
There's one there, so the next one.
Which one?
One right, big one right in the center. Center screen.
Okay. So pilot's choice, let's collect a rock. We have to get some really good imagery of it on the
Something about
seafloor
that rock.
before we take it out. That's okay. Oh, perfect. So our protocol for collecting rocks is to get a really good image of it on the seafloor, look at it all the way around, come up with something descriptive about it. You're talking about this one or this one? This one? Or this one? Or this one? Or this one?
The smaller one.
This one?
Nicer.
Hit that.
Right there, yeah, there you go.
That's our rock. Can we get this one?
Yes.
I like that one.
Okay.
I actually quite like the one that was kind of hexagon looking.
It is hexagon looking, but I like the
Alright.
botryoidal.
We're going to Predatory.
I said pilot's choice though, so if you're really feeling attacked by this, I can live with it.
Women first.
First, Randy?
What? Yeah, let's image it first. Okay, let's take a look. See, now you know what these are. We've got another. Focus.
That might not be your best choice. It looks like it's attached.
That is certainly attached. Okay, let's go to the hexagonal rock. Russ's choice. What do you think, Russ? Is that going to come up?
I think it's kind of like a thought.
That one's attached too. Looks like on the bottom.
What about Adam's choice?
Straight down. Right center, right there. That one might be attached too. Now that I look at it.
I don't know.
Let me
Let's
poke
get-
these
let-
rocks and see which ones aren't
That's a
attached.
good idea. Poke the rock. You have reached the official rock poking hour.
that's alright.
Oh.
Oh,
That's good.
That was good.
Can you see if this one is this too big to pick up?
This was your original one. Here.
Yeah, let's see if we can get it.
That's,
Oh, yes.
yeah, there you go.
Is it pick upable?
Yeah, I think I grabbed the nub on the top.
All right, let's give it a shot.
Do you want to image it first, or? I guess I
Yeah.
just stirred up the dust there.
well, you can pick it up and bring it in front of the camera.
I think we're supposed to image it on the sea floor, so let's wait for the dust to clear for a minute. You know, which is also informative, right? You can learn something from everything. Look how finely silted this is. This is not a heavy sand at all. You know, and I think part of that is that what you're looking at is not just sand. We're looking at all kinds of things that fall in the marine snow, and you get these really fine silty sediments, partially which have been ingested multiple times by different organisms, like sea cucumbers, for example, and you end up with we're sort of repluming some of this, which it'll fall back down, but it's going to take a while because it's so fine. So when we re-release it, it does take a little bit. All right, I think we're ready. I'm going to zoom in an image. I think I can get a good enough image of the rock. So the reason we want to image it really well on the seafloor is it helps us to figure out later which side was down versus what came up. So I think we've got that. You can really see that this top part was sort of buried in the sediment before we poked it, and now we can pick it up. Thank you. This is going to be in that FK399. Oh, great. And if we can take a good look at that while it's in your hands, that would be good too.
Yeah, that's all you.
Okay. That's terrific. Thank you.
What was the bumpy texture on the top? Would you call that bumpy texture? Or?
I would call it a bumpy texture. Great.
Is that manganese? What we're seeing, the red?
I don't know. Some kind of iron, probably. It looks oxidized. Perfect. They look like such big, strong hands. You've seen the movie, right?
What's that?
The Never-Ending Story.
Oh yeah, long time ago.
Oh, come on.
Back in the day when he looked young.
We are on the Falkor, which is named for the luck dragon aboard the Never-Ending, or aboard, jeez, I'm tired, in the movie The Never-Ending Story, and in fact our ROV is named Sebastian for one of the main characters, and we're quipping about the rock biter, which you guys are too, so we have it. Okay, let's go flying. I have a 15-minute meeting that I'm going to step out for, so I'm going to leave you in Anna's very capable hands.
Which means Russ and Adam, can you fly? Because I haven't flown before.
There you go. Have a good time. I'll be back.
Do all the zooming. You have amazing tracks. 1, 2,
just sighted past the flyby.
Yeah.
I still see stuff though.
Right, we're gonna go a little faster just because we wanna make some tracks. We have not made it to our first waypoint yet, and we have been in the water since 3:30 this morning, so hang on to your seats. Here we go. I just did see a seat back there. It's crazy how different this side of the Titov is compared to our track yesterday. It's night and day. Then again, I did hear at the beginning of the dive there was a lot less than when Tim and I came in at the end, so could very well be we just haven't reached the biodiversity or abundance yet.
Yeah, it just depends where we're at.
Urchin. Yeah, looks like it dead as well.
Sponge.
Yeah. Oh, Chrysogorgia. Gorgia. At least I think so. Do a quick flyby. Yeah. Another stalked crinoid. Yeah, and an umbellulo. Yeah, it's the land of the stalked crinoids. It's happened to us when they seem to be everywhere. Maybe a bamboo skeleton.
Oh.
It's like glass sponges, encrusting sponges. So we're making headway just in hopes of maybe Seeing more biodiversity here. Got another tulip sponge, glass sponge with a crinoid attached to it. Is that? Oh yeah, look at that. Nice big Iridogorgia off to the left there. Those are always quite beautiful. I mean, they're so delicate. You can get them up into the lab too. I mean, they just look almost like a flower, like baby's breath. Plexaurin, Nostroschema. There we go. Here we go. This is a good sign. Is this a black coral? Yeah. Good bathies. They're increasing our diversity here.
Is that?
Yep, all set. Thank you.
I just gonna do a show.
And make sure someone can ID it correctly later. Just get close enough. It's just,
It's a big thing.
I don't know.
Quidditch.
Isn't that
Yeah, it's a snitch.
Yes.
Oh,
One of those things.
it looks like a little jelly. Yeah.
Collect it?
No.
I'll flip that thing up. Looks like a ghost from Pavement.
Does actually
Hinky winky.
Mm. There's another one. A jelly.
I know, it should definitely go on, because like
Yeah,
there's two.
there's plenty.
Do you want to take it out?
That's fine.
I was like, I am not going to touch a jelly that's potentially going to sting me.
Last week or also.
I'm here to have Australia. I'm not here to anything around
No,
here.
no, no.
Yeah.
Everything we picked up is dangerous. Even sand. You go on land and everyone else wants to kill you as well. So it's like, it's
It was a jelly. No Dumbos yet this time, I don't think, but correct me if I'm
wrong time of day, they're sleeping. Yeah.
Yeah.
Don't come out to lunch then. Yeah, I don't have to look where last night. It's 24 inches away. Country, clear to the bottom. Also, it's Grey Goose and Ice Cubes.
Those things are
Oh, I like to see chicken out like that too. It's like a old fast. Building.
A whole fast or a sponge?
Looks real nice.
Looks whole fasty.
Yeah, it does do that.
I was thinking on the other side. What did it? Oh, that's a sponge.
There's a little coral going to be growing there, I think. That one?
Oh, the Anthomastus, yep. Nice. Good eye.
fishy thing.
Yeah, yeah. Fish.
Yeah, I like the Anthomastus. They have that nice bright pinkish red color. And they're cute. They're scoring like little balls, which I wonder what evolutionary trajectory that was compared to the other soft corals. Filled some niche. Sponges.
Yeah, southeast and have driven behind it, but the current is blowing to the northwest. So which would be for
I think bringing it down that was
Alright.
good because then when we get down to 8.1, it's a straight
shot.
from Australia. Like that better. Yeah, well, honestly.
Bridge ROV, that is a new waypoint for you there.
New waypoint, thank you.
Yep. We'll leave that on this.
Fair enough.
Come on, Banter.
When was that you seen the broken piece from the lab, man?
What? A broken piece of glass? No, I've not actually seen it.
Corner. Giant blue.
Spongivory?
The lamp.
Oh,
Someone's playing.
What is it?
One of the corners of the lamp. I cleaned it up and it's broken.
Oh, really?
They have to find the broken piece. Hopefully, then throw it out.
What's our incline? Are we steep right now?
we look down or
Yeah, it's not bad.
Is that a little hold fast? Yep. And a sea cucumber, it's always exciting. I don't know what these little floaters are personally. I'm sure someone does. I'm still a learner when it comes to deep sea ecology. I'm more experienced in microbial biology and molecular techniques. But this is my second cruise and I feel like I'm finally getting my corals down. Some of the more obscure
trying to get
things
that up out of your way. So you don't get smashed into it. It's moving
I am
towards you.
still picking up on.
Bye.
I'm
seen
panicking,
her going
it's
though.
like, well, give me a second, I'm gonna get your wife for you so you don't smash it anymore.
Anytime I see a funny rock shadow in the distance, I'm like, ooh, I bet it's a rock shadow.
Yeah.
The older I get, the more my eyes do play tricks on me, I will say.
Yeah, there's many other glasses outside.
Never happens to me.
No, but you got 10 pair of glasses. All of these different focal lengths.
He broke one side. Maybe I'll let
That's an expensive prescription outside.
That's why I bring 6 pairs.
You never know which one you might need,
That's right.
or what combination of ones you might need.
Absolutely.
And he's got style, he's nice, so he's got to be trendy. So he's got different ones, different days, different weeks.
Yeah. Hey, well, glasses have become a little more affordable, at least in the non-prescription range, the readers. It was like, you're like, yeah, right, for mine.
$500 a pair.
You have very nice glasses, though.
Yeah, not bad.
a pretty stocked crinoid, bright red. Sunflower. But again, I mean, these are pretty common, so. There's plenty of shrimp in the water column. Looks like we've got another sea pen and a malula, which are beautiful and great. But it seems like things that prefer sediment, settling might be favored here. Or at least more abundant.
Phone should be turned off by working with Fauna.
It's still on. Hello. 7 feet away.
5650. That's the main one we have. It should be, I think, a 16. It's 16 port triple standard, so it's 48542.
That's on the
Sorry, what's that?
I don't know if you can hear that in the background, but that's our lead marine
The end port.
Jimbo.
Yeah, end port. Yeah, 5600. Yeah.
Awesome person to have on the boat with you. The ship, excuse me.
Yeah, what if you ask that question?
That's like the most obvious question ever. Why would you even ask that?
What's this one?
Nothing we were talking about.
16 port? I mean, I would go for 32 personally.
That's not an option, they don't make.
We do the mix 64 port now.
64 port triple standard boxes. Are you really serious?
Yeah, well, you get
Alright,
128,
okay.
so maybe
I
that's
think you're talking for the ship, but then.
Hey,
guaranteed is 128 now.
No, I don't think so.
Well, 256.
Probably.
64
I actually do. It's got
I
extra
don't
RAM.
think it exists. I think more.
I think it's got double RAM.
You can say
Yeah.
full of it. That's fine.
Yes.
Just because it has a minute to your alligator doesn't mean
I know, exactly. Either way, it's like just barely got DSL.
Yeah, tons of small rocks here. Whoever said that compared to our landscape yesterday, it did seem like there was more continuous flow. But you know, the second I'll say that, we'll get out of the small
Sorry.
rocks and.
It's just one of them things.
Tim was doing something yesterday where he was wishing for things and then they'd show up. So maybe I should wish for something like.
Gold bullion.
Yeah,
Great
I don't know
Pacific
if money
Narwhal.
works. Yes, we have been looking for the Great Pacific Narwhal for years now.
Shockingly, never found it.
But I'd settle for a giant squid.
I would too. That would be
Very-
awesome.
very considered to
Oh, yeah, it's just right
Yeah,
there.
I try to be reasonable.
Oh, well, giant
Well,
squid.
instead you can have a cucumber.
There's loads
Yeah,
of them.
that's
It's not gonna cut it.
Loads of them.
Sea
Get different colours.
fever.
Very good at cleaning sand about it. Well, if you want to do a sample of rock, I think the time is now.
Oh, well, tell me if you see any particularly like good-looking one that you think would have a center that is desirable that won't crack upon grabbing.
Thank you, Russ. I wasn't able to find a 32 or 64
You found a 20.
port boxer, but I have found another boxer in the port with multiple Ethernet switchboards.
See?
And I didn't know they could do that.
Exactly.
Again, just because you don't know.
Again, just because you haven't used it doesn't mean it doesn't exist.
But what you said doesn't exist. So
No, not in that brand. You have to go to Amazon. You can get it in there.
Oh, really? Okay.
Oh man. Yeah, so we're just saying if this is we want to do rock collections. We've collected 2 rocks so far, and we have slurped a potential tunicate. But that is all we have collected this dive so far. We can probably fit at least 4 rocks, depending on the size in our rock basket. So,
Or one really big one.
or one really big one, but we already have 2. Could also try again for one with an encrusting sponge on it. That's something another member of our team is interested in, is the sponge diversity in these areas, and it does seem like we have a lot more encrusting sponges in general in this area than where we were at the end of the dive yesterday. Because it actually took us a while to find an encrusting sponge on a rock that we could collect. But we did find one.
So,
I think we could talk about this. No, it's not.
I like that. I do actually like Thunder's butt.
What's that?
Can you top
We're
off
looking
that?
at rocks.
It's a
Can
bit
you
like
top
this.
off that?
Yeah.
You want you draw?
Without a hacksaw woodwork?
Yeah, this is our basket sheet. 2 rocks and 1 slurp.
Well, you wanted a comparison. It's a pretty stark comparison.
Yeah, it's
Yeah.
really stark. So,
It's
but
the
we
second.
have not made it to our first waypoint yet.
I know.
So,
It's a
which-
little long.
I wasn't here on this.
But we have 6 hours left. We'll see.
yeah, this is a longer dive.
Yeah.
That's it's only calculated for the next job. No, it's not.
What are we doing?
I just put it on the hook, just it's dragging it down.
Oh, okay, good. I was like, did I request this? I don't remember, but I am
I just
tired.
did it rather than drive over it. It's,
Gotcha.
Yeah, it's not all about.
It's gonna be spicy today.
Yeah. I like don't even have a comeback. I don't even know what to say to that. I'm just in shock.
Did you sprinkle something new in your oatmeal this morning, Ben?
Yeah, whale fall would be something I personally have never.
never
Oh,
bring,
it's
never
such a
bring,
horrible,
do
horrible
not bring
smell.
the bones up to the ship.
No.
Do not cut them with an iron grinder.
Oh my. You'll never
Okay.
appreciate
Not
that
very
smell.
nice smell. It's rather horrific.
Was it you, Adam, that?
Adam did that.
Yeah.
Cut it with a
When was that you?
that grinder never.
It smelled. It
I
smelled.
just like to observe it on camera. Let's put it that way.
Best way.
Yeah.
You don't think culturing bacteria from a whale fall would be
If there
advisable?
was worms in it, they were terrible.
Ugh.
Did I cut it? Yeah. Yeah, I cut it with the chop saw.
It was awful.
It still must be.
The burnt hair smell.
It's
Oh, but yeah, it was worse than that. It was worse
Oh.
than that. It was
Oh.
something else.
Oh. That's years ago.
If you guys can't hear that, one of our ROV pilots, Adam, had the distinct pleasure and opportunity to
Madness.
cut some bones from a whale fall, and it was
Hideous.
been decayed for quite some time, and it smelled pretty bad.
Pretty bad. Scale of 1 to 10, I'm about 84. Maybe. What's that?
Oh man.
What would you say that last rock was? Flat?
I would say it was probably flat on the underside, maybe I would certainly ripple on the top. About, I don't know.
Look at you.
Yeah, it's
Another rock, huh?
Do you want this one or is it too big?
I think it's good.
Should we call these the same sample number or give them separate sample numbers? Same location.
Same location. They say different things on this one.
Okay.
I don't know what's going on, so you're in charge.
Yeah, I know.
I just came in after spilling coffee all over myself.
I'll do another number. What do you think is on this one that defines it as different? Just so we can.
Growth or something like that.
I think it's got some.
And on the upper left knob is the iris.
Got it.
There's definitely a U-shaped growth in there.
I don't know if that'll stay, but I think you can definitely see the white encrusted spots everywhere. Okay.
So 400 was the first rock, and this is 401.
Oh, you got 2 right here?
Yeah.
Okay, no more rocks. That's it. We're done for the dive. Not because that's our permit conditions. 4 rocks per dive. That's it.
Yeah.
That's it. Perfect. And this one is white spots. I didn't see the first one, so you'll have to tell me what the first one looked like. Okay.
So the first one was a flat rock that was slightly rippled on the top.
Okay.
Oh, I got it.
Cool. If you're exhausted. Oops. Hang on, folks. We're just going to take ourselves offline for a minute as we get through a little bit of a watch change and just convey some information, and we'll come right back.
Dive track is a little long in the beginning, but I thought it would be over sediment. But
No, it's been rocky.
that's what I'm surprising. That this is a second dive on these north-facing ridges. Barren.
it's interesting. You know, Chris Moss sent me a note saying that the pygmagonid observation on the sea pig might be new and worth a note, which is pretty cool. And it looked, that tunicate that we saw, I'm pretty convinced it's a tunicate, but I don't think it's the classic predatory tunicate we've been seeing. I think it's a different kind of tunicate.
Yeah.
I don't know if you've seen it before, but the pygmagonid feeding on that is also...
Oh, I didn't need to see the picture
Oh, yeah.
on it.
So, well, we didn't see it feeding on it, but we saw one, that the pygmagonid right next to it, and it was very clearly not okay. It looks like it had been eaten.
Yeah.
So I don't know. It's going to be interesting to see what we see, but I think we're getting a lot of pygmagonid behavior and a little bit of their generalist feeding habits and ecological observations, which I think is really interesting.
Mm-hmm.
Yeah. So that's cool, and I think it's valuable to understand as much, like, as we always say, right? How much isn't here as much as there is how it is. And
It is. It is. But I
different
also thought
stuff.
that, but even, so when we were diving yesterday, we popped down,
Oh, can I take a little look at this price of please?
we popped down at, in a similar terrain, and if we'd gone off the ridge at all, 30 meters one way or the other, stuff dropped away. So I'd be curious to see what the bump looks like.
Yeah, I think it's pretty great.
Yeah, I think it's going to pick up terrain a little bit.
It'll certainly be interesting, and we've seen some stuff. Like some of the, I don't know if we've seen a solitary hydroid yet this cruise, but it was the first one I'd seen.
Oh yeah, yeah. They're rare, but yeah, we see them once in a while.
Yeah, so but I hadn't seen one yet this expedition, but I used to know if it happened and I was asleep.
No, I haven't seen one.
We
Either.
saw one. We've seen those tunicates. We've seen a lot of pygmagonids. We've seen. I saw a really small. 30. I think it counted like 35 polyps or something like that. Chrysogorgid with a very small associate, which was really a pyrostylid. So it was really cool to see that. Ooh, I can't get focus. So it's been, while it hasn't been Coral City, it certainly has not been uninformative. I would say there's been a lot going on.
It's really interesting from a habitat suitability perspective about what can live where and why. I think.
Totally.
Yeah. I mean, this seamount probably more than any other is pretty clear that if you get on top of the ridgy parts, even like straight on top of them without having any to look over the side at all, that's where all the stuff is. And I'm very curious as a hermit crab.
Yeah, we've seen a lot of hermit crabs too, but this one's in a different shell.
There's a black coral right there too. Right?
Oh yeah, there's been a lot of little corals, little corals. A couple of Emballula sea pens too, and we saw a baby stalked crinoid. Oh, you're right. Oh, goodness, it's so little.
I can't tell now. I'm doubting myself. It is some type of...
don't know.
Or crinoid? Stalk crinoid? I'm not sure.
I know this sounds crazy, but can we get close to this teeny tiny itsy bitsy little thing from the max zoom? Yep. Use the hermit crab for perspective.
So habitat suitability seems to be like right on the rim of the seamount and right on the rim of the ridges.
Yeah. Right. I'm like dancing for you. Happy. There's a mucus net. God, again, showing your prowess, pilots. We have Adam and Russ on the controls here today, and we've asked them to look at something that's probably about 2 to 3 centimeters. Not certainly, if that, 1,494 meters below the surface of the sea in the absolute remote Central Pacific. and they're getting it done.
It's
I
a
can't
little bit
even
easier with
believe
a 4K camera, but not
Can I
much.
zoom in? Am I good? I see it. Yeah, I got it. I just didn't know when it was ready to zoom. It's right here. I just gotta get the focus. Thank you.
I think I'm leaning more towards stopped crinoid.
Me
Yeah.
too. But let's, and just in case it's a baby black coral, let's take a look at it. Even if it's a baby stopped crinoid, right? The ability to see something so little. It's just amazing. I agree. I think it's a crinoid. Look at
Yeah,
that.
I don't know. Well,
I was just worried that was
I
occasionally it stops.
What's the
don't.
what's
I don't
bothering me
even
is
know.
this.
Yeah.
Right?
No, I don't know.
What's that?
Cool. Thank you, sir.
I mean, it could be a hydroid. I have no idea. Honestly, it's so small that I can't make out really any characters.
I don't know. See, but it's that extra wisp.
Yeah. I mean, it could be a coral because it does have some sort of polyp structure that I can't make out any detail on.
But look at
Could be a bryozoan as well.
what's amazing to me is how much structure there is for something that frankly is like the width of 10 grains of sand.
Yeah. Yeah, it's very small.
It's very small.
Yeah, I don't know. I'm also now leaning towards bryozoan, so the fact that I'm switching my mind every 30 seconds doesn't really offer a lot of conclusive support. So
No.
your call.
I'm still going with crinoid, but I'm perplexed at this little, I don't know, branched tail arm
Yeah.
thing,
Denuded, possibly.
or growing.
Mhm.
Well, it shows how much we know about the deep sea. Let's keep going, shall we?
I have no idea.
I love that. We've had a couple mysteries. Look at this. It's basically the same size as this pteropod gel.
Hmm.
It's ridiculous. Just for perspective, everyone. This is how teeny tiny this thing is. Here's the hermit crab. See, blink and you'll miss. You can't even find it. Oh, but there's a fish. Should we look at the fish? We should look at the fish.
Sure. I didn't even see the fish.
There's a fish. Hey, fish.
Looks like a small synephobranchid cutthroat eel.
Cutthroat eel. Why are they called cutthroat eels?
I think it's when they gulp. When they gulp,
Ah.
the
What type of eel was it?
cutthroat eel.
Now the cutthroat eel, do they not also use mucus bags to store their egg sacs and stuff like that?
I wonder.
'Cause I remember we were doing some project before, and they were mentioning about these little sacs, these little mucus membranes, and this
Mhm.
cutthroat eel was actually producing some of this.
It could be. Could very well be. Yeah. I don't know enough about fish
Likewise.
to speak informed about that.
Alright, I'm just taking some notes. And very teeny tiny. I'm gonna write crinoid question mark. Okay. Let's go flying.
Sounds good.
Thank you. We are making tracks. We're almost at waypoint 1. Which,
It's
which
it's
says
more than
something.
half the dive. Yeah.
Yeah, but that says something because for such a sparse habitat, we keep finding things to zoom on. It's just that it's more subtle.
Yeah.
And I actually think that subtlety is there's a lot to be learned in that subtlety.
We
I'm all
started
for it.
at about the same depth as yesterday, maybe 100 meters deeper, but yeah, just the difference in terrain. We're not seeing as much cohesive stuff here. It's still a lot of talus, I
Mhm.
think.
Is talus the word for unconsolidated rock?
Talus is the rocky stuff that builds up at the base of a slope
Oh,
when you have stuff that falls off.
you've been hanging out with geologists too much.
Yeah, too much.
But that's great.
It puts my work into perspective.
Tallus?
TALUS, I think.
Okay, great. Thank you.
Yeah.
I didn't know that. So it's the rocky pile at
Rocky
the bottom of the
pile
mountain.
at the bottom of a slope. Yeah.
Got it.
Yep. And
Learn something
so
new every
we
day.
are approaching a slope on the bathymetry. Pretty sizable one, probably the biggest one of the dive so far. It would be interesting to see if this turns into some more consolidated vertical structure.
We're gonna take a quick look at that coral, guys. That's okay. Teeny tiny. Just to make sure we can identify it. It looks like another unbranched bamboo. We've been seeing a lot of those. But with Steve in the room, he can confirm.
That's what it looks like.
That's what it looks like.
Mm.
And
There's also
look,
primnoid
branches.
primnoid
Yeah.
branches there.
What?
Those are definitely fallen from somewhere, so that bodes well for our
And they still look
wall hypothesis.
alive.
Mm-hmm.
And that's another one of those tunicates, just so you know. Which we were calling
Oh,
an
yeah.
unidentified gelatinous blob, a UDB, for a while, but now we know they're tunicates. But they're not the same predatory tunicates. Just here.
Did we get one?
Yeah, I got two.
Okay.
They're very cool.
Yep. This is Narella here. You can tell by the sclerites how they're arranged
So
on
what
the body.
clipped its branches? And
Don't
let's go
know.
find the colony. There's so many mysteries. Let's go. I'm excited.
Narella. Crinoid.
Where did it even fall from? It's such a delicate, wispy thing. You'd think that the colony would be close by, and just uphill.
We were so we sat down yesterday. We were manipulating a rock in the beginning, and we thought it looked fairly flat. And the rock slipped out of the manipulator, and it fell down probably about a meter or two before it was caught on another rock. If it wasn't caught on that rock, it would have been gone. Just gives you some perspective of how steep some of this terrain is and how important it is to handle collections delicately, which isn't always easy. But
we have the best pilots in the
expert
world.
pilots. Yeah.
Yeah, but still, I mean, those wispy little things, and there were 3 of them, and they fell together. So I find this implausible, unless somebody picked them up and dropped them.
There's a coral over
Yeah,
there. Maybe a
let's take a look.
crinoid.
Maybe we did it and didn't know.
I don't know.
There's
I'm
no
just
way
seeing
we
a
could've-
branchy thing over in the
Yeah,
distance.
I think it's a crinoid. So,
Yeah.
speaking of our amazing pilots, we have Adam and Russ in the room. You guys want to say hi again?
Hello. Hello.
I already did this, and I'm just that tired. I apologize. But always happy to hear your voices. And we have Jimbo, our amazing marine tech in the room, who is managing our live feed. Say hello again.
Hello.
He's also the world's biggest Beatles fan.
That's the stare heard around the world right there.
He's got lots of friends in the space station. How would you Jimbo?
Did you tell everybody how much I love Windows?
Yeah, he's a Mac fanatic. He loves the Mac.
We love Jimbo so much. The only reason... Ooh, there's a big fish.
We love to talk about fish. Well, I see how your priorities are.
I was trying to find
You
a
don't
fish
even
for
like
you.
fish. You don't even like fish.
I was trying to find a fish for you. All kinds of fun here in the control room. But in all honesty, Jimbo is managing our live feed. He is managing our data collection. He's managing our imagery and communications with the shore team.
Some days makes tea.
Does many things, and we are very, very grateful. He's the lead technician here aboard Falkor, and I find every day I find a new reason to appreciate all the amazing things that happen as we spend 34 days on board, and I get to, every day it's like another layer unpacked. You learn a little bit more. Did he just run into that crinoid and swim away? Did that just happen? That happened. And so very, very grateful for all of the support we have here. Jimbo, you are the best. We love you. And look at the big fish.
Yeah. So you can see when it gulps like that, it has these gill plates open up, and it looks like its throat's been cut. It's a cutthroat eel.
But the body looks really...
Something like
Yeah, okay.
Hagfish usually are not swimming either. They're kind of sedentary.
I wonder when we're going to see one. But there's none in this area?
I've never seen a hagfish.
Well,
out here.
then, we're not going to see one.
Yeah.
Okay, but let's see what else we find. You never know.
Hagfish are really deep though, aren't they?
They don't have to be. I mean, you can see them at a few hundred meters off the Pacific Northwest.
They're the ones you see in carcasses and stuff.
So let's
Yeah.
make an observation that cutthroat eel did not eat the crinoid.
I
Even
guess
though it-
we saw one eat something earlier in the cruise, but I don't recall what it was.
Which one, is that?
It's another. Oh, yep. We're gonna keep going though. I'm curious to see what's over this edge.
Interesting. The dive track length is about similar to yesterday, maybe a couple hundred meters longer. But since we finished the last dive track in about 9 hours, I was surprised that this one's taking us that long. Are the currents really different from yesterday on the surface?
No, we just keep stopping.
No,
Oh.
it's just there's so much to see.
Okay.
I think he's being sarcastic.
I was being
Yes,
sarcastic.
there is a
I
lot
have been
to
looking
see.
at things. It's like it's been taken out.
We're just looking at really different things, and
Exactly,
they're
it's
little.
diverse.
So it takes a while to, every time, it looks like there's nothing here, but watch. I'll zoom in and we're going to find 5 things to look at. In 5, 4, 3. Hey, look at that. What's this? It's a stalk. All right, now I'm just making a point. Come on, let's go. We're also joined in the room by Constance Sartor, who is a graduate student at the University of Guam, working on shallow water coral reefs, but she is also an excellent artist and is the artist on ship, artist in residence, and she's doing some incredible artwork on the things that we see in the deep sea. You want to say hi, Constance?
Hello.
She says hello. But she has a lot more to say too, so if you haven't seen Constance's artwork or read her blog, check it out on our Schmidt Ocean Institute cruise page. You can see a little bit about the work that she does, as well as you can read Steve's blog, which is up too, about our findings from the 2017 Phoenix Islands Archipelago expedition working in Kiribati waters. Still looking.
Can I zoom out a bit more, or do you want to be closer?
I wanted to be closer just to see stuff, but when you're zoomed out like this, whenever we zoom in, I really have been finding little things. But,
Yeah.
But I'm also trying to make tracks. It's always that balance.
Yeah. I was just thinking for post analysis, it helps to be full wide.
Mm-hmm. It's true. Sorry about that.
A little something there. Bamboo coral also.
Yeah.
Same kind of.
Do you want me to go back to it? Okay.
Sticky bamboo coral.
Sticky bamboo
Terrain is
unbranched
kind of
Isididae.
have a little bit of a step change in terrain here. It's picking up quite a bit.
Sure is.
This is talus.
Yep.
Collection of.
Got it. I like it. And Anna's just sending me a note.
Now this like a Bathycrinus,
Oh, yeah.
Crinoid. That's what I was thinking
Yeah,
that was.
I
But
think you might be right.
it's so tough to tell
When they're little.
so small. Yeah.
Yeah. We can keep going.
There are stocked crinoids that are very, very lightly pigmented, like that one was, but most of the ones down here are brightly colored.
You can, but the problem is I can just switch user when you go through. I know this is YouTube anyway.
You know, brightly colored. Is that what you just said?
Brightly colored, yeah, with like red, yellow.
So one of my postdocs, Stephanie Kennedy, who I think you've met her on Zoom, right?
Yep.
Yeah, she's awesome. She's really interested in, she's co-mentored with the Kagan Lab at Harvard University, and she's really interested in thinking about color, which is a question we get asked all the time, but she's a totally new hypothesis and a totally new way of thinking about it. So we'll see. But what I love about Stephanie is she's going for a really big question in a totally new way. And sometimes, even if you don't end up answering your original question, you end up just hitting on something totally new. I love that kind of blue sky science. I'm a big fan of it.
Yeah.
I always have, the safe science, and then always something really, really risky in the works. Ooh, there we have. Can we take a look at this crinoid just because they're beautiful? Yeah, we love watching
Just send
them.
up your camera and I'll swim in.
Yeah, sounds great.
Or you can,
No.
you can send it for me. 'Cause
Okay.
There's some directionality to him, him or her or they, swimming on the bottom.
I
Usually they
love
just kind of
watching them.
indiscriminately swim up and.
cool.
The shrimp is almost like, ooh, structure. Like he's parachuting down. Right on a zillion of those tunicates again. That's what's on those aren't sponges.
So
a lot of those are tunicates.
Some days I have talked to some crinoid experts, and the ones that have the very fine arm tips
Yeah.
might actually be a new or a separate family, maybe from the Camachalids. So I'm wondering if Yeah, this might be good to get some imagery of.
You know,
it's the fine hairs on the edge of
I
the
was
armpits.
noticing that, and I was wondering if like they had been like crunched or eaten, and or if it was just growing.
No,
I was
that's
sort
how
of
they
thinking
are.
it through.
Yeah. I
that's
want to say it's like Telocrinus or something like that. Sounds familiar, but we can send a photo to one of our colleagues, Chuck Messing, who's a world's expert in crinoids, and he will get back to us.
That's great.
We saw someone like this, Telocrinus or Telocrinidae or something like that from the Caribbean, which he was very excited about. So this is neat observation. Yeah, that's not grazing. That's actually how the arm tips are.
I really thought, that it was some kind of growth trajectory, and then it would eventually fill out. We were just seeing it at some stage of its development, but that's really interesting.
Yeah.
I bet what it helps. This is a crazy idea, but I bet it actually helps it to kind of, in some ways, it can poke itself down and move on sand better potentially, or
Yep.
it might actually give it some current sensing,
That's a good point.
and actually give it the
And,
opportunity
like,
to position itself better.
Self-attached weather veins or something.
Exactly. So I just like thinking about the adaptive potential of these kinds of morphologies. I think it's really cool. All right. I think we are ready to move on. Thank you. Flying up slope here. We saw a couple of nice scale worms, some big ones. There's been some neat stuff in here.
Yep.
All right, so my internet search has failed me. I do not know what the little squiggly parts are coming off of a Walteria sponge.
I don't really know either. I mean, as far as I know, they're spicules, but I don't know, what you would call them.
But spicules are really embedded within tissue.
Yeah, I mean, we have used the term interchangeably on a lot of, when we're doing these live narrations. I don't think that's true, but it sounds good enough for the untrained among us.
I know. I just feel the need to invent a new word for it now that I'm thinking about them, because our experiments went really well last night. Yeah.
Silicious projections.
Ooh, silicious projections. I like it. Well done, Steve. Well done.
As opposed to salacious projections, which we don't want to talk about.
No, we certainly don't. Nice. Yep.
Yeah, silicious meaning made out of silica. A lot of these glass sponges are silica, composed of silica, which is essentially glass. Some other minerals thrown in there, but essentially it's glass, which is one of the most abundant resources that some animals can build their bodies out of in the deep sea, since it has a very, very high what's that? Very low dissolution rate.
Oh, yeah.
I forgot what the other.
Oh, let's
opposite
take
that
a look
was.
at this right over here. Sand is essentially silica. It breaks down, and so people always talk about glass versus plastic, and I always think to myself, well, it's not like glass is going to break down at all, but if it did, it just breaks down to silica. And essentially it's just fused sand. Just fused silica turns into glass, and so we call them glass sponges, and that's not just a silly little descriptor. They really are glass. They're full of very sharp, spiny spicules that are amazing. So here we have our first Iridogorgia, I believe, of the dive. And while we've looked at a gazillion of these, I'm going to give a quick scan for a jelly, but I just want to note this because it might indicate a change in habitat.
Mm-hmm. Yep. Now we're starting to get up in the upper
2 meters.
ridge here.
So I am looking for a small corallivorous jelly, as yet undescribed, that likes to hang out on chrysogorgids, and we saw a bunch of them in 2017, and I have not seen a single one this trip. Which makes me wonder about the ephemerality of them, and maybe time pulses, and maybe they have some strange cycle that we're unaware of. Yep, I can go. Thank you. I can't help but scanning every single one of these just really quickly. It's this little reddish purplish. Yeah, that's an amphipod
Want me to take a picture of
thing.
your work?
That's another amphipod.
If possible.
Stare into the belly of the beast. Okay.
I'm gonna jump in the sea logger.
Do your thing. Okay, and then we'll take a little look at this bamboo, which I believe is a bamboo, just to make sure that it is. Whoops.
Can you let me camera and show me where you are.
So Wally Fullweiler, who is one of my colleagues at BU in the Earth and Environment Department, is an expert in silica and in silica biogeochemistry. And so if you're interested more in the properties of silica and how marine organisms use it, please check out her work. She's got some really stunning work thinking about thank you, that's great, about terrestrial silica and how it enters the oceans and how marine organisms use it and how it gets recycled and repurposed and fits into the whole biogeochemical cycles of things. So I really recommend her work. That's Wally Fullweiler. Okay, let's move on. I'm just going to quickly check Slack and make sure there's nothing from our rest of the ship. One of the great things about the ship is that you can watch the live feed from anywhere, from the mess, from your room, from the library, from the lab. And so we have a Slack channel so that wherever people are on the ship, if they say, hey, what's that? Or I know what that is, or collect that, or take a closer look at that, we can respond in real time. So keep our Slack channel open. I'm going to take a look at this. So I think we are seeing a bit of a habitat change. Suddenly we've got a little bit more action here.
Yep, bamboo corals. That stalk behind the Iridogorgia was actually a plexaurid.
Was it a plexaurid?
Yeah, and I was surprised that it had no branches on it. But it looks similar to the species that we've been seeing at this depth. Plexaurid species 2, I've been calling it.
Do you want me to go back?
No.
Okay.
No.
So I was wrong. This is a bamboo, but that other one wasn't. I just assumed that it was. It looked a little strange to me, but I just assumed it was because it was small. But Steve, ever the expert, which I'm so grateful for, has pointed out that it's actually a single-stalked plexaurid, an unbranched plexaurid. Here, I just want to get a close look at the associate for Tim. I'm going to try to zoom in on its head so we can see its rostrum with some focus. So I don't think I'm going to be able to get much better focus here, but maybe just a little bit, and I think we can work with that. There's 2 of them. Is that what you just said, Steve?
Yeah.
Right, and one right next to the other. They look a little different from each other, but maybe one is... I don't know, is there sexual dimorphism in the deep sea?
Sometimes, but... Yeah, sometimes. But I don't know about crabs.
I don't know about crabs either. Alright, thank you. It's great.
I mean, there's sometimes, for example, on one of the collections we had there today, it looked like there were possibly more than one generation in a colony, and that makes sense, from a habitat and evolutionary perspective. You have access to potential future mates.
Yeah, makes sense. I think this is another little Chrysogorgid. See it to the right? I'm just gonna snap it quickly just so we can identify that that's what it is. We don't need to take a careful look at it, but yeah. So they're starting to show up in the habitat more and more. Just want to catch up on some notes. Okay.
I was just looking at the rocks so far. I think
We're done.
4 rocks is plenty.
We
Yeah.
can't take any more anyway.
They're
4
that.
per dive.
Oh, 4 per dive. Yeah.
In the monument.
Yeah. No, that's good. Yeah, they get confusing after 4 rocks. I have to go back.
We tried to take some notes on what they were.
No, plenty. Yeah. I don't think we're not covering enough depth range to really need
Is
to.
this another cutthroat eel?
This is
No.
a helisar.
It does look like a. Yeah. Okay.
It's the
That's
flat
better.
face.
Thank you.
And if you are able to zoom in on the head, whether it's scaly or not, suggests one genus or another.
Oh yeah, working on it.
If you could see the brain, that would be Aldrobandia, and if you can't see it because it's scaly, it's probably Halosaurus.
Well, it's just not showing us its brain.
No, this is like 90% of the pictures in the deep sea about fish, like fish butt.
Yeah.
They don't like the imaged well.
Well, let's keep going then. See an urchin, some sponges. I'll zoom out a little. What is this? I'm gonna zoom in on this very quickly. It looks like a dead sponge. Yes, dead sponge. Anemone. Oh, thanks, Adam. Oh, yes, whoever did that. That was great. Look, we get this transition from this talus to some more consolidated pavement. Is that correct? All right. Look at that. Speaking like a geologist now. Thanks to Steve. And I'm gonna guess that we'll start seeing some different things, but you really never know until you get there. But even if we don't, again, these observations are so valuable for us because we don't know what to expect in different parts of the mountain. We know how to put ourselves on steep, slopey, hard ground, but then the biology, what we're going to see at each of these spots, so it's always a little bit of a mystery in terms of how much we get.
So for the dive trek, we don't need to hit the waypoints, right? We can just kind of curve around. Do you have a preference?
Exactly.
Okay.
But it looks like we're starting to climb here. I see another stalked crinoid coming up. Yeah, we can just curve around. It doesn't matter. We're just trying to sort of follow the basic curvature of the line, but we don't have to be on it. There's in fact something on the top here. It'd be good just to confirm what it is. It might be an Iridogorgia, but it might be a stalked crinoid, or it might be something else. I can't tell yet. It's an Iridogorgia. No, it's yes. It's a yes. And no jellies. Okay. Moving on. Yeah. So this terrain is really weird. I thought we were getting into more consolidated pavement, but we really didn't. Suddenly we're back into, I mean, kind of large rubbly rocks.
Broken pavement, I think.
Yeah, broken pavement. So there's a question in the chat about hermaphroditism in the deep sea, and I think that there's certainly a lot of it because it does make sense, but there's also quite a lot of gynocorism where you have true males and females. So it's a big mix of strategies and reproductive timing and reproductive behavior and reproductive. I'm going to take a look at whatever. I think this is a little. That's perfect. That's perfect. There's a couple things to look at right here, so we're good. So it gives us a chance to. So I'm going to check this out first. It's really hard to study reproduction in deep sea organisms because we get a quick, fleeting glimpse, right? We can't kind of look at them for long periods of time, and we can't get out, at least here, very, very often. I think more coastal areas we'd have more luck, but it took us 3 or 4 years to plan this expedition. We're literally out in the middle of the Central Pacific. It's a 7-day transit from anywhere. Well, that's not true. A 7-day transit from Hawaii.
Some regrowth on that stock.
exactly what is going on here, and it's just really so we can't kind of be here at all different seasons and all different times. This is crazy. I'm at max zoom. So, you know, whenever we clip things, we assume that they regrow, but we're not here all the time to know. And this expedition, we've seen a lot of evidence of regrowth. I've seen some on Analopsamia, I've seen some on other corals, and now we're seeing a crinoid that must have gotten clipped somehow and is regrowing, which is really cool footage to see this stalked base have enough animal left to be able to regrow ahead. That's very cool. You know, I always think about energetic resources and how much energy has to be left in order to be able to reform a major part of its body. And there was obviously enough left here, and the minute these features, right, were formed again, it can start catching food, which can replenish the energy, which can enable it to grow larger again. So that's a really cool observation. Thank you so much. 21, 35, 14, 33 meters. Regrowing top Alrighty, parades. So we've got some sponges here. Let's take a quick look at them. We've been noticing them a lot, but we haven't had a chance to snap zoom on them. So here's a chance to take a really good zoom. Let me change the iris. It's great. Let me see if I can find one a little closer to home so we can get a better look. Oh yeah, there's lots of them. Let's take a look at this right here, which I believe is another one of these crazy tunicates. It is. So Steve, you can see why we were confused in the beginning. This is a tunicate, but there are squillions of them. Oh, I'm going the wrong way for focus. I'm really excited to see so much evidence of regrowth. It's really amazing. It suggests that A, there's enough food available to do that, and that B, these organisms really can recover in a way that I wasn't. So this is a tunicate, and then what do we have here? This stringy. Oh, that's not an urchin. That is a benthic siphonophore, right? Dandelion siphonophore. Is that what this is?
That's right.
All right. I love these. They're so wild. You can see it spreads out its tentacles. It's trying to sort of capture particles, and you can see how successful that might be. It's a big net, and it also benthically attaches itself. It's almost like makes a sort of a spider web structure.
Is it not stinging?
Oh yeah.
Yeah.
Ouch.
Okay.
You want?
What?
No.
You just think maybe it's devil? Try it.
I'm like, I don't even know what to do with that, Russ. I'm just like, look, like, I'm too tired to. Yeah. That's cool. We have a crinoid over here, which also looks like there's some evidence of regrowth. Take a look at this. It looks like there's.
See you saying that.
Up.
Sounds great.
Here we go. There's an anemone in the back. See, you zoom in and there's like all this stuff to see. Can you help me get focused for a minute?
Yeah.
Are we flying again?
I got it.
Sorry, I thought we were sat down.
If you zoom out real quick.
Yeah. That's all right. No, all good. I saw what I needed to see. There's
Alright, we'll start making like
Yeah, let's go.
position.
Let's go. Let's make some tracks. That was a crinoid with a regrowing arm. And there's our little regrowing stalk again. It's so cute.
Good morning, Mr. J-Rock.
J-Rod, hello. We have amazing ROV pilots on the ship. We've got Russ and Adam flying right now. J-Rod walking into the room. If you haven't seen his very cute Father's Day video, you should check it out. It's on the Schmidt YouTube channel. Oh wait, that was one of those big shrimps. We just passed it. Pneumatic Ursinus shrimps, exactly. That's like the same kind that ate the fish, right? Yep. giant predators. But we'll keep flying. Another Iridogorgia, just to note.
It's like yesterday when I was flying, it just flipping in and out, and then come on back. Since the one back there, just keep so you notice that.
Steve, what makes these mucus nets, these 3-point like connection triangular mucus nets?
No idea. I've never seen any evidence of.
You've never seen what?
I've never seen any evidence of what makes them.
But they're all around.
Yeah.
Here's another little Chrysogorgid to the left. See it?
Yep, I got it.
And there's cucumbers
They
everywhere.
are all around, but yeah, it's.
If this is a sparse dive, maybe we should collect one and see if we can figure it out. Slurp it up. I've always wondered. There's a lot of tunicates on this dive. This is clearly their habitat, you know, which again speaks to the fact that while this may not be corals and sponges, there's a lot of other organisms taking advantage of this landscape and using this using this terrain. And it's clearly, excuse me, got enough falling marine snow to support all of these other animals, and they're pretty happy here. This is their spot. What is this? That's another Crinoid. Okay. Can we take a look at this right here? I just want to see what kind of sponge that is. Thank you. We've got some kind of hexactinellid glass sponge, again, highly silicaceous. Or silicious, but not siliceous. Just borrow the Steve's joke. Okay, and then I'll just say silicaceous. Great. I'm gonna see if there's anything on it. Really interested in, we collected part of one of these yesterday, and it was bright white just like this one, which indicates that it's very alive and happy, and by the time we got part of it to the surface, some of it had browned, and I don't really know what that means.
Yeah, I don't know.
Do you?
I've never seen it. You want to do that so quickly?
It was still alive because we used it in experiments. We were able to actually, we're measuring respirometry rates, we could actually see it breathe. So sure, it was still alive, but it was very interesting to see the color change. Okay, that's great. Thank you.
It's a euplectellid for sure. I
A euplectellid?
couldn't get further than that. Yeah.
Okay, I'm going to take a quick look at whatever this is up here. I think it's just a dead sponge. Yep, it is. Okay, thank you.
So looking at some sediment samples from Howland, now that you see the sediment patch, we did observe that it's mostly foraminiferous tests.
Oh, it is?
Yeah.
Okay,
Almost,
definitely.
Almost exclusively, you know, and it's really fine and powdery. It's
Yep.
yeah, it's really neat. I saved some from the bottom of a bio box.
Excellent plan.
If you want to take a look at it under the scope. There is some fine silty clay stuff too, but it's usually associated with their, you know, these collections where we have to dig like Seamount or stuff like that.
You know, whenever we look at the sand and sediment close up, you see sometimes the structure and it looks really round. And I was thinking, is it oolitic? Is it foraminiferous tests? And it's really great to have the confirmation that it's foram tests, so, which tells us a lot about what's falling and what was on the surface and is falling down. We are joined by Dr. Tim Shank. How you doing, Tim?
Good morning, everyone. Great, doing great.
You can.
Except we're not seeing any corals.
We're not.
Well, I mean, we are.
We're seeing lots of interesting things.
It's not coralorama, like, but
Like
it's
yesterday
a
was
noteworthy
fields.
comparison. It's a noteworthy comparison,
Yeah,
right?
right.
This is
That
the
was-
second dive we've been on where the north faces of the north ridges.
What was the other one?
Not,
Well, this was our goal.
I can't remember.
To be at the same,
it was recent. It had
Yeah,
to
I
be
know.
Hollander Baker.
Exactly. I just can't keep track
to
of
be on the
what-
other side of the same depth, and comparative depth, and then we see
It was the one that
so
I
much
picked
more on the
where I
other
was like, oh, I
side.
did a terrible job, and then you picked the next one, and it was great.
I think it's
We just
there's
reversed.
some really complex habitat suitability effects around the seamount. Probably
take a quick
currently,
look at
and that's
this,
what I'm thinking about.
stocked
How
crinoid?
these days,
Yeah.
current.
What with this?
Oh, you're so
How
good.
does current move across the seamount?
What was the current like going down this morning?
Okay, I love how you can anticipate
Maybe
my
that's
every need, and then I
typically
want us to check out
strong
whatever that is
equatorial
behind
undercurrent,
us.
but at the
Which
bottom,
looks like it makes space
I wasn't
bend, but-
awake when we got to the bottom.
Yeah, right.
So,
So, yeah, we can look back at it, but yesterday was almost nothing. But yeah, we
Tim,
had
is
tons
this a
of
little
goals.
shrimp
yeah, when you
inside?
get over that ridge, it speeds up.
Yeah.
You got a
right.
shrimp associate?
I
Pretty
don't.
ragged, huh?
Yes.
It's pretty ragged. Helps.
Right there? In the middle? No?
Randy, you have a really good eye, but I just don't. Oh yes, my God, yes, yes. Well, my goodness, it's actually a Caribbean shrimp.
Is that
You
normal?
don't. No,
Oh.
it's not a common thing that I've seen. Or
It's
even.
really like going at the center of this crinoid.
Don't know what is it? All your is that all your zoom?
Yeah,
no.
it's all
No.
retracted. I wonder if that's
Exactly. This whole crinoid looks very odd.
Can you focus on that?
I'm working on it. I'm working on it. I'm just slow. Bear with
There you
me.
go,
I'm just
there you
like
go.
the deep sea.
That's
I'm
nice.
slow today.
the current is picking up though. You notice it.
Uh-huh.
Really
the
long.
Camera and tilt is starting to vibrate a little bit.
Really long rostrum, and it's Caribbean. It's I mean, the legs are moving around this the
I mean, this thing is clearly.
It's not trapped or anything. No, it's
No,
just
no,
it's
it's
just
where it
hanging out
wants to
right there.
be.
Yeah,
Yeah.
it's just hanging out there.
All right.
Yeah, that's a new observation.
That is a new observation. So let me write
Good
that
eye,
down.
Randy. Really good eye.
Well, you can thank Russ who predicted that we'd want to go look at this thing and got us right in position.
Well.
Russ now. Russ, yeah. What is this? This is a crinoid with a Caribbean shrimp.
Yeah.
It's a Phrynochrinus or Phrynocrinidae family.
It's definitely hanging on there, that's for sure.
For sure. Nobody's gonna be able to read my handwriting later. I'll try. Okay, let's take a look at the blob behind it and see, make sure we know what that is.
Let's
This
look
has
at
been a
another-
very blobby dive.
Let's see another hymenaster.
Think?
Yeah. I do.
I think you're probably right.
I don't know. They're
Uh,
slam star. Yeah.
You are
I
correct,
don't know.
sir. Ah, I can't quite get focused, but there we go.
It's close.
Other way. Ah, there you go. Slam star?
Yeah. We found one of these at 8,000 meters, and it was just respiring like crazy. The center of it opens up,
Wow.
and water flows water through.
Yes.
And I haven't seen them during this dive sequence, but
There they
I
are.
keep looking for that thing to respire.
I was just sort of taking
up
some
against-
lasers on them just to get a sense of scale.
Yeah. Yeah.
All right.
Wow.
Huge.
Is this that iceberg?
A
All right.
blobby dive.
A blobby dive.
Seen
We
lots of
had
blobs.
well now we know they're tunicates, but it took a while. There were these, what were we calling them? Unidentified gelatinous blobs for a while, but they're very
Yeah.
odd tunicates.
Yeah.
They're not like the predatory tunicates we normally see.
Right.
And there are squillions of them. We collected
Yeah.
two.
Okay.
They're very cool. We saw a pygnogonid right next to one, and it looked completely
Was it-
shriveled. So it looks like a pygnogonid
Was
was
it-
eating that
oh,
too.
really? Right.
Pygnogonid's have just been really
Was the
interesting.
proboscis out?
No, I
See
think
it?
it had just finished feeding. It's an assumption. It really looked
Yeah,
wrecked,
yeah,
and the
right.
and it was just moving on.
Wow.
So now we're getting like what I thought would be the most interesting topography of the dive.
Mm.
We're kind of on the peak of a little mount, and then we're going up another ridge to another little local topographic high, and then we'll go up another steeper slope to the rim of the seamount. But we've only got maybe 200 more meters of vertical to go, and maybe 500-600 meters
Let's check it out.
in the horizontal. So it'll be interesting to see what the effects of these local highs are.
we are traversing between waypoint 1 and waypoint 2.
You didn't see any hard corals this morning. Did you see anything?
So
Isn't
the
that interesting?
pilots are noting a current change, a change in the current flow, which is always potentially very interesting for what we might
This is
see
super
biologically.
This is super interesting because of yesterday, right? I thought we had patches of it in Alexandria yesterday.
I can't even remember what we saw yesterday. And here we go, something different. Look at that.
It's like a
Crinoid, yeah, right.
The two lasers are 10 centimeters apart, for those of you who are wondering. And am I okay to zoom?
Just let it get settled first to know easier.
Sorry, I'm going to try to let my impatience. Just tell me to wait. It does look like some kind of Crinoid.
Yep.
Okay.
Should get that.
We've got, let's see what we've got. We've got all kinds of things going on right here. We have a Crinoid with an ovaroid on it, but we also have 10, what's this? Come on, be focused.
Is that a little branch at the end there?
What's that?
Just to kind of keep it slow and set.
Some schmutz that's caught.
I was thinking amphipod originally, but that may just be a piece of schmutz. I thought it might be your
I did
your
too, that's
medusae,
why I went right into
right?
it. But look, there's some stripped tissue. Or maybe it's just growing tissue. This looks more like it's growing than stripped, don't you think?
Yeah, that looks normal.
Yep. It just caught me off guard. And this just looks like some gelatinous schmutz. It's gotten stuck on the coral.
I think I'm gonna go with that.
Gelatinous schmutz. I like it.
See how it branches it toward the top?
Yeah.
Every once in a while?
Yeah.
Tim, what's going on with this ovaroid? It's got some bulbous pieces
got-
that I'm not used to seeing.
Is that part of the coral? No.
No, it's part of your ov.
Yeah, are you all the way zoomed?
I'm all the way
Oh,
zoomed.
come on.
But do you see this? These
I
little
do.
pink
I
spots?
can see it.
Don't you
This
have
is
to
just,
make it
I don't
closer?
know.
Do you want me to ask
Well,
them to
let
get
me sh-
closer or collect it or?
We haven't seen enough to collect anything, right?
Well, we can, as long as we can collect the whole thing, we can collect a part.
Yeah, sure, if you want to get it. That may just be part of the central disc and the arm connection,
Ah,
but I don't, but you're right, it's not on every arm though, so I don't know.
no,
can't
that's
get a
very
sense of it.
unusual, that's why
Yeah, good
that's why I
call.
pointed out.
And that's where the arm would swell too if it had a lot of gametes in it. That's where they periodically accumulate their gametes,
Do you
right?
have
So
interest in,
yeah, maybe we do see it. Is it that we're looking onto the side of it? And that
Yeah.
is that?
yep, you're right,
Is that just-
okay.
I don't know, because I see it's also in here. See that? So maybe it is on each arm.
Okay, well then maybe we
You
can
know,
just-
but if anyone has, would you like to collect this?
Yeah, I'm fine with that. It's Norella. I can't put it a species yet, but it's Norella
Okay.
for sure. I'm fine.
I
You're good. If you want to collect it out.
Just turn around and look at
No, I mean I have no preference. Like, whatever.
All right. Yeah. Can we snip it just below the?
Yeah, I think we're gonna collect. I think we're gonna try to collect the
What's the
ovaroid
next
and a
number?
little bit of coral, maybe a branch point.
One. No, sorry, two. No,
Thank you very much.
I can't.
This is gonna be sample FK400.
I think
Nope,
370.
402.
It says 378
No,
there.
400, 401.
You're right.
Those are
Okay.
reps.
Okay. All right. So it'll be 402.
Mm-hmm.
Yeah. So no Victor Gorgia today.
CNA, and
Right.
I haven't watched very much. Just the past
Yeah,
hour.
I saw one crowd.
Yeah. Really? Okay.
Well, yeah. Yeah. So there were a bunch of whips, bamboo whips. So no black corals.
I didn't see one, but there was some like questionable coral crinoid dry zone we saw. Couldn't fit it to anything.
Yeah.
Could have been a black coral, but
And you could get one different.
yeah,
That's the point. It's really amazing.
yeah. I do wonder if maybe this area, since it's
far up current, like the currents that goes to the north and east, maybe it just doesn't get the supply.
I mean, the water column is so much clearer today.
Yeah.
We're not seeing all this particular come through.
Well, also,
Like we did. No.
but the current here is ideal. I mean, this is exactly
think
what we
you would've
saw
been-
at the
yeah.
ridge, top of the ridge yesterday.
Yeah, for
Yeah.
sure.
Right. Thank you, Don.
That'll probably do it. So I think that would be where I'd go.
Yeah.
So it'll give us 1, 2, 3, 4, 5 branches. Yeah.
Lots of rock.
Yeah. That's good. I mean, we didn't get any rocks at 3 dives on the carbonate platform, so
Yeah, that's true.
that's fine.
That's good.
There should be predation. That brought it in.
Simple 402, right?
Yes.
Yeah, exactly. If you So we're just discussing collection strategy, and we have a big experiment going on with primnoids, and so we're going to take a little more of this colony, probably close to about 1/3 of it, so that we can get this strange ophoroid, and also so that we can run some really cool respirometry experiments, and also so that we can identify it taxonomically, because when we know that it's a Narella, we don't know what species. And so there's a whole lot of reasons, there'll be 3 of us using the same piece of coral tissue
Mm-hmm.
and preserving it in different ways, doing live experiments with it, and keying it out morphologically. So while we always try to take as little as we can, in this case we're going to use it probably close to 15 different ways.
Mm.
So we're going to take a larger chunk.
So that rock is
That rock is
not stable.
correct.
No.
That's neat. Good to know.
Oh, great collection.
And Bob also says again,
Yes.
Yeah, firebox is great. Just let us know which one.
we'll do biobox in.
Thank you. Firebox A. So Sebastian is about the size of a car. You can take a look at what it looks like on the Schmidt Ocean Institute's webpage. What is it? SOI dot schmidtocean.org. And you can take a look at Sebastian. I think we're actually going to put Deb's is amazing. So quick. Right there, you got a link.
Quick fingers.
Quick fingers. I used to keep wondering why it's not like a thing for everyone to have like sports team T-shirts with like nicknames on the back. Quick fingers, Deb. I don't know. It feels like a thing. Everyone should have the Schmidt Ocean Institute T-shirts with our ship names on the back. Science Steve. Andy.
Andy. That's right.
I'm in. Im.
I don't know.
I don't know. That was a great collection. Thank you very much, guys. That was so quick. So we have Adam and Ben flying, our fabulous pilots, who just made a very, very efficient collection, which is going to be really helpful for us. So thank you.
I mean, we sort of take it for granted now, but that collection was made easy because of the modification that they did of the manipulator arm, of the claw there. They have a cutter that's on either side of the fingers, like a blade that comes down together and cuts the coral axis. But it also has these, I don't know if it's rubber or not anymore. It's a type of plastic rubber that I think they 3D printed it, and so it holds the coral while the blade cuts it. And something that we started back in 20 years ago.
Yeah.
Yikes.
It's amazing.
With Hercules. Yeah.
Our last cruise, we were out here with Daniel Vogt from the Wyss Institute at Harvard, and he was developing a new kind of manipulator arm called squishy fingers. It was like a grabber that could actually, it was a soft-bodied grabber to grab soft-bodied animals.
Right.
It was these robotic arms. And he was, remember, 3D printing different iterations every single day.
He was modifying the thing constantly to pick up squishy
amazing.
Holothurians and gelatinous things.
Yeah, so he would like grab something, we'd watch it fail, figure out why it failed, then he'd talk with the biologist, we'd say, oh, well, this organism needs this, and he developed like fingernails and all kinds of other modifications. It was really awesome. I miss Daniel.
Yeah, I do too.
He's a great guy. Squishy fingers. Okay, and now the landscape has totally changed. Cue Pavement. Let's see what we got. Come fly with me.
So we're still heading for 1150 meters, something like that?
Yeah.
And we're currently at 1436.
Yeah.
Yeah.
Come fly away. Right? Sorry. I just feel like, we need a soundtrack to the deep sea. What?
You cut an album? You could cut an album?
I've written like a bunch of songs about the deep sea already.
Really?
Why not?
Oh,
Well, I wrote one, the Oreo song from last time,
Yep.
which I still have the lyrics to. It's in my lab notebook.
Yeah.
So in the back.
I'd forgotten that. That's,
I started writing one last time too. I haven't written anything this time. The most recent thing I've written musically is a pandemic song about.
you wrote a song about the pandemic?
I did. We got chickens, as you know.
Yeah.
I can't believe I'm about to tell this story live, but why not? We're waiting for things. And my kids were like kind of sad that their pets don't have any special features, right? They're just chickens.
Right.
And my husband, ever the dad joke kind of guy, said, chickens have a really special talent. If you put them in a dollar store, they can tell you the price of everything. Buck.
Oh my gosh.
Buck, right?
Oh my
so
gosh,
I
yeah.
immediately just grabbed a pen and wrote a song called Dollar Store Chickens. And it's a country song.
It's a-
The whole
it's a
thing just
country
flowed
song.
out. It took about 30
Oh,
seconds.
my gosh.
And my whole family sings it now. It's kind of
It's
good.
been performed for the family and everybody's.
Oh, well, my whole family
Does
knows
it?
all the
Yeah.
lyrics and
Yeah,
sings
yeah.
the whole thing, and it's just
Oh, that's cute.
Dollar Store Chickens. I really think this could make it big. If I ever don't make it as a scientist, I think Dollar Store Chickens might make the big scene.
You gotta send it to Taylor Swift or something like that, right? You gotta send it in.
I should.
Yeah.
It,
Nothing to lose.
I'll sing it for you later. I'm not singing it on air, but it's
look,
good.
I look forward to it.
I'm very proud of my dollar. It was a collaborative effort, right? Had
All right.
Jeff not made the joke, it wouldn't have
We'll
worked.
we'll have a show.
All
Is
these
that a
dead
C-
sponges.
got a C pen there?
Yeah, let's take a look at this pen. Wow.
Oh my goodness.
So this is going to be my path to getting dollar store chickens.
That's it.
Off,
This is
right?
where-
I've
this is
announced
where
it
it starts-
on
this is the
YouTube,
launch
and
of it.
now it's a thing.
That's right.
We're going to take a look at this pen. Okay. And now it's stuck in my head. It's quite the earworm song. What is that?
So we did see this yesterday. What?
Yeah, what, just gonna wait, I'll come back to it. Let's first look at the C pen.
It'd be great for a white balance.
It's like, yeah.
Yeah.
It's like a percentage, Randy.
You think that it's very little?
Yeah. We saw little percentages yesterday.
Little percentage.
Alright.
One that only had 2 arms that were fully, saw that.
Alright. That is beautiful.
good.
I don't see any associates, Tim.
No.
There have
Typically-
been some on some of them,
Yeah.
but I don't see anything. Alright, let's take a look at that star, and then see if the pilot's going to white balance. Is that a Pavement too?
I don't know. I got it as you were zooming in, but
I'm working on it. Yes.
I'm calling it percentage. Yeah.
I'm agreeing.
Yeah.
Right.
Good.
Let me ask. Yeah,
Zoom
sure.
straight in. You got it.
Oh, sorry, I went the wrong way. I'm just trying to make you dizzy. Ah, perfect. Thank you. Yeah.
Okay, there you go.
They don't care about dollars for chickens.
No.
Not even a little bit. Maybe it won't be a hit song.
All right.
All right.
Yes.
You might care about it, or you're just placating.
No.
It's funny.
I look forward to hearing it. Recording it.
Interesting.
I'm gonna record it
I should.
on my
I bet
phone.
you.
I'll have the first recording, and that'll be worth something someday.
Not the first recording.
Oh,
We were at a
right.
friend's house, and when the whole thing
Yeah.
came out, I just grabbed paper and wrote it immediately. All right, I think we're ready to fly whenever you guys are. All right, let's see. Look at the landscape. It's really linear, right? And it's got all these. It's almost like a spur and groove, but it's flat and rocky. I think this is a dead sponge that's kind of just decaying into the sand, turning into sand, actually.
Were you coming over walls and things were you coming over ledges and earlier and whatnot?
Mhm.
Any verticality?
Yeah, lots.
Yeah. Walls like even?
Mountains,
Yeah, was-
Rocky Mountains,
Rocky Mountains. Up and all right.
Rocky Hills, but they were steep hills. Oof. Sparse landscape.
Seeing some particles in the water coming toward us from left to right.
Yeah.
It seems altogether clearer than yesterday.
I mean, we are in oligotrophic waters in general,
Yeah.
except for the fact that these equatorial currents do bring nutrient pulses, right? There's
Yeah.
upwelling that happens, and
This one I'm seeing too is also a function of our lights and where the camera is and all that. So
for sure.
Can you square up the
I was gonna say.
camera?
Sorry.
Is
Yes.
it full wide?
Yep. They just didn't want to look at the arms at the same time. They wanted just a full frame.
Mm. It also helps because we have our heading sensor makes sense when your camera's
Yeah.
parallel with the
That makes much more sense.
axis of the thing. I keep hitting this thing.
It's
over.
okay.
Oh, it's the tabletop.
It's the table. It's not you. It's the table. But they're trying to do like a psychology experiment
It
on
is,
us, right? You know,
I
like.
think I'm a lab rat. There's all this stuff in here. They put all these tablets are everywhere, and I'm like, I never touch them.
I had to use them because in my room the only way to control like watching the dives is on the tablet. And I finally have kind of mastered it, but it's
really
it's
good.
temperamental.
I'm not. I'm the lab rat that doesn't go down that part of the maze. I'm not even playing.
I didn't until I had to.
Yeah, right.
Right.
I've not
It's
been
very
forced to yet.
temperamental.
Yeah.
It doesn't turn on. It doesn't turn off. I have to figure it out. But anyway, I think it's my inadequacies. So here we have another benthic siphonophore. Take a look at that. Oh, sorry, I just zoomed right in. We're gonna look right up there if we can. Okay. Oh. See it?
Just hanging on to something.
It's got these urchins in the background. They're just cool to watch them feeding, but I wonder how he's setting up in this incredible current. It looks like instead of having links on both sides, he's kind of ballooned out, right? He's only got
It's just-
three major strings. This is a pretty small net for a siphonophore.
So this is a rhodalid, you think?
Yeah.
It's a rhodalid siphonophore, like the
Dandelion.
yeah, that was really made famous when the hydrothermal vents were discovered.
Yeah.
Really?
Oh yeah.
That's
And
the
this was-
story?
They were these. That one's called Pericallia taraxica, and it hangs out in the areas that are dying, like mussel beds, and that are where the vent water was waning. They were scavenging, and they sampled it and into a suction sampler and trying to get it up to to a container. And by the time it got up, it had dissociated.
Oh wow.
In these fragments, there was like nothing left in the bucket when they got to the surface. So those are all different. As a siphonophore, those different things waving around have different functions. Like some are for digestion and other things. So
But they can handle
and if you
vent heat.
no, they're not. They're on the outskirts of the vent fields.
Okay,
They're on
got
the
it.
periphery of the fields. Again, scavenging material that's coming out.
That makes more sense. Got it.
So they'll stick like that between rocks and just sort of feed what's the dying material coming out in the water column.
Yeah, it's attachment points. That's cool.
There's some long tendrils there.
Yeah.
Yeah, very cool.
Very cool. All right, let's keep flying.
Right?
Zoom, zoom. I like it. What was that, Chris, out there? Yeah. That's a great story.
Yeah, well, there were all these biologists on board too, and they were really surprised to see there was nothing in the box. So,
Another crinoid.
or looked like nothing in the box, right? Just a, So I think we saw one sea star earlier. Any other sea stars you see?
No.
Nothing. No predation. From
Nope.
sea
A
stars?
couple picnic
Slime star.
Here's another.
Oh.
Can we take a quick look at this? Just a snap, quick snap. I'm pretty sure it's a. Actually, wait, no, it might be more than a quick snap. Because now I don't know what it is. I thought it was a little Crixigorgid, but maybe it's not. Maybe it is.
Let's take a look.
Oh yeah, it's just a little Crixigorgid. Do you want to look at the associated design, or are you good?
I would like to, if it's okay, if you got a minute there.
We do.
Just because
I've been trying
you
to get
saw
zooms
one
on
of these
them.
earlier.
Yeah, please.
I'll try to help.
Thank you. Please
I'll
help.
do this.
Please help. Help me.
Okay, now go in. You got it.
You can see the arm waving right down here.
Come on now.
That's about all you're gonna get, unless you want to poke it.
That's fine. That's fine.
Okay. There's two of them.
You become a
One
master
here,
at looking
one
at the
here.
coloration and what little structure you can see of
Yeah.
that. There's a crab down in the bottom left corner.
Yeah, two of them.
That's a
And
Europe,
couple
it's a
shrimps.
Europtychus crab. There's a couple of mysids flying around, which often do.
Uh-huh.
I think in the lee of these things.
All right.
But anyway, yeah, great. Thank
Great.
you. Mm-hmm. Sometimes it's a shrimp that lives in there. I just want to make sure who's who.
Yeah. So. I think we're ready to fly.
Thank you,
I'm
Randy.
gonna let Tim take over.
Thank you.
There's an outreach event today. I'm sure everyone's on it. So I'll send it to Slack.
Just if you see anything going around, just make sure. Remind them to leave for lunch.
Oh, yeah. I will.
Cause I have to come back on at 12, but I'll still be here.
Sorry, Steve. Okay. Yeah.
Juggle that.
Do you want to see this one, Steve? Or you know this one?
No, that's the only sample.
Yeah, it is.
I will come back in 20 minutes to relieve for lunch. I'm just literally going to take a 15-minute out. I'll be back. I'll set my alarm.
A little bit of a fish here. A little bit. So we're just on our way to waypoint 2.
Yep.
Yeah.
I'm kind of doing a lazy arc.
Yeah, I see the track.
I think once we get waypoint 2, maybe we could pretty much just that it books it straight up
Just
the wall.
drive. Yeah.
There's these are small divots actually. They're supposed to be in a divot and now climbing up again.
Yeah.
Maybe very small, like
They
10,
are.
20 meters. And, yeah.
No.
I was
No, we're good to go. No.
hoping there would be something remarkable on these
Keep
bumps,
going.
but there's nothing really
Yeah.
to speak of. Maybe it's why we're seeing these Narilla, but I can't imagine anything else.
They're
Get
so
this terrain favorably, swimming here.
yeah. It's just really sparse. We saw that Praxigorgia before. Seeing, Narella every one colony every so often. Thought I saw that encrusting sponge on some of the sides of the rocks. I don't know if you've been seeing that too. A few dead sponges we're seeing now. Saw 3 in the last minute. Just a little sea pen here. The one we just imaged.
Everything's the same.
It's the same, yeah.
Same definition.
There's a big rock here.
It is a big rock.
It is a big one. Look at that.
Where? There's a little white branchy thing.
You want to see it? Yeah.
Just quick.
Take a zoom on that, Ben. Thank you.
That's a big block.
I think the rocks yesterday hosted a lot of diversity. Like
Yeah.
this rock over there might have a lot more on it.
Current's kind of stagnated a little bit again.
Yeah.
I don't know what that's all about.
There you see those dead sponges, and you see the live encrusting sponge on that big rock. There's a few shrimp here. We're gonna creep up to this thing. You ready? I'm going right here.
There's your Naloxone.
Yeah.
First one.
Got the lasers on it. It's got it.
There you go.
Now you can see that sponge close up too, but That's not the one I want. Yeah, it's just a new bud.
Yeah, I did see another one, slightly bigger on top of the rock, but it's good to know that they're here.
Well, can we image that from here or not?
No. I don't think we need to stay and look at it. It's
Right.
looks the same, just maybe a couple centimeters larger. Didn't see any associates.
Nope.
Ready to go.
Yep. Thank you, man. Nicely done. Over the rock. There it is. Yeah.
So that one little coral comes to well, they're confused.
Yeah, I think that rock, that boulder was just a shadow. The currents picked up again.
Oh, we were just sitting in the leaf for a second. I mean, yeah.
Exactly.
Yeah. Working on it. Have you back out current data from ROV? Before, like I've done it with AUVs where we've gone and
Yeah.
determined the current.
It's been somewhere around 600 meters right now. It's a rock pad.
Yeah.
I've collected one of those.
So Adam, what's the favorite thing you've seen so far?
Not wireless.
That was early on. How about you, Ben?
Favorite thing I've seen
What do you think you've seen so far?
was this
Well, on this cruise, yeah.
was the whale shark.
We saw a whale shark at 471. And, yeah, 671 meters and 400 meters. We saw a whale shark diving down, and that was extraordinary. It was on our way of. We were descending with the ROV. Caught somebody on the camera. It's a first for me too. Another. Hand up a little bit here. There's a few corals up here. Is there Gorgia?
Yeah. We saw a couple earlier. And
All right, you did.
These are much bigger though. Everything now is much bigger.
Can we just take a look at this? There's something red on the other. Is that a crinoid on the left? Let's take a look at this for just a second. Just want to see if there's any big. There's a rat tail fish. There's a large anemone, and then we got another coral on the right. Just looking for some associates in here. If there's anybody home. Didn't see anybody in the early Iridogorgia on this. Nice job, man.
Coral fish shop.
Yeah. Often there's a couple of Europtychus crabs or Bathypalmonellid shrimp. I see nothing here today. To also look for those little medusae that may live here that have been thought to be predating on corals like this. I don't see anything there either. Alright, I think we're okay. Just coral on the right over here. Right into the plane of it. Sorry, I'll let you drive it. I'm sort of pushing it. Take a look at that one, Ben. See what's. Okay. All right, we're good. There's a homalid crab underneath that rock too, over there next to it, with an anemone on the back. And we have an ophiuroid on a coral. Ophiophenthaka. And this is Nalsamia. I'll go into some of the, look at the polyps here. Yeah. So Ophiacanthid ophiuroids on a Nalsamia. Great. Nice, Ben. We can keep going. It's our biggest colony of the day, right?
That's the one.
That's the one. Oh, wait a minute, Ben. We can holly crab. Just a quickie. Yeah. All right. He's well under there, but that's another
I'm
anemone.
curious to see the homalid relationship differences between coral-dominated habitat and spare rock-dominated habitat.
Yeah,
They use
right.
anemones more.
Yeah. Great, Ben. Thank you. I think I'm all. See if I'm lined up with the basket or not. I'm just going to point down for just a second.
I'll get to your
I got to go left. There's another C pin. Oh, I see. Thank you. Yep, I got it. There we go. Yeah, I agree with you. You know, when I see them in coral-dominated habitats, they've got even different kinds of corals on there.
Yep.
You know.
More to choose from.
Yeah.
But yeah, I noticed that that grenadier was around and all the homalids were under the rock here.
Yeah.
I wonder if that's something tells you something about food webs.
And anemone is not enough to protect them. They've got to go under the rock.
Maybe.
All right.
I don't know if a grenadier would go after a crab.
I don't know either.
Usually macroinvertebrates.
There's a long whip coming up here. Maybe it.
Oh, yeah, there you go.
Another Nalsamia, right? Want to take a look at this? It's a bamboo whip.
Yeah.
Let's take a look at this. I see no branches. Yeah, I'll let you. Yeah.
Yeah, I think you know
There's a couple
we have
of
a
them.
we have a pretty good handle on what these are. But if you ever see one with an associate, I'd be interested in the collection. But
Yeah.
I'll have to sample it otherwise.
Okay.
They are humongous.
They are, and it's
A couple meters tall, most of them.
there's there can be a couple of crabs on these. Look at Irish Town. I don't see any here though. So here are the polyps.
There's one off to the right in the background if you want to scan that one too on the way up.
I see one coming out of a rock hole there, right there at that same level of the lasers.
That upper left knob just above the finger is the ISO 2
Yeah, I'm trying. I mean, I'm coming.
I think that one might be, oh yeah, there. That one might be shuttered.
Might be what?
Or iris. The very top left one is ISO. There you go. And then the bottom left one's focus.
Right.
And then the middle one is iris.
Oh, thank you. All
I
right.
wasn't backseat zoom. just so you
No,
know.
that's great. No, that's. Yeah. Okay. So you have a thought about this one, Steve?
I would for a collection of an associate, but if it's not an associate, I don't think it's worth the time.
Okay. I'll just go out here. I'm all the way up when we come out.
Yeah, do have some questions about, we have two kinds of curly Q bamboos like this. One where polyps are kind of biserial arranged, so like only polyps on sides opposite each other.
Yeah.
And then these which are a bit bushier. So
Yeah.
I think they're different. I have good collections from 2017 from both of them. But
We could still sample this one if you wanted to, but
I don't want to sample right now.
okay. All right. Then I think we can probably move on. Let me just get a snap on this, and then okay. All right, great. Thank you. Oh, that's good. Thank you, Ben.
You can change the name of it, but it's not good to do it on both my sheets as well.
Okay.
But
There's a couple of
I
them
need
here.
to know what it is, Jim, though, so that's what I see, that's.
Yeah. Happy to scan around if you want to any of the other ones.
You want to see the other ones too?
No, only if there's associates.
Oh.
Scan around the different colonies for associates if they occur in the area.
Yep.
Dark.
Hold on. That's me.
A little later. Yeah, right there.
Yeah. All right. I see an Sammy on the left over there.
Left.
I'm just saying what I see. Can you scan a little to the right though, in case there's one of those bamboo whips over there too?
Corals on that rock left as well.
Yeah. Did you turn the light off by any chance?
No, but what happens is like the other piece of the ice is so there.
Right. So it gets grainy because they got the ISO up.
Yeah,
Right?
just because it's so far out there.
The same thing we saw.
No.
Oh,
Yeah, let's
you
go.
know what it was?
What?
One of auto and both. And then there you go.
Now what? Yeah. That's a lot better. Another big
One
rock.
gets super out of line with the other, it'll get grainy in the far distance there.
Yeah, I was clearly there. So a lot of Anapsami on this big, this whole area, right?
Yeah.
I'm getting into them now. Depth is. I think we don't need to look at any of them, do you?
No,
I'm
no.
okay with not looking at them,
Keep
but
going,
we should keep going. So 1406 is the depth.
Yeah, it does lend some viability to the hypothesis that higher flow areas permit deeper occurrences because of greater surplus in their energy budget.
Yeah.
Whoa. I leave for literally 15 minutes and everything changes.
It's changed fast in the deep sea.
Yeah.
And also changed slowly.
That nap was also way too fast, but I'll tap out. Why don't you go get some lunch?
I'm impressed. I can't nap like that. I would never be able to do
I
this.
had hit the pillow and I was down in 5 seconds and then I went off.
Yeah, it was too fast.
What?
I just crashed.
Yeah.
I just went out like a.
Well, why don't
Light
I
bulb.
let one of you guys go eat and then I'll.
Well, he should probably go. And then
Yeah,
you
I'll
come back in.
probably eat and then
I'll log.
come back around 12.
Okay, another anemone.
We'll go. I've just been, Victor Gorgia, there's the first
No,
one.
what? Can we stop and look at that? Sorry, I missed it. Ben. Sorry, I missed that.
It's very sparse, so it's hitting the button every couple minutes.
Sounds good. There's probably an override on that. Can you tell me what kind of oak? Because I can't see it.
See, now if I try to iris down, I can't. Is there a reason why that is?
They're an auto. Nah, try now. Sorry about that.
No. It's okay. I don't mind auto. So this could be our first Victor Gorgia coral of the day. We saw quite a few of these yesterday. We even found a form that's more of the flat and bushy kind that I like. It's got a nice ophiuroid in it. Okay. This is the one that to me looks more like a plexaurid in stance in its pose. So I'm just going to image this one. I'm not doing any collections yet. There's typically a different ophiuroid species that lives in this Victor Gorgia colony or on it, as we found out in 2017 when we were south of here in the Phoenix Islands protected area. That's great shot. Okay, so we got that documented. I want to look at this crab quickly. I think it's a Eumenidae coming out of the rock. Large claws. You see the shiny eyes, and sort of the multiple rostra, if you will, on the head. All right, that's great. Anything else here?
Nope.
Still seeing that encrusting sponge on the side of the rocks. And now it's Sammy. Great job. Thank you. 1403. Victor Gorgia. That purple coral will sort of lose that color when it comes to the surface, and we handle it in the lab, and our hands get purple, and the water turns purple. And as many of those corals do, they change their color when they get on board the ship. A lot of the plexarids, if you've seen the yellow and the purple plexarids, will lose their color rapidly and turn brown to black after just a few hours off the bottom. It'll be a temperature response. Okay. Do you want to see this sponge?
Yeah, I'll take the images.
We're looking at sponge too, right in the center.
Sure.
Okay, I won't touch it, sorry.
Tim, do you want to eat first?
You can go ahead, Randy.
Are you sure?
Yeah, go ahead.
I'm fine.
Go ahead.
I'm really
You've
fine.
been up here quite a bit today.
All right.
Go ahead, because then Steve will take over.
Okay. The log. Expenses.
Nice job. So we're also looking at sponges on this cruise, and things that eat sponges and live on sponges. And here we can see there's lots of things inside we can't see. I see an ophiuroid arm. I see a claw up there at the top left. But here's an obvious conspicuous inhabitor, and that's another one of those Eumenidae crabs. Looks very much like the one we just imaged on the rock. So that's a glass sponge. Great. Perfect. Thank you.
Moving forward.
All right. So we're still about 80 meters away from waypoint 2, which should be 1391 meters, and we're at 1399, so it's 8 more meters over 80 meters upslope. I'm going to tilt up a little bit so we can see uphill more. Another dead sponge with lots of associates on it, lots of ophiuroids and hydroids, things like to colonize that. There's Anthomassus. It's a mushroom coral. Very small colony. So Ben, what was your favorite thing you've seen so far? I didn't get that.
Whale shark.
This was the whale shark on this cruise. Deb, what was yours? Let's take a look at this.
I was telling me to say whale shark, but the octopus are always my favorite. Sorry, it's not cool.
Which octopus was it? There was a Dumbo and there was
I
a.
only saw one, so I only
You
saw
didn't see
one.
the wall with 3 or 4 on it, the red.
Well, I saw that was sort of, I think, the moment where I was leaving the other day, where there was like 2 or 3 you saw it before
Yeah, that would have been in the afternoon.
change rotation. There was that one like city looking like it was brooding or something, kind of hunched down in a hole.
Thank you.
That's ginormous.
Just gonna check this for. Yeah, this is very tall here, but I'm just gonna check this for some associates. Does that look in focus? No. Are you in focus, Ben? I'm all the way up. I don't.
I can't see
see anything here. All right, that's great. Thank you. So corals are picking up as we climb up the side of this thing at 1393 meters. Oh, Ben, can we look at that sea star? Sea star. We stop for sea stars.
Obviously.
That's good.
Like wait for sea stars.
We break. Yeah, we need a bump. That's right. Thank you. We do. There's another Nalopsamia out there. We've seen quite a few of these now in the last 15 minutes that we hadn't seen on the dive previously. Ben is sitting us down. This is something different than what we have seen through the course of this cruise, I believe. I'll try to iris down. All the way in. It looks like a little 2, 3, 4, 5, 6, 7. It's a little octocoral that settled there too. Do you see that? On the right, a single polyp was colonized here. I'm gonna pop this quickly. That's crazy. But there we go. All right. It's possible that starfish will go there and just have a little snack. I know. Can't even get started. All right, here. That's more of those encrusting sponges, and we're going to take off from here. Let's see what we see. Nice job. Proceed, please. Thank you. I can tilt up too. These colonies of hard coral sclerotinian and Nalopsamia are getting larger as we go along here. See some smaller ones and so different size ones. That's not something we see very often. Another Anthomastus on the rock there. Another bamboo whip. Things are picking up. Nice flying.
Is that waypoint 2? Yeah, we like for the first few hours we were about 100 meters. I don't know.
Anyone see anything different? I'm seeing a field of Nalopsamia hard corals here. We often ride with the iris up pretty high so that we can see further into the distance and see corals that give us time to set up on them, to image them or collect them. Another mushroom coral. It's a nice shot. Big angular boulders here with the Nalopsamia on them. Not seeing a lot of animals living on the Nalopsamia. Rarely, if anything, maybe a crinoid or two. See one over there now. There's a euplatellid glass sponge, a large crinoid, some holdfasts that where corals used to be. Looks like it slopes off away from us, doesn't it? Ben's driving right down the line here to waypoint 2, which is about 30 meters away. So we expect a depression where we are right now to come down slope like we're doing. And just gonna tilt down a little bit. Then we're gonna come up. The waypoint 2 is actually a little bit up. Just having a watch change here of the pilots for our lunch. It is 1:15 where we are. We're on Samoan time. Good morning, J-Rock. How are you? Doing great. Came into this now up Sandy Field. Did pretty good. Seeing some bamboo whips. Saw one Victor Gorge. That was gorgeous.
Oh, that.
How long ago did it pass?
I heard 4 p.m. And I
Yeah,
heard
I mean, like,
something like
yeah,
that.
she's a
We're just changing out pilots here right now, but you can see there's a anemone and a crinoid on a bamboo coral. May still have some polyps on it. There's an Alpsamia with 2 kinds of crinoids on it. You see there on the rock in front of it. Just getting set up. Hey, Jared. Yeah. Oh, what is? Can we take a look at this, Jared? Own it. Own it. All right. 2247. Is it up there? Just the map? I don't see it. Oh, okay, yeah. So just showing people where we are right now. You see the track line we put together here with the blue dots are their waypoints. We are going from right to left, and we are the yellow icon ROV. And so you can see that we're going to be in a depression here for a minute. We're going to come up onto the darker ridge part. For the end of the dive, it's going to be exciting to see all the corals up on that top ridge. So I just want to give you a sense of where we are. We're just sort of the beginning part of this dive. We probably have another 6 to 7 hours to go, I would think. Okay. All right. You're still moving? So I think this is Acanthogorgia. Hang on here. You're edging up on this coral trail. It's all right. I'll get it.
Oh yeah,
Just so this one just caught my eye because I think it's Acanthogorgia. The family Acanthogorgidae. It's got really the polyps have sclerites in them that are really sharp and pointy and large. You can see these. Well, better in the lab, but you might be able to see some here. Deb, are you back to video?
Yeah.
Thank you.
It was a picture anyway.
Oh, great.
Back to the full sign.
Yeah, thank you. So we see, I'm going to call this Acanthogorgia. Come on in. You can see a crinoid here with its light-colored cirri grabbing onto the coral. You can see a shrimp in the background. That's a Caridian. Can't tell if it's got eggs or not. It's a little hot. Let's So, as you can see, you can see the polyps that have these straighter sclerites in them that are really pointy and sharp. There we go. I'm just going to hold that for a minute. This helps us diagnose who this is. See, some of the polyps on the left are pulled in, and then left to the crinoid. On the right, they're open and extended out in their feeding posture, with their pinnules outstretched, waiting for particles. And you can actually see particles that are flying by in the water that they may be able to catch. I'm going to go down here a little bit and see what else lives on here. Over on this side, I thought it might be an anemone on the left. Trying to focus to the back. There's the front, there's the polyps. All the way zoomed in. Yes, that's absolutely an anemone, but I think it's on the rock behind and not on the coral. So, I do see an ophiuroid arm down here. And then possibly another shrimp. We're just going to image the base here. There's a anemone on it. I'll go to the back here with focus. Like on the center axis here. Yeah. So, nice colony with lots of associates living on it. These corals are habitat foundation species that host a lot of species. More than 4,000 species worldwide are living on deep water corals, and that number is just really a postulate. There's more being described every day, every week. So that's fantastic. That to me, folks, is Acanthogorgia, which I don't think we saw yesterday here on Titov Seamount. All right, gonna hold that for frame grabs, and I think we're done. Great. Okay.
Ready to roll?
Ready to roll. Thank you for that tour
Let's do it.
of Acanthogorgia. You see some of those are now Sammy here with also crinoids on them. Not seeing it. Haven't seen an Biddle star yet on these now Sammy hard corals. I think you may have my. Oh yeah. You got it? Okay. Yeah. There you go. Thank you. These guys are, J-Rod's exceptional at getting the camera to do what we want it to do. Yeah. Oh, it's James.
That's right.
So we are flying more of these Anaup salmon colonies that were not in any part of our beginning of our dive on the way to waypoint 1, and now on the way to waypoint 2 we see them a lot more. Here's a bamboo coming up. I don't know if we had seen that before. It's going to be, is it branching or not? Wow, look at that. I'm going to let J-Rod just fly that one. There's no branches. There's no crab associates. That's just, looks like a bamboo base, old holdfast, where these two whips have coming out. It's fabulous. The colonies of the Anaup salmon are getting larger now and smaller, but see up here. Let's make sure that's not Magipora. Here, run a quick flyby of this to see if it's Magipora or not. Magipora is usually recognized by the zigzag pattern of the polyps, of the branches. Just gonna do a quick zoom here and there's one that's down on the floor. Oh, I think it's Magipora. You see the zigzags, especially on the living colony up here. Let's image this one up here, J-Rod. I'm going to back out and let you just set up. It's our first time we've seen this. These usually host a lot of associates, mostly crinoids, shrimp. The matrix of the the branching pattern really facilitates. I didn't even notice that.
Is this one of those ones
It-
that, like, was somebody hiking
Look
around
at
here?
that. It's-
We gotta look at the other side.
Alright,
So
yeah.
there's gotta be something.
Yeah, I
We
can't.
can go over there. Let's get the, can we get the ISO right or something? Somebody adjust, because I can't, I'm not getting it over here. I mean, this is the side, right? But
It is balancing on
balancing.
this.
come on, it's been weathered down to that, right?
Mhm.
That's crazy. The
No,
girl's
Yeah.
holding it up.
This is
Somebody
a
was
good
hiking
side
around
for,
here,
this is a
and
good
they said
side.
follow the trail.
This is a good side for imaging, yeah.
Alright.
Yeah, there we go, great, thank you. Yeah, it is, it's just perched there.
That's another one to touch.
Do you want me to,
Camera's all yours, we'll darken
all right,
it.
yeah, you can darken it, great. Should I zoom in on the rock first?
Yes.
Everybody. I know I'll have a couple minutes left before I have to go to watch change.
It looks like the coral, like there might have been a rock like right there before. You know what I mean? And it was probably sitting like that, and then that other rock fell out. The coral's holding it.
I mean, is that this right here? That's like
Yeah, the
that
holdfast.
thing that holdfast is holding it on?
That's what it looks like.
Because that's part of it right there. That's astounding.
Rock wall.
That's amazing.
I don't want to poke it.
Now I call
That
this
whole thing would come tumbling down.
I called this Madrepora before. I'm gonna look at it again. I think
On the other side, when the other rock
I still think it is, and I don't know where to start here. Okay, so let's just go up. This makes me feel like it's Madrepora up here in the corner. The branching pattern is totally zigzagged in the same plane. There's no turns on the branches. I'm gonna go in. I told you this is a great place for associates. Look at this. It's ridiculous. We have anemones, we have shrimp, we have amphipods in the top right corner, we have ophiacanthid ophiuroids. We've got more amphipods. There's a emunid crab in the back up here. I love that this is a very delicate colony, very brittle. But you see the live polyps at the top that are out feeding? Some are pulled in. It's a dense matrix in a plane. It's totally in a plane that set up to capture particles out of the current. Crinoids love the top of these things to get up off the seafloor and get out into the current. And there's another type of ophiuroid here, another Ophioplanthaka type. Just astounding live polyps all the way down to the base here. Usually there's a part of this kind of coral that's older and dead and like at the base. Looks like it's connected there. There's a base there. It kind of extends down to multiple bases. You see one here at the top far right, one a third of the way over.
Could it be multiple things that grew together?
It could be, but sometimes they lay down additional bases. But you may be right. Yeah, it could be multiple colonization events right here. So here's
The same kind of coral growing next to each other, will they fight each other? You know, like,
some do, but not the same species typically.
yeah, the same species,
Yeah,
they'll
they'll just
work together?
I think so. I mean, that's just anecdotal, just observational. Here's the older part of the colony here, right? You see some of the pieces. Some of the pieces on the floor are falling down already. There's a snail, I think, on the right. Gonna go have some lunch probably on this. This is just fabulous. Because here's the older part of the colony.
I do not want to kick you off because this is incredible.
This is
but
amazing.
If you'd like to eat some lunch,
I know, I'm gonna
offer
go.
is there.
But I thought I would just set you up on something nice, right?
I'd appreciate it, Timmy Shake.
Yeah, look at this.
Did you see the stack of rocks?
The what?
Stack of rocks?
No, I haven't
We
seen
got a name
anything.
for it. It's like a monument.
What are those things called?
There's a name for them, right?
What's it called?
A cairn?
No, when you're on a hiking trail and people have like...
Cairns.
Like, there's
How do you spell it?
What?
Spell it. C-A-R.
C-A-I-R-N.
Oh, like, okay. There's a little hermit crab. I mean, this is.
Cairn.
All right.
Am
Well,
I
this is.
crazy? Hang on,
this-
now I
this
gotta
is
Google it.
YouTube probably
More
already
hermit crabs up here.
mermaid rock
See,
art.
this is what we look for now.
You
See
wanna
this
talk about
crinoid here. Now you can see the cirri at the base. Right, I'm full in. but that's where those things come down and they abrade the coral, the soft corals, and create places for the barnacles and whatnot to come in. So just a wonderful, it's a Venus flytrap anemone up there. So many crinoids. Look how healthy they are looking on top of this thing. Man, this is great. This is a big house. This is a master apartment complex.
This is huge.
For
I'm
a
just
lot
getting
of,
a look through the sit
right?
cam. This is huge.
It is. Yeah. I
How
think
old do we think this thing is? Thousands of years.
I don't have a guess. I don't even have a guess. Probably. So,
What?
I think it's about as old as I am. So, somewhere in the second.
Almost Ben's age?
I can do it.
Almost Ben's age. There we go.
Guys.
Great madrepore. I know. I should have held it. I hold it right there. I did too.
It's cool, right?
Yeah.
Enjoy your life.
There's like 8 forms of this kind of coral zigzags, right? So it's just amazing. They figure out strategies for making themselves plainer for catching more food.
And imagine stronger too.
Yeah. Like a lattice like that would be stronger, but I feel like if you go up and touch that thing, it's gonna just, you know how brittle this is. So, nice amphipod up there.
That's just like a wine glass, really fragile, set it in the wind and it's not gonna blow.
I don't know how you thought of that. There's a hermit crab. All right. Oh, Randy doesn't want the chair yet. Good. You see the old piece in the back that's sort of fallen off now. This thing must be really old from when it really colonized. Right? Hundreds of years old. And then you see the one in front, they get planer on themselves too, and it's just falling off and falling down. So it was once a really larger colony. All right. And there's our rock. Can we come down on the light a little bit and get a
Come down a little bit more?
come down?
Yep, no, from it.
You mean me or him? I want to come up to the black more.
You wanna get it black?
Just like set it up here, Rod. You got it. And we-
With the lasers?
Is there any flesh on the outside of that structure, Tim, or is it just in the... Is there any flesh on the outside of that structure, or
What
is
do
it
you
just
mean,
in the polyp?
flesh?
Like the...
Yeah, when the polyp comes out?
Yeah. It looks like tubular. When the polyp comes out,
Yes.
which is kind of different to a lot of the...
Yeah, it is.
structures we see.
No, you're right. It is. All right. Go ahead, J-Rod. I should write now. 2302. 1385.
Good with that?
Good for that. That's the carrion rock. That was an amazing rock, isn't it? I still don't know if I really understand how it's doing it. If that hold fast is holding that rock
I
up.
mean,
But
with the building up there, and there was a pile of rocks there, and it was doing that down over here.
Yeah.
That big rock fell over from the
Let-
extra
left
weight.
that.
Yeah, see that big rock? Like
Yeah.
this, right here.
They're coming in.
Yeah, right.
You get it. Yeah.
So if this rock right here
Yeah.
used to be up there, and then the extra weight from this coral and the current
Oh.
would have brought it over, right?
Well, yeah. So I was looking here, and how is that? How is this rock isn't even touching those rocks?
I think this one used to be up there. See all the brown where it matches up?
I get it.
Yeah, like,
get-
right there.
Oh, right.
Yeah.
And it was up
this
there.
used to be up there
Right, but how is that standing up?
because it's on the hold pass. The hold pass has it.
Coral's holding it.
I got
What's
too
That's a
that?
big-
first.
got too
That's a
big-
first.
got too big for the current.
I haven't seen that. All right.
Crazy.
Thanks for the ride. That was great. Thank you, Rachel.
Sure. Yeah, he will. Wow. I'm finally getting a look at this thing.
We'll show you the other side of it is what really intrigued us because this side you can see that the hold pass is
There's, I
holding
think
it.
Yeah.
there's more coming too.
This side, you can't see that hold pass.
Hang on, I gotta get my head in the game. I got other stuff going on. Down. Head. Here. Okay. Thank you. That's what I do it. Really. Anemone, have you introduced yourself yet?
So look at it from this angle, like you can and tell the coral what's going on.
Hi everyone, my name is Hanamere. I'm an undergraduate student from the PRIMNM University of Hawaii, and I'm here on board with Dr. Randy Rochen, training your team, scientist team, which is a great experience for me. This is my first time.
It's so great to have you here. We have been working with Kiribati for many, many years, and to work with you on ship is even better. And so it's so fun. You know, I've spent a lot of time working in your country, and now here you are working in mine.
Yeah.
This is partnership. And that was an incredibly cool coral. My goodness, it was huge. And yeah, it looked like it was built on a rock cairn. So, you know, last time we were here in Pippa, actually, we found a bunch of corals like that, and they were evocative of vertical reefs. Like this idea of like these reefs just living on nothing, just this, and they would just slowly drop rubble over the millennia, like thousands of years. They would just sort of drop it and keep building themselves up and up and up, and that's what we're seeing here. Look, for that big one, and then all these little ones around it are they have the potential to turn into that, but they're young still.
Anemone. It's just like this rock full of salt, stony coral.
Mm-hmm.
Instead of driving down to my left, I spun around and grabbed down.
Yeah, it is a stony coral.
So the light up is
So
deeper water.
I am Randy Rogen. I am a faculty member at BU, Boston University. I'm the chief scientist of this expedition. I'm here with Anemone Tanaba, and here with J-Rod and Jared, our amazing ROV pilots, du jour, because you're all amazing, and James, who's our MT in the house, woohoo,
The most amazing.
who is managing our live stream and helping process our data, and helping to manage our imagery and our maps and
These are most
many
amazing
things.
MT from Wales.
I don't understand. Is there a joke on this one? What?
Oh, we were saying he's our most amazing MT from Wales.
He is our most amazing MC from Wales. All of our MTs are from slightly different places. So, all right, here we go.
Actually, everybody's from different places. I mean, him and Jimbo are the closest. Outside of that, everybody's.
Mm-hmm.
Her work.
How you doing? Oh, there we go. Good eyes. Oh, and an Iridogorgia. Look, your shrimp finding brought us to a coral.
Yeah.
Do you want to look at said one?
Nope. I want to follow the shrimp for a minute. It's an Iridogorgia, and there was no jellies on it. So, I'm good.
Swimming like
Those feelers work pretty good for you. You don't run into the rock like this.
Oh, there he goes.
Dead sponge.
the dead sponge.
Shoot that.
Yep, and a couple sea stars. There's another Iridogorgia. Let's take a quick snap zoom on it.
That's the same one.
Is it the same one? Never mind. We're good. It doesn't have any jellies. Or other associates, actually. Maybe we should note there's no associates. An anemone, a crinoid, a dead sponge. We're making some tracks here. Oh, let's take a... Oh, that's just an analopsamia with a crinoid on top. Kind of looked funny for a second.
It seems like it's pretty much like rocky, but I feel like corals
There are.
can found.
Yeah, well, there's more now than there were before. Yeah, I'm gonna take a look at this one. This little orange.
Which angle do you want?
This one.
What
Oh,
angle?
this way.
Okay.
Thank you. Wow. This is a whole new level of being spoiled. I say I'd like to look at this coral, and I get, what
It's
angle
flatter-
do you want?
It's flatter terrain.
You have options, and we have options.
You've been having me mountaineering for like the last 5
So
Okay.
unity.
Let's take a look. All these little urchins in the bottom, and then what do we have here? We have... What do we have here?
that a black hole?
I
Oh,
don't think
there's a
so.
shrimp.
It's not.
That looks black, but
I know, but I don't think it's a black coral. But nope, not Steve. I will check Slack, but I feel like I just saw this.
It's not the Swifty one.
It could be a Swiftia. That's literally what was in my head.
Now you're focused.
Thank you. Did they sample this something like this already?
I just had it.
Okay. All right. Hold on. Slick Slack. All right. Well, I think we'll just take good, careful imagery of it and note it, and we will ID it afterwards. But that's my putative ID is it's Swiftia. It could be wrong though. Okay. Thank you.
Not as branchy though.
No, for sure.
There's a shrimp out there, right?
I saw it. Yeah. Okay. We're gonna go look at. There was another coral just behind us, somewhere behind this one,
Behind this one or behind?
like that way. I think. It is not a black coral. An Antipotherian. Yeah, that's just Anaerida gorgia without a jelly on it. So we're gonna keep going. Thank you.
Very gorgeous. Sponge, crinoid.
Bridge, RV. Yeah, the new waypoint.
RV reach, I see object moving by accident. Is that the right place?
Stony coral.
Yep.
That will work.
Oh, look at you. You've got this all. You should start running the dive.
Jeez.
So Anemone is a first-year marine biology student at Brigham Young University in Hawaii, and you know I work with a lot of students, and most of them don't pick it up this quick. This is pretty great. Like within a couple of weeks, you're a deep sea biologist. You know what you're looking at. You can name it all. You know what you're doing. Oh, I want to look at this little red thing right there. I think it's an amphipod, but I just gotta check. And it's
There's a little shrimp.
I think so. I'm just taking a quick look.
No.
Yeah, it's like a little amphipod. It's not a shrimp, but it's a little crustacean.
Okay.
Okay, cool. Thank you.
You know why I think she's getting so good at it? From my observation.
What?
She's not afraid to get it wrong too.
It's true. She jumps right in there. I
Yeah.
love that. That's the way it should be.
Yeah.
Oh, and a really good friend of mine is on the chat. Hi, Felix. Hi, everybody. Hi, Kara. Hi. Aw. It's so fun. A friend of mine from college and grad school and his whole family are watching. So it's great to have you guys on. We are, where are we? Where are we? We're on Titov Seamount in the Pacific Remote Islands Marine National Monument, just south of the equator, which we crossed. Can we take a little look at this pink coral, please? We just crossed the equator, so we're just below it. We crossed on solstice, halfway through the trip, halfway through the year.
That's nice.
Crossed the equator. It was pretty cool. And what did we do? This is our last dive in the monument, although we'll be in the USEEZ for a couple of days still afterwards. There we go. Nice. It looks like a corallid, I think. I don't see any closed polyps, do you? That's sort of the diagnostic.
Closed polyps.
Yeah, they are all. Oh no, it could be a bubblegum coral.
Yeah.
I think it's a bubblegum coral. I call bubblegum. Wait, that's consensus in the room. Bubblegum coral.
Bubblicious.
Bubblicious.
I'm good with these
Bubblegum.
But that was beautiful. And what do we do? I'm so tired. I'm losing my train of thought. You gotta save me, huh? Sure. Okay, where was I? We're in the Pacific Islands, Pacific Islands Marine National. Oh goodness, PRIMNM, Pacific Remote Islands Marine National Monument, the Helen Baker Unit, in the USEEZ. We're on Titov Seamount, which is a GIOT, It's our second dive here. It's our last dive in the
It's a what?
monument. The Giat. It's a flat-topped mountain that used to be exposed at the surface. It was how it got flat-topped. The waves, and when it was exposed, and then it came down through subsidence, and now it's, I don't know, wherever we are, 1,100 meters below the surface of the sea. We can fly.
Fly, fly away.
Fly away
Set to the sky.
to the sky. And we, what are we doing here? We are looking for sponges and corals, as well as anything else that's interesting. We're looking for their associates, so organisms associated with corals and sponges. And we use the word associate because it doesn't denote a connotation of like being a symbiont or a predator or a commensal, because we don't know what a lot of these associates do. So Tim's really interested in that. I'm interested in a subset of these associates called corallivores or spongivores, things that eat them.
There's a sponge right there.
Ah, you got it. That's a hexactinellid glass sponge, and here's another Iridogorgia, which we'll do a zoom by to, and then look at the sponge. Am I good to zoom? I'm good to zoom. Oh, where there's a little associate. We're gonna take a look at this very little chirostylid crab.
Crab.
You're going to want to have a look at it?
Just a teeny tiny bit. Yes, I think so.
Just right over that.
So it's not a quick zoom, it's a real zoom. Sorry. And we're also looking at, when we're looking at coral predation, we're looking at the wounds made by predators and wound healing, and also asking about the wound recovery potential and how corals fight infection when they have an open wound. You know, we are surrounded, all of us, by microbes.
Did you see the little black thing on the coral right there?
Yeah, I did. I think it's a crinoid, but let's take a look at it.
Okay.
So first, let's look at this crab.
The
Woo!
crab.
Stick with me. Don't get too sick.
Hold on real quick.
Oh.
I'm not-
They're not. I'll zoom out. Sorry.
it's also-
And so we're doing experiments with live corals and sponges, looking at their response to different bacteria on the ship.
All right, get back here.
Yep.
It's a cute.
It's very cute. Let's see if we can get him in focus. We can see exactly how cute he is. Oof. Come on, Randy. Oh, so close.
I got that.
I got it. It's so little and it's so obscured.
And there's another little thing on the other side.
You have good eyes. All right, there we go. This shot's for Tim.
Sample Institute or
Huh?
frame crab?
No, it's not a sample. It's just a
Okay.
frame grab. Oh, it's very, very little.
That's some
It's
great-
extremely young. So now you can see the
that's some good
head, the
footage right
rostrum.
there.
Look at that! Wow, this is great footage. So Tim will be able to interpret what kind of squat lobster this is. It's some kind of chirostylid crab, and it's pretty interesting to see one so small and a coral so big. We usually see them adults with adults and little ones with little ones, but this was otherwise, I think, a crab-free coral, and this lucky little baby found a good home. Pretty good place to be. All right, where's that other thing that you saw?
I think it's just like a small X, like on the end of the.
I can back up to you. Get
Ah, right here. Little red thing.
Kind
That's just a little amphipod.
Yeah.
But yeah, they're cute.
right there.
Oh, that too. Okay.
The,
Yep.
Is that what you think?
Yep. Let's go look. Aha. Coral schmutz. Just some black or brown, like it looks like just some kind of stuff that got caught by this coral, but let's get a better look. Oh,
Run away from it.
it is running away from me. It doesn't want to be imaged. It says, no, get my good side. I think it's just some like little flock in the sediment. I don't think it's anything alive. But while we're here, always a good shot.
frame crab?
Focus on the crab right there.
Yeah, all right. Focus on the crab. Focus on the crab.
There you go.
There we go.
Okay, good shot.
Beautiful. Thank you very much. All right, didn't we see something else that was right here that we said we were going to look at? The sponge.
Which on that way out there?
The glass sponge.
Yeah, the glass sponge. We are interested in sponges too. Brian Kennedy, who's a PhD student at Boston University, PhD candidate, I should say, he's passed his qualifiers in my lab, is really interested in the distribution and abundance of and diversity of sponges. So we're trying to snap zoom on a bunch of those as well.
We'll try to get you
Sequel
right
corridor.
over so you can
Yeah.
get down there.
Yep. I was looking for the predatory jelly, but that was not what we saw. I promise, when we see it, I'll be jumping up and down. You'll know.
Did that rock that we got yesterday have that sponge on it? Did it come up okay?
That's a Steve question. I did not process that rock.
You got it. You got it. Look straight down.
I'm looking. Here we go. Ah, beautiful.
Ooh, it's beautiful.
Uh-huh.
I guess the spines was like too close to each other, like very.
Yeah, they are really close to each other, aren't they?
Mm.
Oh. Here we go. Focus. So these are called glass sponges, partially because of these little spicules. They are super ouchy. If you were to touch them, you'd get them all embedded in your fingers. Eesh. But they are gorgeous. This particular one probably has a pair of shrimp living inside.
That's a wedding one.
Yep, a wetting sponge. They get trapped in a pair of shrimps for life, but they probably don't feel trapped. They probably feel protected and lucky and like they've got a really good home. Okay. Whenever you're ready, let's fly. Look at all those urchins. So many. 4. So many. More now. Samia in the background.
Mm-hmm.
There's been a lot of that. So I'm looking at the chat, and I think a lot of you are asking about some of the collections, and those are great questions. We're happy to talk to you about them. We have been working around the clock, as you might have noted. We have dives all the time going. So in the lab these days, everyone's sort of processing what they process and then quickly tapping out and trying to get some rest or get ready for the next thing. And so we are, I did not process the rocks last night. Steve did. And I worked on an experiment. What did you do last night? Did you filter water? Or
No,
did you,
I did help out with Steve.
You helped out the rock? So do you know about the rock with the sponge on it? What happened to it?
I didn't.
Oh, you
Like,
didn't see
we
it.
didn't do it.
Oh, you didn't see that one? Okay. So there's a bunch of us, and we all sort of, many hands make light work. That's kind of the philosophy, where everybody helps. Oh, right. Hang on. Yep. Oh, good. Yep. Basically, that's whoever's around. One. Perfect.
Jimbo's gonna set up mount, give him a hand as well.
Sounds great. So you and I will do it, and see what's around. This is great. This looks like maybe it's a Calyptrophora or a Pericallia. This is a Crinoid. Enter Steve. What do you think? Caligorgia.
Oh,
Wait a
okay.
minute. How do you know? Teach me.
So the branches have alternating branchlets coming off,
Didn't even look at that. Yep.
and it's a primnoid. Has that texture, right?
Okay. Do you need a sample of this one?
I don't.
Okay. So this is a Caligorgia, and I totally see it now. So Steve, the minute he said it, it made absolute sense. I was
They have
looking at
these,
the shape of the branches.
Triangular branch tips, yeah.
Yeah, but actually, it's what Steve was looking at. His key character was looking at whether or not the specific splits were alternate or opposite, which is a botanical term that talks about whether or not the little branches are opposite from each other exactly, or alternating one and then the other in a zigzaggy way. And these are alternate, which
In fact,
is
I can probably give you a
Caligorgia.
species. There's one that was described from the Howland Baker unit, as well as from material in Pippa called Caligorgia crescentus. And as far as we know, it could be endemic to this part of the world. It's never been observed elsewhere.
That's really cool.
Yeah.
What? Yeah. What are you doing? Oh. Okay. Yeah. Steve, there's a question for you in the. You can drive if you want.
Sure. Yeah, whatever you want.
Yeah, we'll switch in a minute. There's a question for you that came on about what happened to the sponge on the rock yesterday, and I don't even know what.
Yeah, those are tough. We can't put the whole rock in preservative, right? So what we did was we scraped it off as gently as we could and preserved it in alcohol.
Did you do better than we did?
Yeah.
That stuff was hard to scrape, right?
It was, yeah. I didn't even look in the jar, honestly, because it was so much sediment. I didn't know if we could differentiate sediment from sponge. So yeah, I mean, it's tough. We'll have to look at it under a microscope to actually see what kind of structures are in there. But at least we have some tissue. They'll probably get a sequence or something.
That sounds great. Okay, we are good. Beautiful shots, though.
Yeah, great.
All right, I will let you take the pilot's chair here, and I'll log for you. But I'll be done at 2.
Can we move the map over a little bit to see the next waypoint? Thank you. This is a way longer dive track than I had thought. I thought we did something similar like to yesterday, or 2 kilometers, but so the waypoint 3 is kind of at the base of a steeper cliff, and waypoint 4 is kind of at the rim of the geo. So I guess we'll see what we can get to in the next few hours, something like that.
Are you want your usual fast forward?
Yeah, I mean, I think most of this stuff is pretty monotonous, so I would be happy going point 2 at least.
Currently, that's point 3 right there. So.
Even better. Okay. Yeah, I mean, if we come into something extraordinary, we'll bump it down to point 2, I guess, or something in that range. But the goal is we want to cover ground, right?
Let's cover some ground. We have a lot. We are doing dive after dive after dive after dive, so I feel no reason to rush and no reason to linger. You know what I mean? Let's just go at the pace that we go. Oh, Amanda back. And we will do what we do.
Yep.
Okay, I'm gonna leave you two, I think, if that's all right.
Yeah.
Just tap me if you want to make a call. And I will do a ship to shore and be back at around 2 if you need something.
Yep.
Okay.
Great.
Although I'll be in the lab too, so just basically call me if you need.
Sure.
Okay. Have fun.
Thank you.
Yeah.
So is the major speed constraint here the ship movement or the vehicle movement?
Currently, right now, because the ship's behind us, it's going to be us.
There is
fly out a little bit more.
anemone right there.
Yeah.
Hang on. So the ship to shore is at 2.
Okay.
Not at 1. So I'm going to go sleep.
Have a good night.
Thank you.
Nice boulders and cracked crust there. This is looking pretty similar to what we've been seeing. Can we look at this one over here with the coral with the branching and the sea star on it, brittle star on it? This might be a different species in Narella, and it has this kind of branching all from the same point. Just want to double check. Or it could be a Calyptrophora that has a branching pattern that's similar. This is a Primnoidae coral
Okay.
with an asteroschema or a brittle star.
Yeah, still.
Crinoid with the Predator star.
Mhm. So I think this is actually can I have a manual? Thanks. Yeah, thanks. So this is looks like Caltrophora, possibly Wyvali. It has branching all from similar points on the colony. It's really tough to tell this clade from Narella, since they have very similar characteristics, and you have to look at the different arrangement of the sclerites and the body wall of the polyp. But this is a really neat observation. I don't think I saw this dive at all, but this is one we can very easily, relatively easily identify from video. So this is great, and we have material from this from the Phoenix Islands in 2017.
Any
All right.
being closer for?
Nope, that was perfect. The one thing I've been curious about, if we see any of these big long bamboo stalks with a crab on it, we want to stop an image and maybe collect that. But if you see like something hanging on one of those, try and point it out though. Yeah, not many collections today. We got some rocks, we got some bizarre gelatinous creatures, as well as some Narella colony that was full of brittle stars, a bit deeper, but not too much, so it should make for light work in the lab today, which I think everyone will appreciate.
Mhm.
I wish we could fill it up. I don't know if there's enough stuff here to fill it up with some places. And there's not really a ton of diversity here for eDNA, but I may actually, if we get up into a little bit higher density, I might take an eDNA water sample. I think that it's diverse here, but I want to wait until we get up to the next bump, because I think we'll see much more up there. A lot of these deep sea corals, they are slow growing. That's something we say a lot, that they're feeding on marine snow, particulate matter in the water column, full of organic, gooey, juicy particles that clustered together as it sinks to the seafloor, made up of all different types of animals and phytoplankton. And then as it sinks down, this is the food. The corals capture these particles and ingest them. But because of this, one of the reasons, in addition to the cold temperatures in the deep sea, they grow at very slow rates, on the order of sometimes millimeters to at most maybe a centimeter per year, depending on depth, linear extension rates. Sure. Yep. Yeah, the biggest, the oldest coral that was found, I think, was found near in the Atlantic, aged somewhere around 4,000 years old. It was a black coral. To the best of my knowledge, that record still stands. But it's sometimes very difficult to age corals. Some of them can be aged using methods similar to aging trees, like tree rings. For example, bamboo corals are very well known for that. However, other ones like precious corals are very poorly datable because they live with a commensal polychaete that burrows in the skeleton, and it basically undermines the skeleton, not really allowing very good dating using concentric ring chronology. And those patterns are really muddled the deeper you go too, because the patterns of coral growth and coral skeleton dissolution are not always very clear. So as you move deeper into the ocean, you also increase the dissolution forces of some minerals, just because of the chemistry of the water, the temperature, pressure. All those effects result in dissolution of coral skeleton over time. And if the coral isn't laying down tissue to keep pace, it's actually probably not going to survive very long. But with greater food inputs, typically, if you have very nutritious food supply, for example, or a high amount of food supply to an area that corals can take advantage of, they may be able to compensate for that depth in their energetic balance. This is especially true of calcifying organisms like the stony corals. So our last sample number was 402. That puts us at 403 for this dive, or for this, whatever our next sample is. Can we take a look at this wispy little branchy thing here? And I'm going to try and go close on the crab.
What's that?
I'm going to try and go close on the crab.
Okay. I'll get you a
And
little bit.
that, yeah, I mean, I think that's okay. But whatever you can do just makes for better footage. Now you can see all that, like all of this stuff up here, this is all crust, right? And you can see where the rock is here. It's impressive.
Did you do the rock with the sponge on it last night?
I did take some of the sponge off the rock. Yeah, it was an encrusting sponge. I didn't really know anything about the sponge, so we had to preserve it the best we could. Usually encrusting sponges don't come off well from the things they're encrusting on. They're very, very thin. And so in this one, rather than preserving the rock, I had to scrape it off with a scalpel and preserve it in a tube. But that should allow us to get some DNA material out of it. It's like a chirostylid with a Chrysogorgia colony. We've been calling these Chrysogorgia bluto. They're very similar in morphology. And we have plenty of material from this cruise for these colonies, as well as from further south in the Phoenix Islands from 2017. Great, all set. Are lasers on?
No, they're all
Can you take them on? Thanks. Wow, where did that come from?
What is that?
That's a umbellula.
stuck on it.
Can you grab it? I'll take a volunteer. I will take that for sure. We can even go in a bio box. Oh my gosh.
Got stick.
Yeah, it's rigid. I mean, the stick part is rigid, right?
I It's just like a flower.
Looks like a rose, right?
Yeah.
Yeah.
Do you need a picture for the?
We'll get a picture in the arm. Yeah, we don't have a picture in situ because we don't know where it came from. This is definitely a soft sediment one, so it might have gotten hung up on the vehicle when we were set down in the mud. So we'll just give it a number 4003.
403.
Yeah. Biobox might be easier. That's a really springy stalk there.
Biobox.
You know.
Any additional text?
The Umbellula is the genus. Umbellula.
Umbellula.
Yeah. It's sea pen.
Sea pen.
We've seen quite a few of them so far, so this is great.
Wait.
You hit it? That's fine, yeah. Can you just hit the sample in situ button too when you have a second? When it's in focus.
I think I put it in Biobox C.
That's okay. We can put it in C. Yeah.
I
Can you
accidentally
put it in C? We already hit the button. If you need to change it, that's okay.
No, we couldn't see, we were just getting you images.
Sure. Yeah, that's good. That's great. Can you hit the sample in situ one more time? That's great. Okay, ready to go. I mean, they had to have come from recently. Yeah. So probably about this depth.
I didn't see one.
They've been around. They're mostly in the sandy patches, but yeah.
And you said put it in C?
See if you can get it. Yeah.
Because I accidentally submitted.
403. Yeah, it's a coral. For folks who are writing in, it's a coral, a sea pen. They live in soft sediment. They have a blob at the end, opposite the polyp end, which is pigmented red. The blob at the bottom end is called a peduncle, which it uses to anchor into the sediment. It's kind of like a balloon that it can blow up, so it has some sort of hold in the currents. Nicely done. You ever gone fishing and caught a sea pen before?
I don't know about a sea pen. Definitely fishing for stuff.
Is this a sea cucumber or different?
Yep, sea
Yeah.
cucumber. Yeah, off swimming. Get a good view of the mouth this time.
So cute.
Very graceful. I think this is Benthidites or something similar. I used to have a good handle on the sea cucumbers, but that part of my brain has now been replaced by corals again. I can adjust it if it's still in manual.
It's in auto right now.
Leave it in auto then. No, I don't need to change it unless you're having trouble focusing on it.
That's a good question. It's a pelagi cucumbers.
Yeah, this seems to be the dominant one at this depth on these seamounts. The deeper you go, the crazier the diversity of sea cucumbers. Even below 3,000 meters, you can start to get multiple different species, different colors, shapes, sizes. It's really stunning. So we're approaching waypoint 5, which is, I think, there's 6 total or 5? Wait, no, 3. Waypoint 3. Read that wrong. Yeah. So the goal is really just to get up to the rim of the seamount. Those are contingency targets beyond that. So if we have extra dive time there, we'll just do that. But this is a good pace. Yeah, so this is the umbellula here when it's full out.
Umbellula?
Umbellula, the thing we just collected from the basket.
Okay.
This is what it looks like normally.
But that one looks like a rose.
Yeah, the polyps, those long polyps, they kind of contract and they ball up a little bit.
Mm-hmm.
So this gives you some perspective of, feeding in a blob in this part of the ocean is not very advantageous. But if you can spread out your polyps and your pinnules, you got a better chance of capturing particles, which is your food source. Is this ridge gonna come? Are we gonna start coming up this ridge, or is this?
Feels like we're almost there at this next waypoint.
Okay.
We'll start coming up.
Yeah, the 3D looks a lot better than trying to navigate on this without contours is like.
Yeah.
A
There you go.
fish.
Halosaur. Probably in the genus Aldrovandia, maybe. You can kind of see right through its head, through its skull. Very little scaliness. I think so far we've seen both genera of Halosaur on this dive, which is actually kind of interesting. definitely one of the more common ones. These and Synaphobranchids are probably the most common fish at this depth. It's very similar morphologies, very long, streamlined, very energetically efficient body plans. The deeper you get, though, there's some really deep water halosars that are just really stunning. They look like dragons. Any closer zoom on the head?
Yeah, you're a manual focus, so go ahead and just zoom in.
Okay.
And then you're gonna have to adjust the focus because it's messing up on me.
Yep. I got it. There it is. Yeah. All right. That's good. Let's keep going. Current's pretty stagnant here, but it looks like it's coming on the nose. Maybe? And a
It's just
stagnant variable. We'll see what it looks like when we get up to on top of this ridge.
Yeah, pretty stagnant.
Studying skills.
Yep.
Victiculture.
Exactly. You're gonna know those like a pro. I haven't seen very many species. There's a couple of species I think in this area so far. I think we've sampled them both, so fortunately we have a good reference. Victiculture.
Oh, that's a sponge.
Yep, glass sponge. Some bamboo corals. Unbranched. We're maxed out on rocks for this dive by permit, so we're not going to be looking for any more rocks.
But this would be a good area, is what you're thinking.
Yeah. If I was going to sample a rock, which I'm not, I would take it here, but I'm not. Yeah, it's when we start getting to like 3 and 4 rocks, it starts getting difficult to tell them apart, and I have to go back to the sample report and look at the bottom photos.
You can always put rocks inside the bio boxes.
Yeah, we could. Yeah, good thought for the future.
There's some fish on the other side.
Yep, another halosar over there.
Mm-hmm. Anemone.
Primnoid coral.
Crinoid.
I can hear you.
The primnoid with the predator star.
Yes. Yep.
Most of the predator star loves to stay on the corals.
They do. Yeah. There's a couple of reasons why we think that is.
Yep, I can hear you.
Tunicate. More of these spidodiadematid long spine, deep sea long spine sea urchins. Yeah, so the brittle stars like the corals because it allows them to get up into the current a little bit and feed if they're planktivorous, and it gives them some substrate to grab onto. Most of them do have associates.
It allows them to go up?
It allows them to so they feed in the same stuff the coral feeds on, right? The plankton and the water column.
Okay.
So if they're higher up off the bottom, they can feed more easily than if they have to stay on the bottom and put their tentacles up or something like that. And it's much more nutritious too.
Conservation International, I think it's the University of Scotland,
Look at those all day, huh?
Oh yeah, plenty of dandelions out here.
No, the benthic, Siphonophore?
Yeah, yeah.
You
They-
call them dandelion?
It's kind of a common name, yeah, dandelion. I don't know who gave it to them. I don't really see the resemblance myself.
yeah, I can hear each other, there's a little, yeah, there's some feedback.
So another thing that I observe is like sometimes like the associate or whatever it is called, like the ones that they usually stayed on the coral, like sometimes the color they have is like exactly the same with the coral that they were living on.
Looks like a
Yep. Yeah. Could very well be something that is defensive, you know, some sort of protection. They're trying to either mimic the shape and texture of the coral, or they're trying to mimic the you know, some sort of color pattern that we're not seeing. I mean, we see the visible light, right? But we don't see all the other wavelengths of light that are
Yeah.
emitted down here. Well, sorry, I didn't know you were zoomed in there.
Oh, golf.
Just a habit.
He took off before I got back in time.
Cool, and then, Eric, were we here on camera, or? That's how we had visions, like,
For the sediment that we're seeing in this area, what is that bushy thing? Looks like an anemone, maybe?
Anemone or sea
Yeah.
cucumber?
I think
Anemone?
it's an anemone, yeah.
Okay.
Yep, anemone. The sediment we're seeing down here is really mostly this marine snow, but it could also be so the stuff on the white stuff on the ground is usually foraminiferan tests. These are planktonic organisms that form shells that get heavy enough and sink to the bottom. But yeah, the marine snow is slightly different. It's mostly organic matter, and it may be filled with other minerals, maybe silica, or maybe even bits of calcium carbonate or chitin from shelled crustaceans and things. One of the things, if we had all the resources, would be to deploy a sediment trap here. We don't really know how fast the sedimentation rate is here, but if we were to deploy a sediment trap, for example, that was suspended in the water column, it basically looks like a giant funnel that can collect a known quantity of water, and it saves and preserves all of the marine snow that falls into it, and it can give us a rough estimation of how much marine snow is falling in a given unit of time in a given volume of water. And those are quite big pieces of equipment. They require moorings to be deployed, anchor it to the ground. And then they also require someone to come out and pick them up after a while, since they kind of work on their own. So it's very involved, and yeah, exactly, yeah. And all the sediment traps that I know are associated with some projects are still out there, just they haven't been prioritized for collection. So those samples are kind of just sitting at the bottom of the ocean somewhere. Yeah, it's really hard to get a ship to come back out here twice. We've been fortunate to have 3 cruises since 2017 in this region, but still, it's very rare otherwise.
Were your wet balance reports?
Yes. Yeah, we are collections light today, and yesterday really, both by design but by circumstance. By circumstance meaning we are not really seeing as much worth of collection. We have sampled a lot of this stuff before, and we don't really see a need to disturb more colonies unless it's a specific need for experiment or some sort of unusual or novel association between a coral and an organism, or even a sponge and an organism. But it's also by design in that this is a marine national monument, and we are required to have permits to operate in this area, and it limits our collection capacity within this site, as well as Howland and Baker.
Is that a type of Brassicocha?
That looks like a plexaurid octocoral. Yeah. This is our last dive, though, in the Pacific Remote Islands Marine National Monument. We will still be in the U.S. territory, U.S. EEZ, exclusive economic zone, for the remainder of the week. And then we will be moving on, but we have probably another 4 to 5 more dives in the U.S. waters before we move on here. But we will be outside the monument, which allows us to sample a little bit more. Autofocus doesn't work on close-ups, not usually.
We hardly seen any corals, like diverse areas of corals.
Yeah, it's not what I was expecting, and I don't think it's. We were talking about this a bit earlier. I don't think it's anything to do with the food in this site. I don't think it's really anything to do with the substrate. It's all very very solid stuff. But I'm just thinking the currents may be slightly stagnant right in this area locally, and maybe since we're on the upcurrent side of the seamount, where currents are coming from the north and east primarily, maybe there's not as much of a larvae distribution in this area. Since typically larvae, when they're released from a coral or a sponge, they don't really have a lot of ability to move against the current. They kind of go with the current, so they'll either settle very close to where they were fertilized, or downstream of it. So that's what I'm thinking, is that this area might be devoid because it might not have that larval supply that helps propagate the population. But we'll be looking at that data along with some of these video when we get back to shore to try and better understand the spatial distribution patterns on the seamount. Yeah, go ahead. Can you hit the fish button?
Image button.
The fish button. Yeah, that one.
Is there any names on it?
No name for it. Just yeah. If you you can type in Helosar if you see it, but it's not necessary. We have a fish button just so that we can send all the fish photos to our colleagues ashore who study fishes, and just helps us find those records later on. We're not doing a lot of really in detailed analysis of the fish IDs, but that doesn't mean we are not interested in the data. Can you pop the lasers back on when you have a chance? Yeah, absolutely. That's fine. It's pretty barren here.
Hey, James, we put a different camera up than the 4K. We're gonna power cycle it. See if we can get that focus.
Thanks.
So we're just moving up this, moving up to the ridge, top of the ridge on Titov Seamount, which is the southernmost feature in the Pacific Remote Islands Marine National Monument, the Howland and Baker Unit. We're just at a depth of 1,343 meters. So far, we've seen a decent amount of diversity of corals on this dive, but the abundance has been relatively low. This is another plexaurid coming up here with a brittle star associate, 2 plexaurids it looks like. And we're getting back into these little encrusting sponges on the rocks here. We saw a lot of these yesterday. We attempted to sample them, and we got one whole one up, but it was pretty hard to see on the rock we brought it up on, and we did get some material for genetics analysis of it. Nice skeleton here. We're just going to do a quick white balance. Since we had to reboot the camera, it came up in auto white balance mode, which is a little off for the color temperature lights and the attenuation of light in the deep sea.
Okay, that's fine.
What's going on?
We're trying to white balance, but they rebooted the computer, or rebooted the camera. So white balancing is one of those things that your eye does it naturally on thing, but cameras need to compensate for the color wavelength of the lights and the attenuation of the water, and so we have to do a little bit extra step with cameras that you don't have to do naturally with your eye, and it matters a lot in the deep sea where you've got different color temperature lights. The lights can change, the water clarity can change. Even the temperature of the camera can cause the white balance to drift.
The pressure of the water is going through.
And the pressure. Another plexaurid coming down the left. I think we just fly by that one.
Did you want to go back to it?
No, I meant the one you're aiming to now.
Is it a Crinoid or
That is probably a primnode. Primnoid, yes.
just the stars?
Yep. Looks like two more plexaurids and a primnoid.
Stars. Thank you.
Sorry to tear away Brian from everyone. All right. This site has got me thinking a lot about doing a three-way comparison between the southeast ridge, southwest, and northeast.
I just really want good current information. I don't want to do that. I'm
Yeah.
all for it, but I really want a better measure of current.
Yeah.
There's a Crinoid. Wait.
Yeah, plexaurid. So we saw today a coral that I really wish I had.
Wait. Is this
You got it.
bamboo? Bubblegum? Wait. Mushroom.
Yes.
Mushroom,
Yes.
I was like, wait, it's not.
You got it. We saw the sparsely branched swiftia earlier, and I kicked myself because I was like probably nodding off or something, not asking for a collection or for that, but
Mushroom.
yeah. Mushroom.
Big to go.
Hey, did you see the one we preserved? It's like the second dive or third dive. It
No,
looks like
I
just like a
don't.
mushroom, like a button mushroom.
But is it a mushroom? No.
It's just the way it looks when the polyps are sucked in. It doesn't have that flowery appearance. It just looks like a mushroom cap with like a fleshy face.
Wait, there's another one, a type of chrysoculture. The one that it's just like very, what is that again?
Is it branched or?
A
I mean, like
branch. The one that we just saw, like with Dr. Ryan. Like the very pinky one, like it could like be a bubble gum, but not left.
Oh,
not
umbellula,
actually.
the sea pen?
No.
No, not the sea pen.
The sea pen, I guess I know. But sorry.
Like you have to point it out to me. I mean, we're seeing some of the same stuff here, so it's not
I think
uncommon to. Oh yeah, little juvenile slime star. Yeah, I think so.
slime star.
Yeah, when they're young, they're translucent like that.
That's so cute.
Yeah.
Made it to waypoint 4.
Waypoint 4, we're just knocking them off. waypoint 5 is kind of like we've reached our goal, and anything beyond that is extra.
Oh, look at this. Steve, you boogied.
It's a lot of similar stuff, and we've gotten all our rocks. You know, just to break it up a little bit, I may want to take a Niskin water sample. In this area is fine. Got 3 or 4 colonies in view. Yeah. Yeah, wherever. So we'll do sample 404 and pick any bottle.
Is it 404?
That's right. Yeah.
I just, we're trying to reduce the amount of mapping things, so I just did year to year, but you said that was quite efficient, I didn't want to go this way to go too close to that.
This the bottle 2 has a new plug in it.
Bottle 2?
Yeah, the screw cap, the screw thing fell out.
Oh, really?
This
Yeah.
file is a file.
Yeah, the last dive. I told Russ about it. He said he was gonna try and get it back up. One is fired at 18 meters.
3,200 kilograms.
We do have a photo for that.
And air. In water, negative 25 pounds.
Really, it's heavy. Aren't they
Oh,
usually
no, sorry,
slightly
positive.
lighter?
But,
It's like, huh?
What do we weigh?
Yeah.
We weigh negative.
Yeah, yeah.
Maybe that's the only thing next, and then there's the dive site, and those dive transects totally exist, and I don't know.
Oh, no, with the rocks.
Is
So do you have like going in on the sea star. So this sea star is something that was noted by Chris Ma as being in the genus Chiraster, and potentially very poorly known from this area. And so I wanted to get a second look at this because we saw one the other day that was kind of pale pinkish, and get some better zooms on it. Since we haven't seen very many of these in this dive, we're not going to be able to attempt a collection here, but nevertheless we can try and get some good imagery so we can satisfy our colleagues who study sea stars here, since they are some of the most prominent predators in coral. Yep, ready. Even better. Awesome. So you can see how spiny it is, and I'll share some of these photos with our colleagues to see if we can learn any more about this potential predator of corals and sponges, possibly. Can you hit beauty shot?
Yeah, sure.
Yeah.
There.
Got it. All right. And we'll get a size on it. Can you go lasers on for a second?
Lasers are on.
Lasers are on. Looks to be about 15 centimeters across armtip to armtip. All right. Ready, let's go. Thank you. Yeah, so we are abiding by the terms of our permit here in the monument, and the terms of our monument state, terms of our contract permit with the monument state, unless we see 10 of something, we're not really permitted a collection of that thing. So a lot of the things we're collecting are pieces of corals, for example. That's okay. We don't need to see 10 of something to see to collect pieces of coral, as long as we can leave some. But if we collect a whole of one thing, we have to see at least 10 of them, or confirm that there are 10 in the area, which is challenging, when we have only so much bottom time.
Mushroom.
Exactly.
Mushroom.
Yep.
Mushroom and the mestis. Oh, I kind of like remember the pronunciation of the coral. Is it like a caliculture?
Caligorgia, yeah. We saw that when we first sat down, right?
That's the one that I was
Yep. Yeah, it's really pretty, Caligorgia. And Anthoptylum, sea pen. It's a rock pen. You see how there's like a suction cup that uses to attach to the bottom?
Mm-hmm.
There's this might be in the, so it's in the same genus as a rock pen called Anthoptylum lithophyllum, which means rock lover, and it was described usually from the eastern Pacific, and it's not known exactly if we have the same species out here, but we have collected them in the past. So we do have specimens from this area. Interestingly, we're also seeing rock pens in this area that are panachulids.
Stand over here. Oh.
Eric, you are currently on mute.
Okay, now I'm talking down. No one run me. Can you hear me okay?
Noise.
Okay, yeah, fantastic. So this
Sorry about that. We're back now. We were just out of comms for a minute while we had a ship to shore interaction moving through. Yeah, yeah. So, so we sampled a couple of corals just now. We're starting to see some greater diversity, and one of the things we noticed was this white coral colony, which is kind of unusual. We're seeing a bunch of different color morphs, from yellow to reddish pink to white, and they're all plexaurids, but they may be different genera and may be different species even. So if this coral was red, I would call it something called Swiftia, which is an octocoral. But we also have some evidence from 2017 of a coral called white Swiftia. Not very creative in the naming, but still, you know, when something is that drastic of a color difference, it's really, you know, we really can't be sure if it's a different species or just a different color morph. So we're going to take a small piece of this one as well, just to confirm an ID. The one we collected, so we collected two corals previous to this. We collected a green plexaurid, which we don't have an ID for, and we've also collected a reddish pink Swiftia, sparsely branching. It's very clearly pink on our screen. But Swiftia can be all colors from white to red, like fire engine red, to really pale pink. So it's not clear whether this could belong to one or the other. It's one of those families that just drives us, you know, coral biologists really, you know, up a wall, because there's so much difference in phenotype and, you know, what's displayed by the coral, even amongst species, that we like to get a little piece of some of these things just to confirm our suspicions. Good. So this is going to go into looks like.
Quivered sand.
.07. There is a brittle star on there. Can you write in the text, also one brittle star? Very nice. And submit it. Yeah, that one's a bit of a mystery. Curious to look at that. I really haven't had any time to really microscope image any of these, but that is on the docket for the transit home. We have about 7 days transit back to Honolulu, where I'm going to try and finish my backlog of paperwork and data entry, and also try and image some of these corals using a microscope camera we have on board. Maybe they might find, some of those images might be able to find their way onto a blog post in the future. We'll see. See how fast I can get my act together there.
I never seen an anemone being on the coral before.
Yeah, there's quite a few of them. That one's really obvious. They're not always so obvious. Sometimes they're very small. They're like less than a centimeter.
Mm-hmm.
This is a really nice plexaurid rock.
It's also
Nematocarcinus. Yeah. Very nice outcroppy thing.
This is so beautiful with the crinoids.
Yep, lots of crinoids, lots of shrimp, ophiuroids. Really nice three-dimensionality here. Can I just zoom in on while you're flying by after you're done with this little pirouette on this larger fan here in the middle? Just gonna go in for a second. Yeah, that's perfect.
I'm gonna be flying you in closer.
No, that's perfect. I know I have what I need now, so I'm gonna pull out. That's Acanthogorgia there, and the rest of these are plexaurids. So you can see how they look a little bit different. That one looks a little bit bushier. So we've got Acanthogorgia next to plexaurids.
And that one off in the distance, the little guy, is the same as the one that's closer to it?
Yes, yeah. So these are all the same. The one, just that big one, from what I can tell, in the middle is different. Yeah.
So Plexigorgia.
Plexauridae, yeah. And also this one here, the A, the first bubble.
This
That one,
one?
yep.
Oh, I just came across with this one. I can look which.
That's all right. Yeah, a lot more diversity here. It's just that this is the moment we've all been waiting for. Like working up this side of the seamount, getting to the rim here is where you have. Seems like most of the diversity. I mean, these Globetrophora wyvili are everywhere, and that is actually one of the things I remember very much from the southwest ridge or the western
Seapan.
ridge. Yep. Is that these wyvili species are everywhere on these bumps, these crusty bumps. This is really nice. Just kind of zoom over this area.
Okay. I'm just gonna do this for the.
Oh, yeah,
Oh.
there you go.
Is this
Sure,
a
yeah, great. Yeah, go for it.
I see cucumber?
Yeah, it's one of those swimming sea cucumbers.
So cute.
Yeah, it's the these are all the legs, and then the mouth is in the middle. And you've got digestive tract there, and there are these tentacles around the mouth that it uses to feed. It's kind of like it's really cool if you watch them feed. They
Kind of
umbrella out, but they will also use those arms to. It's essentially if you were lying on your back, covered your hands in peanut butter, and just wiped them across your mouth. That's kind of how it feeds. Whatever. Yeah. Don't try that at home.
Cody tried that last night when he was making peanut butter jelly sandwich.
He's not lying. Stuff everywhere.
It's so cute though.
Yeah, I need to figure out more of these human analogies. Invertebrates are just weird body plans. Really cool, but just weird sometimes. We sneak a peek at this ragged-looking one on the far left.
There's a ragged one right here to the right as well. It's not as dramatic, but just
Yeah. Let's look at this one first, since it's it doesn't matter which side, left or right. Yeah. But there's a Pelagothuria caught in a coral. Now we have an interesting scenario here. So, can you identify this coral? You said it already several times.
Which one?
This one right in front of us?
The white swifter? Looks alright.
Do you remember the coral we first saw when we sat down with Randy?
Culture?
Exactly. Calogorgia.
Caliculture.
Yep. Yep. Yeah, so what's neat about this,
Wait, is it from wool? Is that a sea cucumber?
exactly, yeah.
What the heck?
What,
Yeah.
was it?
That's the same one.
It is the
No,
same
no,
one.
not the exact same one.
Yeah, yeah. Same species, but different individual. Yeah, this one just got,
Is
yeah,
the Caliculture started from
wandered
sea?
into the,
Okay.
Calogorgia starts with a C.
Okay,
Yeah.
Caliculture now.
Yeah.
And I see cucumber and then plenty of like
So,
Predatories.
yep, lots of these
And-
ophiocanthids. So what we're looking at here, it looks pretty ragged. It's probably been there for a little while, but the mouth is here in the middle of the sea cucumber. These are the arms, and it's kind of the head end, I guess, or the bell part of it is in the colony. It's really curious. Yeah, I mean, if this coral could possibly eat this thing, it would be a huge windfall. But it's not clear, you know, with these bigger critters, if they're actually able to consume this, or if this is something that would fall off eventually, or be eaten by other predators. It's kind of neat. But you know, if you were to touch this, it would fall away. That holothurian. I want to look at the skeleton here, because what we were seeing last night were these black bumps, which I don't see here, that were possibly evidence of some sort of infection or disease, or maybe just pitting.
Is that the one with the black dots? Last
Yeah,
night?
similar. So the skeleton type is exactly the same.
Mm-hmm.
It's just in a different state of decay.
I need to take a photo of this.
Let's zoom over here. There's a shrimp there.
A shrimp.
Lots of these amphipods, though. Lots of
Are you gonna
amphipods.
get a sample of that?
I'm not gonna get a sample of this unless I hear from someone else. But it is interesting to note, since we don't have Calogorgia from this site, but we have primnoids for the respiration experiment, and there's just a bunch of Asteroschema on here, which we have associated with Calogorgia. So I'm inclined to say probably no to a collection. Oh. Just gonna zoom over here, grab an image of this coral. So this one looks like Pericallia, just based on the arrangement of the polyps and branching patterns. All right. You autofocus me?
Go ahead. Microphone.
Okay. Thanks. I'm at the end of the zoom here, but I don't need to move over. I'm just gonna try and get an okay shot of this. Okay. Just wait a second. There's a couple other things. I'm just gonna scan around here one more minute.
Yeah, you can see like there's a lot of marine snow.
Right. So there's that.
Oh, mushroom.
Yeah, mushroom coral. there's that Swiftia button red now, so that's what I was talking about, the variable color. So I think that white one was actually also that color.
Mushroom endomestes. Like
I would like to sample something here.
Hi. Sorry about the lack of warning. Everything changed timing.
I know, I was so nervous. I didn't expect it.
You did a great job. Can I jump in for 1 second?
Yeah, I do want to collect this coral here. This is Pericallia, a new species we haven't seen so far, or that one is the same. So one of these two, maybe with an ophiuroid, would be useful. But otherwise than that, you can do whatever you want here.
Great. I just want to say hello to our friends from the Conservation International phone call, who they just had a live ship to shore. And we have friends from all over the Pacific who are joining us right now, live from Fiji, from New Zealand, the University of South Pacific, Kiribati, let's see, I don't know, Australia, Samoa. So it's incredible to have you all here. You're here at a perfect time because Anemone from Kiribati is actually working this shift, this watch, and so she's helping to narrate the dives along with Steve Askovich. So you've got two very expert now. Anemone is new to deep sea science, but now she's 3 weeks in and is basically a pro. And you have Steve, who's an expert coral taxonomist, here narrating the live dive. So if this is your first time with us, we're so happy to have you. And if you have any other questions, feel free to ask. You're looking at some amazing deep sea corals and crinoids right here live on the seafloor, 1,254.9 meters below the surface of the sea. I'm going to the lab.
All right.
See you guys later.
Welcome everyone. We hope you enjoy some deep sea TV here. We're going to set up for a collection now of this Pericallia colony that we have not yet observed at the site. So the goal here, it's kind of brambly, but we're going to try and get part of the ophiuroid, the brittle star, in the jaw with some branches. We don't need all of the ophiuroid. Yeah, that's fine. I can do it. We don't need all of it, like all the arm tips, but maybe getting something with the center disc.
Okay.
Yeah. And quiver size, no need to go big.
Alright, so you just let me know where you want to snip.
Yep. Can I go? Sorry, I didn't take photos. It was my fault while we were waiting.
You're a manual focus.
All right, I'll manual focus it. All right, we are good to sample.
Okay, where would you like it?
So I'm thinking, yeah, it's a tough one because it's kind of just a bunch here, but I want this part, the center. I don't need this, so something like here, cutting it here, but maybe lower.
Okay.
It's like right in the middle, so it's a tough one, but if you happen to not get the center disc, it's not the end of the world.
Oh, that's cool.
But anything you can grab, yeah. If you get it in the cutter, it should cut easily.
Yeah. You want that oak though, right?
Yeah, the center disc and you know,
Coral?
some of the arm, yeah, whatever's the easiest.
You want the coral too, right?
Yes. Couple branches is fine.
Right here.
Yeah. The arms might put up a fight,
Yeah.
but do what you can. If you pull them hard enough, it may they may unravel, which is fine. Oops.
Oh, well. Hold fast isn't great.
All right, let's go BioBox then. There's plenty here that we can collect a whole one. Let's go BioBox B with the whole thing. Bio box B.
Box. This one.
Yeah.
Any additional text?
Yeah, the name is Pericallia. I'll write it here.
Wasn't a very strong base.
Yeah, it's tough, right? You know, it's tough to judge. Sometimes they're really cemented, and sometimes they come off.
Nitro for. Okay.
With brittle star.
How many stars? Just one.
Just one.
Brittle star.
FK21. This is number 408.
8. Skin erosion, organization, international.
Lovely.
Don't do it, don't. Oh, I'm sorry. Oh. When somebody was on, I guess.
No, we'll do it later.
Oh, did some fall in box A?
No.
Okay. All right, so we're just trucking up this line, mostly just for video right now. We saw a couple of things here that are different, so it may start to get higher density as we move up, but we're just gonna straight up through the shallowest point.
I'll just limit this one.
Yes, yeah, that's fine.
I wanted to know like what type of like the crinoids.
The
Like is
crinoids
it
are
a coral as well, or it's a different species?
the crinoids are more closely related to sea stars.
Oh, okay.
So, sea stars, crinoids, sea urchins, sea cucumbers,
Sea urchins.
yep, and sea cucumbers.
How to spell them?
Urchin, Urchin. All of those belong to the same group, the echinoderms, which is basically just means spiny skin. Even though it doesn't seem like it, they all have some characteristics that make them similar. They have ossicles of calcium carbonate that are embedded in their tissues or make up this bony plates on their body. Brittle stars too. Forgot that group. Brittle stars are part of that too. The ophiuroids. Oh, there's some diversity here. Way more than enough to allocate to a collection of that colony. Haven't seen very many crabs here. Just a couple.
Sure.
We saw some homolids earlier, but it's mostly brittle stars. Yeah, so if we see any crabs on coral colonies, we're going to take a look at those in more detail. Sure. Yep. This is great.
They do make one bridge.
So we've got here a pretty mixed assemblage, mostly this yellow plexaurid octocoral, some Acanthogorgia mixed in, which are practically indistinguishable in certain cases. Calogorgia, Pericallia, saw Narella in the area, Calyptrophora in the area. So it's a pretty diverse little environment we're seeing. It's mostly just occurred in the last maybe 100 meters we've been traveling. It seems like we've gotten up and over the lip of the seamount, the crest, and started to come back into the flow a little bit. Can I do a quick snap zoom on that thing, that pink blob down there? Yeah. Alright, so we've got, what is that? Can you get a little bit closer?
You're rooming with Constance, right?
Yeah.
Alright. All set. Can you guys tick lasers back on when you have a chance? Yeah, this is a fantastic little assemblage. We've been seeing a lot of this on other sites, but not so much on this site until we've gotten right about here. But when Brian and I were here in 2017 on Okeanos Explorer, this is exactly what we were seeing on the direct opposite side of the seamount. So we're on the northeast side now, and we were seeing this assemblage on the southwest. But we didn't see so much of this yesterday. Similar in species, some of them, but it seems like, oh, there's a sea star on the left-hand side there. I'm gonna set down and image that well.
I'm sorry I pulled you out. I need a couple shots of you working.
Oh, okay. I can set up another.
Great.
So we're gonna zoom in on this corallivory going on here.
I feel like
So if I zoom in slow enough, it should autofocus all the way in, or is that? Does it? Yeah.
It doesn't want to do it right now.
Can I have it in manual,
It's in manual.
or is it in manual?
Yeah.
Whoops.
oh,
Yeah. So we've got some predation here. It looks like this coral.
And the top over there is the iris. You know that too.
Yep. Yeah, looks okay to me. So what we have is this sea star probably started somewhere here on the rock and started working its way up, eating, following the external most branches here.
Did you see the shrimp over there? Trying to stick up to the branch?
On top?
No, no,
Oh,
like right next to the.
it was swimming.
Mm-hmm, swimming.
Yeah, we get all kinds of little mysid, like shrimp in here. Yep. Very nice. I don't think this is going to be good enough to get an ID on the sea star, so we might rotate around a little bit and get it from the top side. But it's good to see this side. It doesn't look like the stomach's everted right now, but good to confirm that. Can we rotate around and try and get it from the other side?
Yeah, sure.
I know it's kind of facing downs, but whatever we can get is a little bit more helpful.
I'm confused with this type. Is this a Crinoid? Okay.
Exactly, yeah.
Okay. It looks so pink.
Yeah, they do have a pink coloration, so Norella and Callitrophora are pinkish usually, or sometimes orange, a very, very light orange, and then the Pericallia, which is the one we just collected, the big fan, they're often
Pink.
darker, yeah. Same colors, but darker, yeah. Cool. This should be good enough. Documentation. You can see it's got some tube feet wrapped around there. So this looks like Primnoid. Let's try and get. Did you hit the corallivory button?
Yeah.
Okay.
Submit.
Yeah.
There you go.
Okay. That looks good. Alright, we can move on. No, I don't want to sample it.
I know, guy.
It's the sea star that's might
gonna
be
sample?
Wait.
stopping us from sample it.
You gotta sit
Did you see that?
Oh, Dumbo?
Is that an
Good
octopus?
Good spotting.
Can you turn the lasers off so we can get some shots of that?
Great spot.
What is that?
Dumbo.
Dumbo octopus. Yeah,
Tumble.
you'll see why.
Got some big old ears.
Yeah, it looks like an octopus, but so big, like the legs.
Mm-hmm.
White pig. What the heck is this?
Bone and water. Looks like floated.
Dumbo octopus.
Dumbo octopus, fan favorite. Ship favorite as well. How could you not love a face like that? So this is a type of octopus that's pretty common at these depths. They fly using these fins that are modified. They almost look like ears, but it's really not too different from you know, how a squid might move, for example. We've seen a few of them so far on this expedition. They haven't been abundant, not as abundant as in 2017, which is very odd. We did see at Howland Island, I believe, an egg case. It might have been Baker. An egg case for one of these Dumbos. They lay their eggs in the coral colonies sometimes. And I'm really surprised we haven't seen very many at all. We saw a couple of them from my memory. But it is interesting to note, yeah, they do use the coral habitat, so we might see some if we look closely, but not yet.
Where's the link of this thing? How can I find the link to share to?
The link of
The
the live feed. It might be on. All right. Nice footage of the Dumbo. This one's we had one a few nights ago that was really like working for the camera. This one's just kind of like lazing along.
Sleeping upside down there.
Oh, Rita Gorgia too. No, it's all right. I was just noting it down since I haven't seen one in a couple hundred meters. All right, yeah, let's keep going up. We may pop a Niskin when we get a little bit further up, but I'll let you know when.
There's a fish. Mostly the corals right now.
Right. Yep.
Looks like Crinoid. I haven't seen the stony coral so far.
Just when the fun starts.
Where are we on the track?
We are right about at the rim of the seamount.
Okay.
So waypoint 5 was the rim. And then we're basically just going to hit waypoint 5 and then keep going kind of along the rim of the seamount for as long as we have bottom time.
Okay.
And I should check on that.
Yeah.
An ROV is never early nor late. She surfaces precisely. Precisely.
Tumble to this. And it's so interesting. There's a lot of names.
What?
So we got in the water about 03:30 last night.
Wait,
Plus.
did you just like being on ship by yourself
Huh?
last night?
Oh no, I mean, we basically
It was just for a couple. It was like 20 minutes. That he was-
Okay.
last. I mean, basically they got in the water. I think they got down 200 meters and then I left. So what time we got now? So 12 hours bottom time. I don't know what time we were
How
at
about
the
you,
bottom.
Princess?
I'm good. How are you?
Good.
Did you get some sleep?
Yeah.
Good.
16:08. So we have about an hour and a half left of bottom time. I think that's plenty of time. Yeah, we're in good shape.
So we're still shooting for like 4:30.
So
Is that-
4:30, 4 o'clock is our, around 4 o'clock is our tentative
Okay.
off bottom. And yeah, and then what, an hour probably to pull it, plus another 15-20 minutes to set up.
Sounds good.
Yep.
Okay, so it doesn't sound like we will be on another control shift.
I don't think so, yeah. but this is pretty spectacular. I will want to pop a Niskin here, I think, though, whenever you guys have a second.
Yeah, we're happy right now.
Niskin 2, 5 liter.
That's a fish.
Can you do
2.5 you said?
in situ?
Or 5?
A 5.
Number 2.
Do a sample in situ photo.
There you go.
All right, and then a sample storage. And this is going to be sample 409.
409. Water.
Yeah. Niskin 2
Skin to
is the target. Yep.
Barnhole.
Looks good. Okay. Did it? Wait. What did you put? You did. Okay.
Is that just for fun? That rotating?
No, it winds up the power and makes the swimming go faster.
Looks like we're the, tell me what you're about to learn something.
About to learn shit.
Do you need any additional text on this one?
No.
Oh, okay.
Yeah. You just put the depth in the additional text and say 1228
12.
1228 and then eDNA.
Fine.
Yeah. Perfect.
Tim was curious kind of what the collections have been and if we have associates on them.
Lots of Asteroschema, very few crabs on this dive. Yeah, lots of Asteroschema.
I think you saw the Slack about if you see a
Crabs, yeah.
yeah.
Yeah. Did he specifically say Chrysogorgia or does it matter?
I think the Chrysogorgia is good, but if you see him on something else, that's fine too.
Okay. Yeah, not many crabs. I think we saw one.
Yeah, I don't think we saw any during
while
our
ago.
morning shift. So
Can I tilt up a little bit?
Yeah, go for it. So you want me to increase speed a little bit then?
Yeah, you can go ahead with the speed increase.
Let me wind up the rope real quick. Do you want to go light 1 or light 3? Light 2 is actually broken.
Light speed 2 is broken.
See, they're all lower. Power up the flux capacitor.
Somebody get on that that pedal bike upstairs and just start pumping in the power.
Powered up. Beam me up, Scotty.
Yeah, once we hit waypoint 5, we'll kind of just cruise across the top. We have no real time constraints once we reach the top.
Okay, well, I'll go nice and slow because there's still lots of stuff.
There
here
is,
and then
yeah.
Once you get to that, 'cause I think you're right. I think this is like the steepest part of the whole section.
I think it
once
is.
you crest over, you're going to get like, I think it was like 50 meters out here.
Yeah.
Elevation gains.
Yeah. Can you hit the rubble button? I don't know what that is, but it looks like rubble.
I'm submitting.
Yeah.
this.
Oh yeah. That once it's done, yeah, hit that.
Rubble button.
Yeah. Should be a coral rubble button. Just hit submit too.
Mushroom. I'm so obsessed with the mushroom.
They're easy to spot. Like, it's such high contrast to the black crust that it's just.
The updated waypoint bridge.
What is a little green thing down there?
It's a sea cucumber.
Oh, okay.
Not one of the translucent ones, but yeah, brown.
Sea cucumber.
Elpidiid maybe.
I haven't seen the stony coral so far.
No, yeah, we saw some.
Before.
Yeah, I was up around 9 a.m. and there were some on the camera between 9 and 11ish. And then they disappeared. Yeah. Now we've got octocorals, which is kind of exactly what we had on the other side of the seamount. Precious coral. Hemicorallium. Yeah, this is
Is that a catfish?
where the fish on the. No, it looks like a cusk eel of some type.
That's right. Is that a sponge? The dead one?
Yeah, that does look like some sponge rubble. Some kind of big sponge. Haven't seen anything like that. Sponges, though, when they do dislodge, they tend to roll a very long way. Unless they get caught in a crevice. Maybe it's because they're lightweight. I don't know. No sea pens in these sandy patches anymore. As soon as I say that, we'll probably see a sea pen then. See how it goes.
Sponges. Crinoids makes the. Is that a crinoid or a sea star?
Crinoid.
Okay.
Yeah, that one's really dense. They like, going back up. I saw something. It's back to that fan that had a bunch of yellow things on it.
Just want to talk about it?
Yeah, I would just want to zoom in on the tips, just for a second. Let's see if these are crabs or crinoids. I couldn't tell them. Alright, the crinoids. Oh wait, hold on. Is that a shrimp? Yeah, it's a shrimp. Alright, no crabs. Just one shrimp and some brittle stars. Alright, keep on going. So, we are diving on a guyot today, as some folks have been mentioning, and guyots don't necessarily mean that the seamount was above water. It could have been just very close to the surface, or very briefly through geologic time had some wave exposure. But there are some characters that suggest that this seamount may have been above water at one point.
Oh, there's a little sea star.
Yep. I think that's another chiraster. Let me look at that bottle brush over there. That's floating right here. I want to take a closer look, see if there's crabs in it. Sure. Okay. I've got amphipods, but no crabs. Okay. Let's keep going. So, interestingly, the carbonate cap could still be here, but it might be covered with this black iron manganese crust, which is why we're not seeing rock that looks characteristically carbonate, like we would see on a more recent reef like Howland or Baker Island. So it could potentially be still carbonate somewhere under all this. But for the most part, the way this looks, it does seem to be more volcanic, basalt underlying this. But this is my impression as a biologist, having looked at these, working with some geologists, but not having geological training. This is really nice, habitat actually for corals. It's got to be a combination of current flow and just shallower depth. We've broken 1,200 meters now, 1,196 meters. Our end dive depth was somewhere around 1,150, so we've only got about 40 meters or so left to go in this vertical transect. See another Dumbo? Oh, I thought somebody said Dumbo over there.
No, apparently I've just missed one.
Oh yeah. Yeah.
I'm distressed.
It's the biggest one we've ever seen, Jim.
Oh,
Yeah. And it was probably a new species.
Right into the narwhal. Bright green.
Yeah.
I'm gonna kill you all.
There's a sea cucumber over
Then
there
dark
on
with the sun meeting stick.
the car.
You know where it's at?
Yes, I do.
What was that?
No, I mean I saw the sea cucumber in the car.
Sea cucumber, another one of those pink ones, yeah.
I am very repentant
Yeah.
J-Rod.
I was very impressed about how smooth some of these features are. The blue one?
Since it's been good,
That blue one?
Looks
there?
alright.
Yeah, it's a plexaurid. Can we
maybe I'll let it
take a look at this blue coral here? I'm pretty sure we sampled it already, but we sampled it yesterday as well. But I just want to double check because, like I said, color is one of these hyper variable characteristics of these corals. Wow, look at these. This terrain is phenomenal. It's like really nice smooth crust.
Let's
Yeah, this is really one of the impressive corals that that I've seen. Hyper variable color. From what I can tell from collecting several of these specimens, from green to blue to yellow to purple in some cases, and it's just extraordinary. It seems to be all one species, but all of these colors are of course just organic materials that are produced by the coral in the tissue, and it's no idea what's actually making some of these pigments, but it's definitely one of these really just like bizarre color morphs you see down in the deep ocean. All right, looks good. Yeah, I'm struggling to try and explain what's happening here geologically. I really don't know. We'll take a look at this one right there, that white bushy one. White bushy. Few anemones on these corals. You were mentioning about the anemones before. That one has an anemone, Victor Orgia.
and the sea star.
There's
Is it a sea
the
star?
brittle stars. This is an anemone right there.
Oh, okay.
Yeah, wrapped around. Sure.
Just checking in. How are you guys doing?
Great.
Great.
You don't even have to set down, I guess.
Pretty star.
It's just a quick zoom for confirmation. Oh. I think I'm all out of tilt. Oh, there it is. And so this is Thurella. It's a primnoid that, interestingly, we saw here quite a bit in 2017. This Thurella actually might be a very rare species, or what was thought to be a very rare species. We're still debating in the community whether it could be this rare species or if it's something that might just be a variation on something that's rather common. So stay tuned on that one, but we have many collections of it from across the equatorial Central Pacific, so I'm very curious to see how this goes. We're still looking at it, but stay tuned. I don't want to spoil the surprise. Yeah, some of these look like pillows, right? Some of them, I'm not sure, like big blocks. The one thing I haven't seen yet, which I thought we would see, were black corals, namely Bathypathes, which was quite abundant on this seamount back in 2017. None of them yet. It seems really patchy for some species. The typical cast of characters are here. These plexaurids, really abundant. Wow. Yeah, this does start to look like kind of spur and groove formations of a former reef or former carbonate cap. I don't know of any lava flows that would do something like this. Coral rubble. Can you hit the rubble button?
Yeah.
Let's follow the rubble this way. So this, again, from a biologist perspective, this might lend some support to the hypothesis that we are actually in like very ancient reef that's been encrusted in iron manganese crust, such that it doesn't look like it's carbonate anymore, but rather it looks like the surrounding crusty basalt that might be underlying it. Yeah. So all of this debris, right? Mm-hmm. All this coral rubble suggests that there's something up here in this direction that's resulting in a bunch of this reef material to fall down. And it's not insignificant either, it's quite substantial.
You're also getting a current above us that we're starting to feel.
Okay. I'm seeing more of these chiraster, and I'm keeping a mental tally in my head. Oh, that might be the 10th one. Can we set up for a sample of this sea star? Yep. So we hit the threshold now of numbers of observations of this particular sea star that would allow us to solicit a collection. I'm going to take good imagery of it first, which is a known coral predator. But to the best of my knowledge, we have not seen it on a coral yet. But it is in the presence of some denuded corals, so this is a potentially useful collection. Yep. Is there enough rack to get the, yeah. Perfect. This is chiraster. Very spiny. We've seen at least 9 of them, this being the 10th of the watch, and just in the past couple meters we've seen 3 in fact. So it's really noteworthy that they are in these high abundance areas. All right, let's so do an in situ Photoshop. All right, the lasers on it. Am I just not out far enough? Oh, there they are. Lasers on it. Or on the rock. There it is. In situ. Alright. Let's go for collection.
Ready for collection.
This is going to be sample 410.
410 by VG.
A suction could work too. So this is Chi-raster, and I'll spell it out for you when you have the window open. Cheirar. Yeah, that's it.
Chi-rest. That's just it.
Yep, that's it.
Is that the name of the species?
It's the genus name.
The genus.
Yeah. No species name on it yet. I think it was not known at the time. So we've been networking with our colleague Chris Ma at the Smithsonian Museum of Natural History, and he has given us some information about this through social media, that this might be an interesting collection for a coral predator analysis. I'm going to put a note that whoever touches that should probably not put their hand in there.
Poisonous?
No, I don't think so. I just think it's really spiny.
Sharp.
Quiver 9.
Quiver 9.
Beware of sea star. Awesome. You could hit it. It's Q9. Yep. And submit.
It's a sponge. Item.
Oh, there you are. All right. Just doing a check-in with our scientists on the ship here. There are about 1 more hour, a little bit more than 1 more hour left on the bottom. So we're just trying to strategize the use of our time. But this is the most tremendous diversity we've seen. I did take one recently, yeah. Can we look at this thing here?
I guess my laptop is running, so I need to open on that computer.
Okay, yeah, whatever you need to do. So, Tim, this is interesting. So this is the blue coral, right? But I think it's got zoanthids on it.
Yeah.
I've never seen this with the zoanthids. I've never seen a plexaurid take over by zoanthids in this way before.
Huh.
That's kind of neat.
Right.
Yeah, you've seen a lot more rosy and a lot more current over here, which makes sense.
Yeah.
Alright, go for it.
Yeah, this is kind of odd to me because we don't typically associate plexaurids with zoanthids. They
No.
seem to be resistant to that, and then when we do see it, it's like this. It's almost complete. There's some live.
It's just
you
a
know,
little bit of blue left.
Yeah.
There's some in the back, I think, right? There's something waving in the back. The
Yeah.
blue.
There's another plane back
Yeah,
there.
yeah, right. There's more back there. Interesting.
I don't have a particular interest in the zoanthids unless you do.
Only for ice tubs. You know?
Okay.
I think that's what we collected yesterday. Had the same pairing of the zoanthid with the blue plexaurid.
We sampled
we got the plexaurid. We didn't get the zoanthid.
Oh, okay. Well, let's do the zoanthid then. Do you want? How much do you want? Do you want sea star arm or no arm?
I'm not sure you can get it from here unless you come from the top.
Yeah,
You
it's,
mean? Yeah,
it's
you
a
can
big chunk. I mean,
You can come from the top.
yeah, we could do something. We're gonna sample. Maybe we can do something like
Yeah.
just a
Yeah.
couple inches in this area. These things mucus like crazy, so I don't want to really.
No, what else? Agreed.
Piece of the arm, right?
Yeah. Can I go in and take a photo first? Sorry.
Okay, I'm gonna give it a little wiggle real quick.
Sure.
Do I need to eat it?
Yep. Hit the in situ button. And then
Jump on that side real quick just to see something.
this is 411.
Okay. Hey, good, Steven.
All right. Good. We got some images while you were
Oh.
pushing it, so we're okay. We can go for our collection. Yeah, I mean, we just want a piece of an arm and some of this this zoanthid here.
Got so many that pulled in since they were just touched them.
Like right there.
That looks great to me.
Yeah.
Yeah.
I think there's some arm in there.
Yeah, you'll get the arm. Yep. Yeah, this one, I wouldn't trust how attached these are, so I might need to readjust and cut again. We really don't want to take the whole thing of this, only because it's just a pain in the butt to
Yeah.
dry them.
There you go. Good grab, there you go. There's something stuck in the.
There we go.
Nice. Precision.
We're gonna go.
Well, I just realized we don't have any crabs from Titov at all. But that's. So no Chrystic gorges really.
I mean, I saw
Saw a couple.
one, yeah, a bluto, and most of them had nothing.
Hmm.
Yeah,
Interesting.
yeah. I mean, like this, right? They're
Yeah.
Right. Not crazy huge. We've sampled that colony before, so I don't particularly need them for corals, but if we see any more with crabs, so keep an eye out. But that's the only place that I've seen a crab on Titov, not here. All right, good collection. You can submit it. It's a Zoanthid, Zoathid, and it went into quiver 1.5.
It's weird that there's no crab.
Yeah, great.
Yeah.
Even a unit on the seafloor would be really helpful.
Not a one. I haven't seen one. Yeah.
So I should have I've seen a couple on this dive. Right? There's one on the back of the Plexor. There was one on the Magipora. I should have. I thought there'd be more. I haven't been checking the Slack. I don't know if that's any action on the Snapchat. I have a phone call at 3, but I can come back if you want.
No, yeah, you're fine. We did. We saw some. There's a massive carpet of coral rubble leading in this direction, so that's kind of what we're following right now.
Okay, there's one there too, right? And that's,
Yeah, but no live stuff yet.
is that? Yeah, it's mostly Magipora.
Yeah, can we zoom on the debris? We did.
Debris?
Yep, debris.
There's a pink one up there. Is that a bubblegum?
I'm only seeing hemicorallium right now.
Okay.
Do you think crab doesn't live there because of some reasons?
I don't know. That's what I'm trying to figure out. I don't
Cool.
understand.
Sorry, I was distracted by a squirrel.
Because
I
basically
saw something
they're
down here.
really
Let's take a look at.
shit. You might come across with a crab.
Yeah.
What is that?
There's a little chrysa there without a crab.
What kind?
I don't know. It's pretty
It's maybe Pleurogorgia,
pretty wispy.
but it doesn't have a really well-defined helix, right?
No, it doesn't.
It's not a type of crazy
Are there two,
I mean,
are
it
there
could
two there?
it could be. There's two. Yeah, it could be. It's just
Yeah.
like it's not coiled enough. I'm not gonna collect it because I don't think it warrants a collection.
It's a funky little
But
branchy.
if we do see Pleurogorgia, I might be amenable to a collection, but I haven't seen any yet. And actually, we're quite shallow for its depth range, where we'd expect to see it.
There you go.
Yeah, Madrepora, I would say. Yeah. Yeah.
Wow.
Yeah. Some old stuff too. It's really discolored and weathered, and some partially manganese coated. Some look, those bamboo skeleton too in the distance.
There's some.
All right, well, that's enough of a teaser for me. Let's get going. Yeah, so we were saying, you know, I think we are on a carbonate cap because we've got spur and groove.
Yeah.
And yeah, these very rugose heads don't really remind me of pillows anymore. They're
No.
more like dissolution features. Yeah.
Those all must have fallen in place, right? I didn't see any holdfast.
Possibly, but this looks like a big sand channel. But
It
I mean,
does, but there's
where
these
where's
rocks
it?
in the way too.
Yeah, I haven't seen a single
Super interesting.
holdfast that suggests that there's been a large
Yeah.
Sclertinian there.
Right.
I mean, we're moving up into their depth range where you start to see these larger reefs. You know, 1,000,
Yeah.
maybe 1,100. Can you rotate back left? Yeah. A lot of these holothurians, this Pelagothuria,
Yeah.
caught in the colonies. Yeah, I mean, it's almost like you need two sets of eyes to look at all the colonies to see if they have crabs or not.
That's a lot. I know.
I did want to, I think I think we overshot it a bit. Yeah, back up just maybe a meter.
Heading southwest, the current's coming. The current's coming.
It's in the lower left corner, kind of on the
south.
this portion. Oh yeah.
I don't want to miss any of this. I gotta make a quick phone call. Back.
Oligothuria on an old coral branch. Everything's dead there. Well, there's a ophiuroid, I guess.
How much of that is our thruster, or is that versus current?
No, that's the current. Yeah.
I'm assuming most of that's the current, right?
Yeah. This is a lot of Thuarella up here.
Yeah.
Small bits of Thuarella. All right, let's go.
Awesome. All right, I'll be back.
Yeah, this is some of the highest densities we've seen. Not too dissimilar in community composition, though, from where we took our last eDNA Niskin. So I take another one in the just past waypoint 5 or so.
Okay.
But keep an eye out. Actually, might be a crab here. Can't go too far. Yeah, this bamboo coral looks like it has a crab on it. Of course, just when Tim leaves, that's when we see the crab. I think we'll do a collection here of the crab and the, of this, but let's do a quick image of it first. Come back out. Yep. Oh yeah, more of this. Yeah, lots of this Madrepora. Yeah.
Is that a crab?
Yep, it's a crab. We're gonna see if there's maybe more than one crab. Take a look, kind of scan the colony from the base up. Alright, can I have the manual focus as well?
Manual focus is on.
Yay! It's a bird.
Yeah, that's a crab, alright. Although it's moving down towards the base, so I was trying to make sure it's not too exposed. But I think I just see the one. Alright, let's do a collection.
Crab?
Yeah, oh, I mean, there's nothing, we don't have to keep this whole, so we can do like a snip and then a snip.
Okay.
So two snips and this in quiver, yeah.
So start at the top.
Do you want a picture for that?
Oh gosh, yes. Good thing. Thank you for reminding me. Gonna do a picture.
Should I eat it?
Hit it now? Yep.
Okay.
Alright, and then 1 second. Alright. Hit it now.
Yeah.
All right. We can go for collection.
Is that sample 412?
412. That's right. The coral is a bamboo coral with crab. Broken, just held on by tissue now. Is the right crab?
Yeah, that's a good
Okay.
crab.
It's clawing its way back down the colony. It's
Okay.
moved about a quarter of the way back down the colony since we just set down, forcing us to take more and more of the colony, I guess.
Yeah. I just wanted to check that you were getting it, so. good.
Mhm.
Good.
Yep.
Good job.
Tim was just in here.
Huh? Oh, it was, okay. I'm gonna go to the next. Do you need anything? You want me to take my cup?
Sure. Thank you.
Anybody need anything? You need a piece of paper? Oh. Let me just bring you an assortment. Did I give you the tray? I think we're out of Skittles.
Those didn't last long, did they?
Peanut M&Ms.
Peanut M&M's down there right now.
Did they grab it in? Lost it in? Wait, a second.
You know, if we could suction the crab off. I don't know, what's your track record with suctioning crabs off?
10 for 10.
Really?
Yeah.
Alright, well, you already have the arm there. Let's go for the coral. It'll stay on probably easier.
Yeah. If it jumps off, that suction. Does the human know? Watch out, this one. This one.
Before we lift off here, I think it'd be cool just to tilt up and just get a landscape like this. Or actually, maybe you could
Or do you fly it?
bump up a little bit more, so we kind of put this ledge in the middle.
I'll just do a full fly by myself.
Yeah, better. Yeah, it's just it's a nice landscape that we're low and the substrate's above us.
Or do you want me to land out? Do it.
No. I think that's fine. Just doing a flyby, but keeping low,
Is
so we
that
kind of keep
River
all the
16?
corals mid-frame. Yes, it's a good collection. Yep. Quiver 1-6, right?
Wait, River 6?
Quiver
Yes.
1-6,
Okay,
16.
sir. Your first class.
Oh,
M&M delivery.
I am spoiled. Yeah. Thank you. Can you hit the beauty shot button?
Sure.
Nice corals.
What's he been following?
Another one of those sea stars. They're everywhere. Chiraster. Very cool. Okay, let's keep going. Try and reacquire that rubble trail. Yeah. It's got bits and pieces all over. It's nothing like in the shallows, but it's enough. Yeah, we have our first bubblegum coral just passed.
Oh, did you see the big fish over there?
This one?
Yeah.
Yep. That's another cutthroat eel.
Quite huge.
The next dive will be somewhere in the next 12 hours, probably. We're going to do a quick CTD south of Titov Seamount here, and we're going to be doing a deeper dive on an unnamed seamount a bit south from here that Nautilus dove in 2019, and we're going to be doing a much deeper segment of newly explored depths that they didn't cover. So we're going to be diving in the 3,000 meter range, but probably launching sometime in 12 hours from now, approximately. Still seeing this rubble trail. I mean, tons of corals. So actually, it's way more impressive here on this side of the crest of the seamount than on the western side, which is where we were with the Okeanos Explorer. And yeah, this is pretty high density stuff here. Let's do a Niskin bottle here. Yeah. Right here. Can you hit sample in situ button?
Sure.
And we're gonna do a Niskin number 3. FK21-413.
Another 5 liter.
Another 5. Yep.
Going for 3. Fire in the hole.
1, 2. Is that 3? No. Does that seat well? It looked a little crooked. Yeah. That's 3? Oh yeah, okay. I just didn't see the right one. Good, thanks.
3. You got 4, 5, 6 left.
Yep.
What number is that? 1, 3?
That's Niskin 3, and the sample is 413.
Any additional tips?
High density and high diversity coral. Yeah. eDNA.
What was target depth again?
1150 actually, so even better. Nice. Okay, so we were just discussing end of dive plans. We're getting close to our off bottom time, and we're just looking at this Pleurocorallium here. Yeah, that's good. Exactly what I wanted. Nope, nope. That's it. Yep. It so with the coralliids, especially Pleurocorallium, they're solid. They have a solid axis, solid core to their skeleton, and if you throw a little thruster wash that way, you can usually differentiate Pleurocorallium or any coralliid from a bubblegum coral because they yeah, Bathypathes too. Okay. Because the coralliids won't move at all. They're solid skeleton, whereas the bubblegum corals will be a little bit flexible in the thruster wash. So that's.
Coral before, like it's not a bubblegum because I thought it was a bubblegum.
They look very similar. They're very closely related groups. We track a little bit closer to the waypoint 6, and then I think I'm gonna get in touch. Yeah, we've kind of reached the top here. This is exactly what the rim of this seamount we expected it to look like. We have some data from the other side, and it pretty much mirrors exactly what we saw on the western side of Titov when we were diving at the Okeanos Explorer in 2017. Yeah, I think we've saturated our dive here. But we'll just keep cruising along, towards waypoint 7, until we can get some info on off-bottom conditions. While we wait, take a look at this hamalid crab on this Calogorgia colony. Wrong type of crab, unfortunately.
Well, write something to yourself.
These ones aren't very pick-upable, unfortunately. They're pretty agile compared to the squat lobsters.
can't see them. It's camouflage.
Yeah, I don't know. Can you see crab there?
Oh.
I'm not sure.
Coral. I see coral.
I know. Fooled me. Alright. Good imagery. So, I mean, he's not really fooling anyone because they've got. So that homalid crab is on a Calagorgia colony, but it's actually holding Thuorella above its head with its rearmost appendages. So, I mean, A for effort, I guess. Just taking a scan through the area while we have a second. I think current's pretty good up here. Probably a reason why we're seeing a lot of this biomass. Oh, you got a crab there? Oh yeah, another homalid. Let me do a quick. Yeah, sure. Have a second. So how far do we got to waypoint 7? Something like 200 meters, yeah. I think we can cover that and yeah. Finish this zoom quickly and then just start trucking it. And this one's Thuorella coral with a homalid crab. Hydroid hat. Again, close but no cigar on the matching of the coral. I guess it really doesn't matter. But yeah, this is the land of the homalids for sure. We're not seeing many squat lobsters at all. Could be a depth thing. Alright, let's go. Let's book it.
Another one for you, Bruce. You're very much welcome.
Yep. Some pretty impressive, yeah. Pretty impressive plexaurids in this area. Blue, yellow, green. Swiftia colonies. Is it still in manual or auto? Oh, I guess it is manual. Can you put an auto before we cruise?
Oreo fish.
Where?
Up there to the top right.
Oh, yep, first one, I think. Those are usually pretty good indicators of these stony coral reef things. $11.27. Some Narella here. We've checked all the boxes for sampling goals, or that we can. Tim didn't say anything about Hamala. It's mostly just a.
Hey guys.
Almost. Yeah, I wonder how they track.
You can see everything.
Yeah. I don't know.
Oh, that's not good.
That is the stare of death. That
Whoa, bud. Whoa.
that was an attack. That was an attack. I thought we were gonna be goners there.
I thought it was enough.
So, if you guys can find me a cup coral in the next, between now and waypoint 7, that would check another box, but I haven't seen very many. Just going through my checklist here. Or a sponge.
There's a broken.
Oh, yeah, there's a big crab. Casion type crab. We saw these very closely associated with Howland and Baker Island sites. Yeah, there's crazy high density in this immediate area. It's pretty stunning.
First grab the
After we're done with the crab, I'd like to look at this right here, this single stalk.
Do you want to just snap in?
Snap on the crab. On the crab, yeah. Corallium, small bits, more of these. Size on him. Alright, that's good. It's going off camera. That single stalk, yeah. I mean, if we see any obvious sea star predation, that might be of interest too. But I'm not noticing any. Can I go in?
Go for it.
Unbranched bamboo coral. could be similar to the one we just sampled. No way of telling really yet if it's a branching bamboo coral or if it's unbranched. That's about it on that one. Actually, these white curlicues, black corals, Sticopathes. Yeah. Good capture on that. All right, let's keep going. I see that cup coral, but I'm not going to collect it.
Where?
Right here. I don't want it. It's one we already know pretty well. We have a couple of them on board already, and abiding by the rules of our permits, I'm not going to collect it unless we've seen 10 of them on this dive. Seeing a lot of this stony coral rubble, but no really large live stony corals.
Got about 150 meters until the lake opens up.
Yep.
Terrain. Roll up. Terrain. Someone said sample probably James is doing with me.
Can we go off in this direction a little bit? Just straight up there. Not finding this stony coral rubble source as much as I thought, but this is really old rubble. It's not like that stuff we were seeing in that overhang, that wall the other day. So there might not be much living here. It might just be something old that is not producing anymore. Might have gone extinct from this site. These are really nice features though. Yeah. There might have been stony corals here at one time, but they are no longer here. Oh yeah, a mullet crab there. Look at that, look at that, look at that thing. A scour mark in the crust from the coral. You just notice that? Seems unusual. Yeah. That looked to me like what it was. All set? Yeah, that's what I was looking at. I was gonna zoom in. So this is kind of like cool and like weird at the same time. This is yeah, I mean it looks exactly like the trace of that branch, right? Like it's just been the action of the current moving the coral has gouged part of the iron manganese crust, which is really unusual. I mean, it's not crazy hard stuff, but I wouldn't expect a coral to be able to do that, especially one that's really proteinaceous. The material is proteinaceous on the axis versus, you know, calcareous, but it's actually kind of neat. Could be something totally different too, but that's what I'm going with. That's cool. Hydroids and Hamalid crab.
2 crabs here back to back.
Yeah. Yep, 2 crabs. All right. Let's keep going. Just like, and we'll do a kind of a last sprint to waypoint 7. About 100 meters. Oreos really like the outside, but yeah, sometimes the I think most of the Oreos are like planktivorous, and they're eating things in the water column, and so it probably makes more sense for them to be in that higher flow. Oh yeah, look at those. Can we image those for Tim, since like those 2 crabs right there? Since we haven't really seen very many on this dive. I got two squat lobsters here. They might actually be two different genera. Europtychus and whatever this other one is, the spiny one. I'm not sure about. the details of what's going on here. We'll get some imagery of both.
It's a full equation. And then if you press this button right here, you'll see like the concepts happening. Yeah, it takes the algorithm and plugs it into the flight computer.
Very nice.
Oh yeah. Yeah, green sea only integrates with green sea. Yeah. It's proprietary tech.
All right. I think we're all set there. And yeah.
Please start left here. What are those?
Sponges. Some pretty dead looking sponges. Got some small chrysogorgia in there too. Yeah, haven't seen many of these sponges. I think this is the first one of the dive. Good to get some imagery on that. So we're about, go. You can go ahead. So thanks for everyone who was tuning in today for our dive at Titov Seamount. We were just discussing our end game plan for coming off bottom today. And it sounds like we will be ending the dive pretty soon. We are just finished our dive track we planned for today, about 2.2 kilometers in total. So we've kind of ended in this really nice, beautiful spot. Lots of corals, lots of sponges. And so we are going to definitely enjoy working up these samples when we get them back on board. But still, yeah, this terrain is way more impressive than the southwestern ridge, in my opinion. Having looked at 2 dives here, well, 3 dives, four dives now at Titov Seamount, this is one of the more impressive parts of the site, just in terms of density and diversity. But we are not seeing any more change in community. It's pretty much been like this. We're not changing much in depth. We actually we got a bit shallower than what we even thought the seamount was at. We're at 1,118, and I think our bathy was telling us 1150. So we're a bit shallower, and started seeing some fish that we hadn't seen in previous dives, like the Oreos, which are started this depth range and move shallower, which is neat.
You know I love the Oreos.
Yeah, we got attacked by one too.
You got attacked by one?
Yep.
What did you do?
Came up and took a chunk out of the lens. Yeah, I don't know.
Did you deserve it? I don't know.
I don't know. Yeah.
All right, so everybody's on board with this plan. So if you're, as soon as you're ready, you can. Yeah, we're all on board with this plan.
Ready?
We're ready.
All right, we're gonna come off bottom, and we'll see you in 12 hours or so.
No, not even.
Less.
Much less.
Less.
Great. Let me just think this through. We get off bottom. We're on the top at 5:30. CTD. We'll start it at 6:30 to 8:30, and then 4-hour transit, which is going to give us till 12:30. Then down 1:30. We'll be back in the water by 2:30 or 3 a.m.
Yeah, and probably on bottom sometime several hours later.
Right.
Yeah.
So, which puts us in at 5. So let's see. That's roughly. You're right. Never mind. Take it back.
Maybe
It's roughly 12
around
hours from now.
breakfast time. Yeah, I would say maybe around 5 to 7 a.m. tomorrow.
That sounds great.
Local time.
So thank you all for a really awesome dive. We are diving tomorrow, today, I don't know, later, 12 hours-ish from now. We will be on an unnamed, partially mapped seamount about halfway down the Tokelau Ridge, still in the U.S. EEZ, but outside of the Pacific Remote Islands Marine National Monument. Thank you again to the U.S. Fish and Wildlife Service, NOAA's Deep Sea Coral Research and Technology Program, Conservation International, the Schmidt Ocean Institute, and everybody on the ship and at home for helping us. Good night.